things like mussel beds, shell hash, things with a coarser grain size in the area surrounding it. So what we've done this morning is just kind of swung our dive track around. So we're going to be focusing on the purple line here, acquiring bottom at a depth of 1,168 meters, and then transiting up to the top of this feature to 1,130 meters. Two notes there, you know, that vertical change is only about 50 to 70 meters, depending on where we end up today. And the other thing is this feature, I had the exaggeration bumped here about 5 times just to pop it out. But if I bring it down to only one time exaggeration, so you have a relatively flat feature here, you can see because it is so flat, you know, the slopes here max out at about 15%. We'll be able to be very mobile on this feature with the vehicles today. So when the vehicles are using their scanning sonar 350 kilohertz, if they see some sort of anomalies in the sonars themselves, we can do some seep hunting today. And of course, our navigators have these seep targets to give us some bearing. With that, I will take questions or hand it back to Diva or Chuck.
Just one quick comment. Don't be disheartened that we have moved the dive site closer to these seep anomalies. It's likely that, which I'm sure you all know, that they co-occur with the coral colonies, which are going to be living on the orthogenic carbonates associated with them. And as you can see, we are still on one of the ridges that are the highest
highest topographic feature, bathymetric feature in the region. Yeah,
exactly.
And also point out the backscatter, the relatively bright backscatter. If I zoom out a little bit, we covered both areas last night. So if you look at the superficial backscatter here, where we're getting some of these brighter areas down on the southern feature, is quite similar to what they saw in the north here, where these previous dives were. So I would have pretty good hopes of potentially seeing some hard carbonate bottom, maybe some corals today. I think this, in any event, will give us our best chance.
And it's going to be really interesting to compare this southern area to what exists in the north that is quite spectacular, seeing how they compare to each other. Yeah. So with that, any questions, comments, concerns?
No, this all looks great to me, guys. Thanks very much for all of that overview. And I guess, could you... Mike, were you able to download that,
and if you're ready,
that anomaly package? I'm curious how anything that shows up in that might compare with what you're seeing in the backscatter.
I am, and Chris, I really appreciate you sending that. I'm still downloading it. The download failed last night, and then I restarted it this morning. So I think by this afternoon we'll at least have them for some later dives here, or in some sort of post-analysis we'll be able to compare them. It's a pretty large file, so I'm still working on it.
Yeah, but Chris, we were comparing that image you sent us.
Just from
you were comparing the screen grab you sent us last night, and we think the ridge that we moved on to now actually has more of the hard bottom anomalies than the ridge we had originally talked about, given that graphic you sent us.
Yeah, it looks like that's the case. So that's great. Thanks.
Okay, any other comments, questions, concerns? We have just touched down on bottom.
Just keep it center.
So if not...
I do have one quick question. Chris, I was reading the description you emailed, and I was interested to see that in that AT357 site, there was described a large Madrepora oculata reef structure in one area of the site, and I'm not completely familiar with the scleractinian buildups in the Gulf, but is that unusual? Because certainly we don't see as much of it. It's mostly Lophelia on the other side of Florida.
Yeah, we were safely on bottom. Let's set bail out to south.
Yeah, I think what I took from those descriptions was that the size of this aggregation in particular was the most unusual kind of feature about it. So there are a couple of images from that same Lophelia 2 report that I could send along that are quite striking, just as far as the size of these colonies.
Oh, very cool.
Yeah, this is Tom Harrigan.
Hey, Washley, just heads up. We're on bottom. We're just finishing setting up lighting, and we'll be ready to go in just a moment.
In the Lophelia 2 work in the Gulf of Mexico that have fairly large aggregations of Madrepora oculata. It doesn't appear to really build up reefs in the same way that Lophilia does, but it may be a contributor to those reefs.
Gotcha. Thanks very much, Tom and Chris.
Video, you want to go for a quick zoom as
anyone else?
we're setting up lighting?
Chris, could you forward that, what you sent to me as well?
Yep, I'd be happy to do it, and I'll when I do that, I'll send these images along as well.
Thank you.
Heather, do you want to say something about this morning's tweet chat, just so everyone's aware?
Oh, yeah, that'd be great. I was wondering whether this would be the right place. We, Office of Habitat Conservation and Fisheries has a live tweet chat starting at 10 a.m. Eastern that we've been promoting, and we're really excited with the dive still in Henderson Ridge, and we're looking forward to seeing lots of cool stuff. And we have photos that we've downloaded from past dives, so we'll be tweeting those out. If anybody wants to send some information to their communications people, we'd be happy to be in touch, and I can provide the Twitter page and hashtag that we're using. Thank you for helping us with this.
Yeah, Heather, would you mind emailing that over or putting it in the chat log just so that we can see? And if you do need anything from us during the dive and during that tweet chat, please do just put it in the chat or just jump on the call or anything.
Video, let me know if the lighting's good or if
all right, we'll do. Thank you.
Sounds good. Anyone else?
Yeah, this is Daniel. I just want to make a quick comment since you mentioned those habitat suitability models for deep sea corals. Right. So the highest suitability is actually in the area north of us, and those dives that you mentioned where we found those spectacular communities as part of the Lophilia 2 project. This area that we're diving on today has quite a bit lower suitability in the models. But this is also something that we're trying to test as part of this project, is to go both the areas of high and low suitability and see how well these models predict what we see. And that's kind of the first step to try to refine these models. These models kind of correlate environmental factors to observances or occurrences of corals. And it's an iterative process. All this information that we collect today will not just have management, but as well as scientific implications.
Understood, Daniel. And we do hope that this dive is going to be really useful for you guys.
Yeah, and this is Tom. One final note about the proposed or the habitat area of particular concern for the Gulf of Mexico Fishery Management Council. Council as a whole hasn't actually proposed this yet, but it is under consideration by the Council.
Okay, thank you for clarifying that.
Kind of a fine distinction, but.
And I just hit send with those images, so Tom should be in your email, and everyone else a couple of additional pretty pictures to look at.
Thank you very much.
Yeah, we got them already. Thank you. Okay, well, if that is all, then let's get exploring. Today's dive should be a really great one, so let's get on with it. Thank you very much, everyone, for joining us on the call, and yeah, hopefully we provide you with some good info today.
Once maybe both of you line up on 325.
Okay, thanks, everyone.
Yeah. Next move, you can just put Fernando, kind of like angle him. Okay. Put him center line behind me. So he's looking. You can move him north.
Okay.
I'm going to do a 30-meter move north.
Bridge,
okay, well, good morning, everyone, from the Okeanos Explorer.
Due north,
we are at a site known as Henderson Ridge Mid-South. We're currently just touched down at a depth of 1,180 meters, roughly. And the site we're going to be exploring today should have a variety of habitats. We first expect a number of potential coral sites, potentially coral gardens. This area has, well, is being considered for
you just want to comment on this
proposal
a lot of it.
To expand the Flower Garden Banks Marine National Sanctuary. And it's also being proposed as a habitat area of particular concern just north of here. The sites just north of here, we have seen past expeditions have seen extremely spectacular coral colonies, some which have corals which are 6 to 10 feet across. It really is meant to be one of the most beautiful and important coral colonies in the Gulf of Mexico. So our site is just south of that, and we're looking into whether there are any similarities between this area and the area north. That, of course, is going to have huge management implications. Additionally, in that site north of here, those corals were found on a carbonate pavement, likely orthogenic carbonates, and so those are associated with areas of hydrocarbon seepage and chemosynthetic areas. And in fact, mussel beds were found at those areas north of here. And so last night we did some mapping over the area, and we did find 4 bubble targets, bubble plumes in the area, which we have now adjusted our dive track to incorporate. So we're expecting today we are going to see not only incredible coral colonies, but also potentially some great mussel beds and other chemosynthetic inhabitants here.
All right, video and
so today my name is Diva Raymond, and I am one of the biology leads on board the ship. I'm a research fellow at the Natural History Museum in London, and my work tends to focus on chemosynthetic habitats such as cold seeps, as well as how humans are impacting the fauna down in the deep ocean.
And I'm Chuck Messing. I'm at Nova Southeastern University in the Fort Lauderdale area, and I'm an invertebrate zoologist with a specialization in crinoids, the sea lilies and feather stars, although I also work on deep coral communities. And just for your information, this field of what looks like rubble has got numerous, just a huge abundance of what appears to be a purple stoloniferan octocoral encrusting. You saw those purple polyps. And the bright white sea urchin is Gracilichinus affinis. And I'm wondering whether, Diva, whether we should go ahead and grab one of those, because this looks like the same thing we missed getting the other day and never saw again. That purple encruster should be pretty easy because it looks like they're encrusting on
quite small pieces of rock.
17 meters.
Well, let's focus ashore. Is this something that may be of interest?
Or is it something that folks have in their collections and know about already?
For zoom, just so we can see what we're looking at.
So just a quick segue, here on the Okeanos Explorer, our #1 priority is exploration. And so that means making as much ground as we can during the dive. But then secondary to that, we also want to be able to get the incredible footage that we are so well known for. And that means we have to work very closely with our videographers on board. And then additionally, we collect samples, not often, but we do collect them, up to 6 per dive, but those have to encompass both geological and biological samples. And so we have to weigh up very carefully whether our samples are something that is important. They have to be perhaps for biology, something that is rare, something that is unknown, or something that is particularly characteristic of the area.
So Daniel Wagner just mentioned that it looks like clavularia, and I've asked him whether it's worth collecting or not.
Did you say that was a stoloniferan?
Yes.
Okay.
And here's a little bacterial mat.
Yeah, Chuck, this is Daniel. So that looks very much like Clavularia
28 meters piles,
and that has been
almost done above
documented extensively throughout the Gulf.
Let that settle out.
Including at these depths.
Looks like you have moved.
I don't work on this
meters.
group, so I can't say if this is something unusual.
You're going off to the
but something
starboard.
very
Looks
similar.
like.
It's documented throughout the Gulf.
Okay. And so if it's been documented, do you think there have been collections made?
I I know of at least some collection through the Lophelia 2 project in their report. That species that I mentioned is documented in
All
right. Good. That's fine then.
Ship move is complete.
Thanks very much. We'll pass on it.
All right, Washley, so we're
Yeah, thank you, Dan.
pass on
It's
that
an
collection,
interesting collection of strange-shaped rubble, though.
We're passing on that collection. That's what I... Yes, we can do that now if it works for you. Great. Hi, everyone. This is the front row. We're all from the Global Foundation for Ocean Exploration, or GFOE. Sitting pilot with you this morning is Sean Henderson, and I'll let the rest of my coworkers introduce themselves, starting with my right.
Sitting as co-pilot, my name is Fernando Aragon.
And sitting to the pilot's left, sitting navigator is Levi Unima.
All the way on the right side on video is Bob Knott.
And in the back on video clipping is Emily Narrow. And we are all with the Global Foundation for Ocean Exploration.
So in addition to Chuck and I, who are sitting in the back row and are going to be narrating most of the dive today, you'll also be hearing those guys who just introduced themselves, and those are our ROV and video team, all from the Global Foundation for Ocean Exploration, as they said. But apart from that, we also are joined by a number of scientists ashore who provide us with a lot of really useful and fantastic information. And basically, we can't have more than 2 scientists on board the ship, and so we are just here as facilitators, in essence. We are using the expertise and the knowledge to communicate that to not only the public, but also to our pilots, so that we can achieve certain objectives for each dive. I'm not sure who we have on the chat today.
I saw, in addition to Daniel Wagner, there's Asako Matsumoto, and Bob Carney has logged on. And he had just mentioned that some of these curved rubble pieces look like they might be burrow casts. And Daniel has told us that that purple encrusting octocoral, which belongs called informally a stoloniferan, looks like the species Clavularia rudis. And we're zooming in on these odd curved concretions, solid pieces. They appear to be hollow, at least some of them. Small polyps on them. And when we pass the purple colonies, if we see any more, we should try to get a good close-up of that clavularia. I don't see any right here. Lots of that Gracilichinus. That's the first we've seen on this cruise. We saw another species earlier. But it always amazes me how very abruptly the fauna can change. So all of a sudden we are out of that area of the purple encrusting octocorals.
All right. I think
can we get a quick zoom on that fish that's hanging in the water column, please?
Yes, we can.
We are going to want to cut across this relatively quickly.
That's what I figured. Video, go for zoom.
That's an odd one.
So I'm inclined to say halosaur, given the weird movement that it keeps, 3/4 of its body pretty stable, and then just moves the latter quarter of the tail. But it's a weird posture. But that's great. Thank you, pilot.
Copy that. Video, come wide if you're ready. It's kind of out there. There's a lot in the water column today.
So Sean, I think we're going to want to cut the slope a little bit.
Yeah, no, I'm perfectly fine with that.
We're looking at like a 325.
Copy, which is, yeah.
That's interesting how the this rubble
totally
is confined to just the edge of
So
this.
thinking 50
here we
meters,
have
do you
a
want to do
flat
0.1 or
area to
0.2?
one side off the
Should we
port
just make some tracks at the
as
beginning?
we pan over,
Yeah.
and then to starboard
we
to
got
the
some
right,
good zooms when
it
they were
slopes
doing
away,
introductions on other
and there
items,
was
so.
less less in the way of
Wait
this
for
rubble.
Fish.
Wait for Josh to get settled in here.
And what do we have there? That's a squid.
Oh, squid.
Looks like the same short fin squid that we've seen before on a mission.
Go. He's out there.
All right, Josh, we're going to want to cheat the slope a little bit. We're looking at moving at 325. And we're going to go ahead and get a decent move in here to pick up some pace. But
oh, you're tugging me, Sean.
I'm coming back.
We're looking at 50 meters at 325.
Yeah, sounds good to me.
All right, I'm going to go ahead and call it in, and you guys will get adjusted here in a minute.
Yeah, so you gotta saddle the port here, Sean.
Yeah, I'm coming that way.
Yeah. Mess with your legs just a little bit.
Yeah, go for it.
Pilots, could you please tell me, relative to where we're looking now, which direction we will be heading on our track?
So to get to waypoint 2, we want to be going, and the ship's moving now.
325.
325, which is
Bridge
this
RV nav.
direction. The direction the RV is looking right
Go
now.
ahead, RV nav.
Yeah, we'd like to put in a move, please.
Go ahead.
We're going to do 50 meters. That's 50 meters,
Right
bearing
along-
325
right along that ridge
at
edge.
0
That's
decimal 2
great.
knots.
Yeah, about right
Copy,
here.
that is 50 meters,
That should
bearing
put us right to waypoint
325
2.
at decimal 2 knots.
That's correct.
Thank you.
Not a problem.
So we will be heading along the
RV nav
sort of
bridge,
edge of
move
this
initiated,
mound
50 meters,
or
bearing
this ridge,
325 at decimal 2
panning
knots.
back and forth as we go.
Can you do a quick zoom? Just anything?
And that looks like more water hyacinth washed down from the Mississippi.
Yeah.
We saw a lot of that yesterday.
Okay, move is
Little
initiated.
squat lobster there. See a number of these little squat lobsters. These are in a group
Copy that,
of crustaceans called the Galathioids. They're related to the hermit crabs, but they don't use a shell. And unlike true crabs, they have 3 pairs of walking legs instead of 4.
Okay.
We'll see more later, I'm sure. The last pair are small and tucked up underneath the carapace.
I haven't seen it too bad today.
And that little shrimp down below as we're passing looked like a glyphocrangon, and we'll see more of those, I'm sure.
Yeah, it looks pretty bare over here. All right, video, go for fish.
And that's a halosaur, yes. That duck bill
Take whatever you
and
want, video.
long, slender tail. And that's the texture of the bottom is very different from what we've seen before. There are no obvious little mounds and craters, as we've seen before. And instead of a continuous layer of tan phytodetritus, tan material that comes down from the plankton above, it's that tan layer is rather discontinuous.
I'm going to range down Sirius sonar to 50 meters, D2 at 30.
Copy that.
And a little bit more rubble here.
Video, go for zoom on fish. Might have seen this one already, but...
And correct me, but this looks like a synaphobranchid eel. And what was that? Was that a piece of human debris down lower left? There. That looks... oh no, these look like broken up... I don't know what those are. What do you think, Diva?
Perhaps they're related to what Bob had suggested. So some kind of burrow cast that's then been exposed in some way and broken up.
Okay. See if we got any comments about that.
A video can come wide. Nice. Lasers on them.
So the 2 points that you see are the lasers, the parallel scaling lasers, and they are always 10 centimeters apart, which is about 4 inches.
It's fine.
And more piles of those possible burrow casts. Strange how they've clustered like this in piles. Bob Carney says they may be just very firm mud, and he refers to them as broken burrow casts as well. That's really strange, though. I'm not completely unfamiliar with that sort of structure. And he also says, Bob also says that he's been told that bubble pipes can make a hard tube too, which makes sense why we should have so many of them and so tightly clustered like this. So they perhaps are remnants of bubble streams of methane from below. That's really interesting. Look at that one standing up right there. Yeah.
This one hasn't collapsed yet.
Yeah, this is Carolyn. That one we're looking at right now would make that what Bob said more plausible. We do see these sort of constructional features sometimes, kind of like a carbonate deposit inside of a tube, and then everything around it erodes. So maybe that is the right interpretation here.
Thanks very much. Yeah, it looks like a chimney.
Yeah, there are some places on Earth where we get very accurate, where I'm sure some of the people on the line have seen seafloor in some of those places.
Yeah, they're about 10 centimeters.
And there are some broad trackways in the sediment here. I haven't seen their producers yet. So this is interesting because this is completely unlike anything we've seen before on this expedition. Those little chimneys are really amazing. And more of the urchins. Another reflection of how patchy life in the deep sea can be. We started out on this the rubble field of possible burrow casts with abundant purple encrusting octocorals.
Video, go for zoom on fish.
And then they stopped. Haven't seen them since. And then we get into this area of relic bubble chimneys with broad trails. And this is a rat tail. And that looks, down below there is a that's a Galatheid, squat lobster with its claws up, sort of pale, almost the same color as the sediment.
It's a pretty good looking rat tail, as far as rat tails go.
Yeah, it's got those big puppy dog eyes.
Looks pretty clean.
All right, video come wide.
Fresh.
Kind of downslope on me.
I think that move is
quite a lot of fish in the area.
Yes, can we have a quick zoom on that? Yes, on that little, well, that's
Bridge.
an interesting fish.
What is that?
Go Bridge.
Wow.
Position
I've
move
not seen
completed.
that before.
Thanks, Bridge.
So that looks like a
I'll wait a moment
halosaur.
before putting in
But
another
those
one.
fins.
Yeah.
Let me just get this.
Well, Kevin told us that the macrurid is the first fish was Corophenoides Mexicanus. Maybe, Kevin, you can tell us what this is. That's something I have never seen before. Look at that weird, sort of a cross between a
tripod fish.
Yeah, a tripod fish, a rat tail, and a catfish with a little armored sea robin thrown in.
Does it have a parasite on it too, or is that just its gill?
Not sure.
Huh. So it's got those filaments. One is the first ray of the dorsal fin, one looks like the first ray of the pectoral fin, and one looks like the first ray of the pelvic fin.
Ooh, and Kevin, interestingly, says he's never seen this before. And neither have we on board.
Drifting away from it.
Wonderful. Look at that.
Should kill lasers.
So this is a great time to remind everyone who may be watching that this area of the Gulf of Mexico has never been explored before. So these images that you're seeing, no one on the planet has ever seen.
If the fish will do us the courtesy of turning round, we may be able to see Kevin spotted a chin barbel as well, little filament off the chin that some deep sea fishes and other fishes have.
Copy.
For sensing the perhaps prey in the sediment.
Oh, and Dan Wagner's also saying he's never seen a fish like this before.
Ken Sulak would be the one to know.
It's like a rebellion star.
So hopefully we're gonna send him a photo, and hopefully we'll be able to get back to you within the dive to give you an update as to what this is.
Now look at that tiny little chin barbel right below its mouth.
And then its gills are like iridescent.
Yeah.
That's a magnificent fish.
Video, do we have any more zoom?
Yeah, I was just talking about how good looking that other fish was, and then this one showed up.
Very interesting.
That's great, pilot. We're happy to move on when you are.
Copy that. All right, video, let's let it swim out of frame.
Oh.
And there it goes.
You can come wide, video.
That was awesome. What a great way to start this dive.
180 starboard.
Oh, and then we've got another nematostomid eel off to the left and a rat tail potentially above us or something.
Get untangled.
I mean, you got time if you want to look at stuff.
We pretty much got all those on the way up. So we're good.
Should I go ahead and initiate another move?
So just in the small area, we've seen, what, 5, 6 species of fish?
Yes. Yes.
I think we...
And this one has multiple elongated fin rays, too.
But that's definitely different to what we were just looking at.
Yes, yes. And the fin rays appear to be confined to the front of the pelvic fin, perhaps? Or the base of the pectoral fin, it looks like. Yeah, look at that.
Go close, tight on the head video.
All of these fishes, you will notice that we've seen, are either dark gray or pale gray. They're basically without pigments, characteristic of shallower water fishes. This is one of the characters of many, not all, but many bottom-associated deep-sea fishes. This one, of course, has very large eyes. We've seen a couple with large eyes. Others, particularly as you get into deeper water, tend to have reduced eyes. But most of them have this more or less eel-like or tadpole-like shape. There, of course, are exceptions in different families.
And a lot of the ones we've seen this morning have had that really exaggerated lateral line going down the body.
And that's between those extended fin rays and the lateral line, which senses vibrations. That's largely all they have, because in
50 meters,
the 1,170 some odd meters of depth we have, there is no light.
Roger that. Position move, range 50 meters, bearing 325, speed 0.2.
That's a good copy, Bridge.
There's a large chunk of rock.
So we're already seeing a good bit more exposed rock than we were yesterday. Tiny
There's
Lophelia?
a bit of, yes, looks like Lophelia, and the rock looks like there's a feather duster worm.
That looks like carbonate.
Yeah.
Quite. What was the term we learned yesterday from Carolyn? Vuggy.
Vuggy, yes.
Vuggy carbonate.
Vugs are little holes.
Position move initiated.
And those little fuzzy brown tree-like things hanging down are agglutinating foraminiferans. We've seen those all over the place before. I wonder if is this Vuggy or is this an old piece of coral? I can't quite tell.
Eric Cordes has just joined us. He's at Temple University, and he has worked extensively in this area, especially those very spectacular sites north of here. And he's actually suggesting that this is not Lophelia. It is, in fact, Solenosmelia. And he's making that judgment because the polyps are a little too tiny, as well as we're at a bit too deep a depth for Lophelia in this area. So I think Lophelia tend to be around 600-700 maybe. Whereas now we're at nearly 1,200 meters.
Thanks for that, Eric.
Yeah, thank you very much.
Video quick zoom on this dark shading, is that anything?
So is that a little anoxic sediment pile there around a burrow?
Yeah, looks like it.
Yeah, so we've already seen
come wide video.
Some areas that indicate to us that some of this area is pretty reduced, and the sediments are, as Chuck just said, are quite anoxic, and we can tell that because of that black color. So that burrow, that animal that's burrowing down the sediment there, has extruded all of this sediment, which usually would have been much lower down, much deeper, and as a result, we can see that it's pretty black.
Let's look at over there.
That looks like a, I don't know if it's active, but it looks like one of the blind lobster burrows with the openings at both ends, like a short tunnel. But I don't see any resident. And a bit of, off left, there was a bit of turtle grass, seagrass, shallow water seagrass washed into the deep sea. Now here we're getting into a bottom that has a somewhat different appearance. We're beginning to see mounds, low mounds, little low conical, low cones, and possibly some craters. Don't see any yet, but mounds suggesting that there may be more infauna here. And the darker cast to much of the bottom is more uniform. There's a burrow, another one of the two-ended burrows. And there's a shrimp above the lasers there that looks like Glyphocrangon, might be.
Video quick zoom. In burrow.
Oh, there's a coral.
Coral sort of washed into the burrow, hanging on for. That looks like the might be the same or similar. It's a paler color than the purple one we saw before. It looks like it has its own axis, unless it's overgrowing something.
I think it is overgrowing something. Considering how dead the skeleton looked, those polyps look pretty healthy.
And maybe a little scale worm up at the top that's red.
All right, video come on.
And could we have a quick snap zoom on that shrimp?
Go for it.
Take a quick look.
Yep.
Yeah, this looks like Glyphocrangon. It's a deep water bottom dwelling shrimp. There are several species, and it's got rather sculpted carapace with ridges running along it. The carapace is quite thick.
And it's also got these characteristic enormous eyes. So working with AUV images in the Central Pacific, these are one of the easiest animals to tell because of those very characteristic eyes and that very characteristic shape and color of the carapace. They may not be the same species as the Pacific, but definitely the same genus, Glyphocrangon. And yes, those are some very large, complex-looking eyes.
All right, video, you can come wide. There's a fish. It's too close. Video snaps in with fish. And we'll let him swim out.
I think we saw that one before. Not sure.
It looked like it had the black ring around the eye, which
Corophenoides, yes.
Kevin said was that the black ring around the eye is for Corophenoides Mexicanus.
Push forward.
Another one of those Glyphocrangon, some bacterial matte patches, which are the bright white areas just off to the left. And then in front of us, we've got some more of these very large urchins.
And a very different bottom.
So we're beginning to see a little bit more bacterial matte, some pitting, some black anoxic sediments. And sometimes what happens in very reduced areas that perhaps could be chemosynthetic in nature, so cold seeps, even though these animals that we're seeing here, these urchins, may not be chemosynthetic in nature, you do get a lot of animals which gravitate to these areas because of the enhanced productivity. There is this abundance of food, and as a result, you tend to see much more of these animals coming towards this oasis to feed in various respects.
And there's that weird, very weird fish that no one has put a name on yet for us.
Just take a moment.
With those long
nav bridge.
Fin rays.
Go bridge.
Just letting you know that the position move has been completed.
Yeah,
thank you.
Come wide.
Ship move completed.
Copy that, nav.
Should I go ahead and put another one in?
Yeah, I can keep pace.
All right.
Unless, yeah, I think
we'll do the same.
Great.
All right, bridge RV nav.
Go ahead.
We're going to do the same move. That's 50 meters, 50 meters, bearing 325 at 0.2 knots.
Roger that. Position move, 50 meters.
So there is a
bearing
little local
325,
relief
speed
here.
Decimal 2.
With
copy.
Low, broad, elevated areas and shallow depressions. Not sure what their relative organization is in terms of whether they are the elevated portions are mound-like or elongated. And there's some yellow.
I'm sorry, I missed the pre-call today. So did you guys discuss
position move initiated, range 50 meters, bearing 325, speed decimal 2.
Going on yellow real quick.
Sorry, would you repeat that, Carolyn?
I was just wondering whether you got back scatter data last night, and could you just summarize what that shows?
Yeah, so we did do collect some backscatter data last night, and the relatively brightest areas were in the region that we're going to be diving in. So along that feature, if you were to look at the dive plan, or if you were on the call this morning or last night, then you would have seen that big feature that we were intending to dive on. And so along the main ridge of it were the brightest areas. And that also correlated with 4 bubble targets, which we picked up last night as well.
Right.
So we're hoping there is going to be some
great.
exposed carbonate, as well as potential mussel beds, as well as corals. It should be a pretty bumper dive.
Okay, great. Thanks so much. Sorry to miss the call.
No, that's okay.
And here are more distinctive Bio-Turbation indications. And a lot of the phytodetritus, the organic material that's most likely come from surface waters, that's coloring much of the bottom a tan color, and then the paler sediment is dug up from below by infauna.
So the seafloor here is undulating in some respects. We can also see these little patches where it looks like there may be burrows or perhaps an area where methane gas is being extruded.
Oh, right, that's an option, certainly.
Being, yeah, gassing out of the seafloor there, and as a result, that's why we're yeah, so they could be areas where bubbles are escaping from the seafloor, and that's why we're seeing bacterial mats around these little areas.
It's a little sad. Come wide. Go for fish real quick.
That looks like a juvenile.
Port corner might be in the mud.
That's a little one. That's probably only
stirring up a little bit of dust.
A couple of inches long.
I think I saw something this way. Thanks.
Those glyphocrangon shrimp were rather common there for a while. There's another one.
So Kevin Rademacher has just given us a potential ID for that fish. The one with the long filaments on its fins. He said that potentially that's Gadamus longiphilis, which is a bathygatid. So thank you, Kevin. There are so many fish around here.
That's surprising. And right here you notice that the fishes and the shrimps were all sitting looking left, suggesting that that's the direction that the current over the sea floor is flowing. And more bacterial mats here as we come up to a topographic high. And that looks like a different kind of fish in the water column. Oops, swam away. There's quite a number of fishes here, much more than we've seen on previous dives per given area.
So dead ahead, there's something that looks a little strange. Perhaps more of those weird burrow casts that we were seeing before.
It looks like we're running along the edge, along this top of a very low ridge, I guess you could call it, as it looks like it slopes away to both the left and to the right. And there's certainly, hard cobbles here, white.
Video quick zoom.
So what tends to happen in these areas where we're seeing the bacterial mats is that hydrocarbons are seeping up through the sediments. So that'll be things like methane,
It's complete.
other hydrocarbon derivatives, sulfide-rich chemicals.
Should I keep it moving?
And those are then
Seems to be a good
used
pace.
by bacteria in this area. And those bacteria also use up the oxygen in the sediment when they are using these chemical compounds. And so that depletes all the oxygen, making this area anoxic. And so then chemical reactions from that anoxia make...
Go ahead.
We're going to do the same move.
Certain
50
metallic
meters.
compounds
That's 50 meters,
more
bearing
prominent than others. And I think in this case, the blackened sediment
Roger that.
usually is
Same move. Range
an
50
iron
meters,
compound.
bearing 325.
And so that's why we tend to see these areas with the blackened sediment. And then after it becomes anoxic, special bacteria move in that thrive in that habitat. And there's just this whole, chain reaction of bacterial succession, and at some point you'll end up with these beautiful white bacterial mats. Wow, that fish has its own little chute.
Nav bridge.
Go bridge.
Position move initiated. Range 50 meters, bearing 325, speed decimal 2.
All right, you can come wide.
Copy.
You saw him a couple times. Video quick zoom.
Is that a bivalve shells? Are those mussels or clams, Diva?
Those look like clams to me. I'm not sure which species, but one of the main inhabitants at cold seeps in the Gulf of Mexico can be these chemosynthetic clams.
Is it possible to get a zoom on that white material under the overhang, please?
Sure.
Right, straight on.
Is that what we want?
Yeah, yes. Can we just zoom a bit more?
Yeah, video.
That's max.
Copy that.
Is that helpful, Carolyn?
Yeah, I was just trying to obviously see whether that's hydrate, because we often see it under overhang because the methane concentration gets very high. I don't think we can really judge anything from that, but thanks for doing that. I appreciate it.
And I know yesterday, Carolyn, you were really excited for potentially collecting some of the orthogenic carbonate. Is that still the case today?
Yeah, it would be, and I just, in fact, my DGS colleagues are watching the video and are supposed to send me an email if they see something good. They had suggested already a place, but I think I encouraged them we should hold out for a little bit better. So we'll just wait. Thank you, though, for asking.
Okay, just let us know if there is something that suits what you guys potentially are looking for. Does heading along this shell hash take us towards waypoint 2? Can we get a zoom on that orange?
Yeah, well, actually, we're going to be moving in the direction that D2 is facing right now.
Okay.
On our way to waypoint 2.
Thank you.
That looks like the Octocoral swiftia, maybe torn off. It's not usually found strewn along the bottom like that. I can't tell if that up at the upper right is its holdfast or not on that bit of rock.
I think it looks broken off.
But this Octocoral is one of the species that is found on the hard grounds. And again, like the, for those who haven't been listening before, like the purple encrusting species that we saw at the beginning of the dive, this is also an octocoral because its individual polyps have tentacles and internal partitions numbering 8. And that is distinct from the stony corals, the sea anemones, and the black corals, which have their tentacles and internal partitions in multiples of 6. And this looks like, yes, that looks like possibly a Chrysogorgiid. Can we get a snap zoom on that?
Yeah, one sec.
Thank you.
Video whenever you're ready.
Okay, I'm ready.
Quick snap zoom.
Yep. On the shrimp or the coral?
Yes, that's a Chrysogorgiid.
That's a partial.
Perhaps the genus Chrysogorgia, and that is that Heterocarpus there, that shrimp on the side?
Does look like it.
And Chrysogorgia is another octocoral. It's got 2 different kinds of shrimp sheltering next to it, and it looks like it has a commensal shrimp tucked away in its branches there, a little bit of pink off to the left side of it. Thank you.
Copy that. Video when you're happy.
And Daniel Wagner has told us that that orange octocoral that we saw just a minute or 2 ago looks like Swiftia coriani, which has been previously recorded at 850 meters in the Gulf. And Kevin tells us that that large red shrimp is probably Heterocarpus ensifer.
So those shells we were seeing were probably vesicomyid shells, or potentially, I mean, we also find thiosauroids here, but the shape of the shell didn't really strike me as being very thiosaurid-like. Now that I've said that, someone's probably going to disagree with me. But those vesicomyids actually mine the sulfide compounds associated with the seepage, those chemical-rich fluid that's seeping out of the seafloor in this area, or potentially just within the upper sediments. They mine those sulfide compounds for nutrition, and they have internal bacteria or endosymbiotic bacteria that use those sulfides to create food for those animals. And when you collect those clams and bring them on board and cut them open, they actually stink of rotten eggs. Like, the entire lab will just smell absolutely horrific.
And what have we here? Is that a bamboo coral whip, perhaps?
All right, video go in.
It's partial.
I think so. Diva? Is that the Lepidisis?
Yeah, it could be Lepidisis caryophilia, which Scott France was saying yesterday was one of a couple species of bamboo coral that live within sediments.
So this is another octocoral, and in this case it's and it has a little anemone on it in that Venus flytrap posture. And these, for those who are unfamiliar, the bamboo corals, family Isididae, are also octocorals. Their polyps have 8 tentacles. And the central axis is long rods of calcium carbonate, solid fused skeletal pieces fused together, and they are joined by short, what are called nodes of dense black or brown or golden protein material. So when cleaned in particular, the skeleton looks like a length of bamboo, or at least superficially similar to a length of bamboo. And members of this family occur as these whips, and they also occur as branched colonies. They can be bushy, they can have long, slender branches, fair diversity.
Let's see.
And what are you?
Is it too close? Go for fish video? It's pretty close.
Any idea, Eva?
Looks new though.
Oh, that is this a near bottom fish or a mid-water fish?
Yeah.
Mid-water fish.
All right.
Is it it's not a cetamimid, is it? No.
All right.
All right, we're full wide.
Great.
Okay, that move's done, looks like. I think we'll adjust our course a bit.
Copy.
I think we just kind of start skirting along. Say like a 300.
Lauren Jackson suggested a melamphaid.
300, copy.
Yeah.
That's a midwater fish.
Nav bridge.
Go bridge.
Position move completed.
Thanks, bridge. We're going to go ahead and put another one in.
Okay, ready.
We're going to do another 50 meters. It's 50 meters, bearing 300 at 0.2 knots.
Roger that. Position move, range 50 meters, bearing 300, speed decimal 2.
Good copy.
Nav bridge.
Go bridge.
Position move initiated, range 50 meters, bearing 300, speed decimal 2.
Good copy, thanks.
Bob Carney has mentioned that the clam Calyptogena, which has that chemosynthetic metabolism, was first collected in 1888. And of course, back then, that aspect of its metabolism was not recognized at the time, but it probably represented the oldest collection of a seep community. But of course, that wasn't recognized until the early 1980s, cold seep.
It's a partial.
I had thought that Calyptogena was only in the Pacific. And in the Gulf we had Vesicamaya. But I could be very wrong with that one.
But in either case, whichever one it was.
I think there's been a lot of shuffling around in the group anyway. Taxonomically, they're pretty difficult to work with, I think.
Well.
And here we have what look like tube worms, perhaps.
No, that looks like a sea pen.
Yes, that does.
Go for zoom video.
And not sure what the other. Yes.
It's a partial.
Stand by. Let me settle. All right. Get a little more.
Certainly a possibility.
as well as a tiny cereanthid just in front of it, which would be one of those burrowing anemones that live in tubes within the sediment. We haven't been seeing that many sea pens during this expedition, which has been pretty surprising, given the amount of soft sediment we've seen.
Very interesting. The dark purple, I'm guessing, is the pharynx, the tube that runs into the interior of the polyp, like a sleeve from the mouth into the interior. And could we take a look, please? Could we take a look, please, at the branched one immediately to the left?
Copy that. Branched, immediate left.
I can come out if it would be
come
helpful.
partial. Here we go.
Okay.
All right, go back in.
Now, what are these? They look like worm tubes
partial.
of some sort.
Copy. Go ahead and take a little more. Go all the way in.
So they aren't branched. I guess they're just close together, and these may be the same. Some of the white ones look empty, and the pink ones may be occupied. There are tiny gooseneck barnacles, perhaps, on them. And if we can get to the tips, we might see a crown of tentacles. They may be the in the same family, the Ciboglinidae, as the tube worms. These are former members of the phylum Pogonophora, which used to be called the beard worms.
Sorry, this isn't
And
working
there's
very
one
well.
pulled back in its tube.
Should I come out?
Yeah.
That's a tough one.
Yeah.
If that's what they are. But they have the chemosynthetic metabolism as well.
All right.
Thank you.
No problem.
Yes, Sarah Cordes says they are definitely pogonophores. Now in the, he changed his, change it to frenulates, but I am guessing, and he can correct me, that they are all now in the family Ciboglinidae, now buried among the annelids, the segmented worms. They are very long, extending deeply into the sediment. And another, this looks like it has, it looks like it has a 3 openings to this burrow. Maybe that's an upscale version of the blind lobster burrow or something similar. 3 bedrooms with bath.
Can we get a quick zoom on that shell, please?
Yeah, copy that video snap zoom shell.
Right. It's a partial.
So that looks like a Bathymodeolus shell, which would be one of the dominant species found at chemosynthetic habitats here. And there are also 2 tiny little brittle stars just above the shell. Very tiny.
Yes, very tiny.
Are you happy with that, Watchley?
Yes, thank you.
Great. That video, if you're happy, come wide.
All right. Full wide.
And coming up on another one of those bamboo coral whips, 2 of them actually. Looks like 2, a short and a long. And this is interesting because my one previous deep water excursion into the Gulf of Mexico found the same sort of thing. We'd be running along a sediment bottom, and there'd be both tall and long and short colonies of bamboo coral whips. If that is indeed the same thing, I'm not sure. The polyps look withdrawn, but you can see the nodes there at intervals, a little darker points along the stalk. Polyps are all withdrawn. Not sure how healthy this one is.
All right, video come wide.
Full wide.
Great.
I think that move is complete.
Copy that.
$50 more meters.
Nav bridge.
Go bridge.
Position move completed.
Thank you. Yeah, we'll do 40 meters at 300, and that'll just...
Are we going to waypoint 2 or seep 1?
Well, our waypoint 2 is kind of arbitrary, just backed away from seep 1 because we're not sure exactly where.
Copy that, yeah.
I mean, this is an estimation from the multi-beam.
Yeah. Okay.
So, but we could head towards seep 1 if you're comfortable going there and basically just scan back and forth looking for bubbles. So
all sounds good to me. You can actually switch whenever you want to.
Do you think you could do a 285?
Yeah. That's upslope.
Yeah, let's adjust to 285 then.
Copy.
Bridge, RV nav.
Go ahead, Nav.
Another move, please. 50 meters, 50 meters, bearing 285 at 0.2 knots.
Roger that. Position move, range 50 meters, bearing 285.
So far we touched down in an area where there were potential stoloniferans, coating some very strangely shaped rocks, which could have been burrow extrusions. And then we've been moving over some pretty sedimented bottom, alternating with a couple areas
scanning
of
sonar at
these
50
chemosynthetic
meters
shells
D2.
that indicate past communities. We've seen a really high diversity of fish, including a couple which did stump and amaze us initially.
Is that bubble status sonar?
Yep.
Nav bridge.
Got it. Go bridge.
And just as an interesting aside, that family of tube worms, the Ciboglinidae, that used to be 2 separate phyla, actually, and are now just a family of annelid segmented worms, includes 32 different genera, very wide range of forms, from the giant tube worms of the hot vents called Riftia to small ones like this one. But interesting, some of the names that have been bestowed on these different genera, 2 of them are named after major former, now late, Russian deep-sea biologists, Bierstein and Zenkevich. So there's Biersteinia and Zenkevichia. And then there's one named after, well, I'll let you guess the name of the person who it's named after. It's called Bob Marlia.
Yeah, so that was actually some Portuguese colleagues, Ana Hilario and Maria Cunha, and they named it Bob Marlia because it has palps that come off the top of the head that look like dreadlocks. Yeah.
There's one called Polarsternium after the research vessel Polarstern. There's one called Oasisia because they form, many of these form oases in the deep sea. Just taxonomy is full of interesting little sidelights, tidbits.
Can we zoom on that strange rock just at the base, please? And then we're going to zoom on some of the corals in the area.
Sure, this one down here?
Yeah, the elongated one.
Yeah, go ahead, video.
All right. It's a partial.
Okay.
Perhaps the base of a dead coral or another one of those burrow molds. Casts, I should say, and a little Galatheid sheltering. I'm out of my comfort zone when it comes to that.
I think that's a pretty good guess. I got really excited because I've been looking at a lot of pictures of ferromanganese encrusted whale skulls, many of which are beaked whales, and they have very elongated rostrums. And so that's what I saw there. But no, I think Chuck's guess is a lot better. Can we have a look at some of these pretty big corals in this area?
Sure.
Well, big compared to what we've been seeing so far. So dead ahead, those little bushes.
Okay, what about the one
as well as
coming into center?
I think those are anemones. We can just pass those by.
Okay, all right. We'll push ahead.
And then over to the left, there's that big area as well, please.
Okay, I'll move over
so
to
this
the
is
left.
a great time to point out that NOAA is actually hosting a tweet chat that is happening live right now.
Great.
If you are on Twitter, I do encourage you to go over there and take a look. You can find that chat at the hashtag DeepSeaTV17 or look up at NOAA Habitat.
But I think you're right.
And so they're talking about deep sea corals over there. And that's something that we're really hoping to see today. A lot of deep sea corals and hopefully some spectacular ones.
Face you, but we're still moving.
All deep sea corals are spectacular, but
all right, you want to zoom in?
Sure.
Yeah, I think that is line. What do you think?
Yeah, that's fine.
Yeah, so that tweet chat is from the Office of Habitat Conservation and NOAA Fisheries. And I know there's some really interesting discussion happening over there.
Let's come out.
Do these look like there's Swiftia and a Chrysogorgiid there?
Yeah, so definitely the light pink bush-like one is a Chrysogorgiid. Then we've also got the, was it the red or the orange that was Swiftia-like?
I think it's the red one at lower left. And these guys also look like Swiftia.
Yeah, and I think one of the Swiftias is Swiftia coriani.
Right.
Then we've got a black coral, potentially a Bathypathes, right in the foreground, that light pink one. Then some more of those purple stoloniferans, which I think...
Clavularia.
There we go. And then off to the right, I saw a yellow coral, which we haven't seen yet for this dive.
Come out a bit. Try to find the yellow one. There it is.
Oh, there it is. Yeah.
Okay. Go ahead and go in.
And Daniel Wagner says the orange ones look like Swiftia. This one could be, there's an astroschematid ophiuroid in there, one of the snake stars. And is this more likely a Canthogorgia with those longer polyps? I'm not, I'll need help from shore. Eric says a plexaurid.
Okay, let's come out.
What's the bearing on the move?
It's 285.
285.
Yeah, I'll push over. Coming ahead.
Yeah.
Any interest here?
We've seen quite a couple of these already.
Okay.
If we could, move along.
Move along.
If we do find any more of the particularly vibrant coral communities like the one we were just at, we'd like to stay and get a couple good shots, please.
All right.
Now that looks like a dead one covered with hydroids.
Do a quick zoom video.
Just one on the way by. And yeah, they're pretty fuzzy. I'm not sure if the hydroids are even alive.
Nav Bridge, we are finished with our position.
And you can see the hydroid polyps are extremely tiny. You can't see them directly, but they're little tube-like openings you can see as the little zigzags along those branches. Hydroids belong to a completely separate group of cnidarians from the octocorals and stony corals and black corals.
Come up.
So for those unfamiliar, all of these hydroids, many of the jellies, and all of the corals and anemones and their allies belong to a major group, a major subdivision of the animal kingdom called the cnidaria, C-N-I-D-A-R-I-A. The name comes from the Greek, I believe, word for nettle, as in stinging nettle, because all of them have produced these capsules within certain cells that can evert explosively a thread, which in many cases is used for defense or prey capture, often called the commonest form is called a nematocyst.
All right, pilots, let me present it. How do you feel about doing another one?
Stinging
and
cell.
just
And there
Sirius
is
right
a
over
deep-sea red crab,
our SEEP1
Chason
possible location.
quinquidens, which
Sure.
is commercially
Okay,
fished.
that's going to be 40 meters, 285.
Okay.
I'll put it in
So
if
within
ready.
the cnidaria, there are two major groups. One group are the Anthozoans, which means flower animal, really. And within that, you have the forms that have a six-part or multiples
why
of
not? Go
six
ahead.
to their tentacles and interior radiating partitions. And that's the anemones, the stony corals, and the black corals, for example.
Good copy. I'm going to read that back.
And then
I've
you
got
have the
40 meters
octocorals,
bearing
which we've
285
talked
speed
about.
decimal 2. Is that correct?
The other major group
Copy. Stand by for input.
is called the Medusozoa, and that includes these little fern-like hydroids. It also includes a wide range of what are commonly called jellyfish, but should properly be called medusae, the umbrella or dish or bowl-shaped planktonic forms
Quick zoom here, please.
that are chiefly in the water column. They're swimmers.
Let's do a snap zoom.
All right.
Let's see.
And just to wrap that up,
There's also a chrysogorgia off to the left, if we could take a look at that.
Sure.
there we have another set of dead shells and a rock.
You want to come out video? I'm not
Yep.
seeing it.
Just to wrap that up, medusozoans often go through a complex life cycle where they are polyps like the hydroid for part of their life cycle, and medusae up in the plankton for the other part of the life cycle. And it gets very, very complicated. And there's a chrysogorgiid, an octocoral with its commensal squat lobster, and another one below it.
And chrysogorgiids are pretty easy to tell apart from the other families of octocorals because they just sort of resemble cotton candy. You know, they're this beautiful little poof with these tiny, small little polyps and these very thin branches. And I think that's two different species of squat lobsters. The one
Yes.
inside is likely the commensal that will stay with that colony, whereas this other one just looks like it was exploiting that little overhang of hard substrate for a burrow.
And one last comment that Daniel Wagner reminded me about, and that is the term coral, which we use for a wide diversity of organisms in this phylum Cnidaria. The stony corals,
just ran into each other.
The octocorals, the black corals, and then over on the Medusozoa side, there are some.
Any interest on
like the fire corals and the lace corals, which are completely unrelated to the stony corals.
Let me know what you want to see.
They are in sort of distant cousins within this major phylum called the Cnidaria. Very, very different internal structures. The
all right, let's go ahead and let the
stony
ship go a little
corals,
further and then
rather
pause it.
the
I think if they want to
fire
get some
corals from
nice
shallow
shots
water and the lace
colonies up here.
corals do not have, and none of the hydroids have those internal partitions that you get in
I can let it go like
the
25.
anemones and stony corals and black corals and octocorals and their allies, which increase the surface area for digestion.
And then
in
that should
the
still put you
interior.
within reach of SEEP1 possible location.
Sure. So you don't want to see this one here? Okay. Okay, we'll do this one and we'll move on.
And this is Swiftia again, I believe.
Not sure if we mentioned this, but deep sea corals are really important for a number of reasons. One of which we're seeing here in that they provide this complex three-dimensional habitat, which is used by lots of other invertebrates. So we saw in that last Chrysogorgiid, there was a squat lobster living inside of it. In the Acanthogorgid we saw earlier, there was a snake star living within it. And so a lot of animals use these corals as a form of substrate to attach to, and also a form of shelter to shelter within, so that they can escape predators that may be passing by. There are also extremely many of them are extremely slow growing. And so they're particularly vulnerable to human impacts, such as deep water oil and gas drilling or fishing in some places.
Average.
But I know in the Gulf of Mexico, this is one of the best managed areas in terms of managing human influence with the needs of various habitats down in the deep.
And Ken Sulak, our ichthyologist colleague, has just logged on and scanned back to look at some of the video we took, and that one, the first one we saw with the long dorsal ray and the long lateral rays is the rat tail, family Macruridae, and the species is Gadomus longiphilis.
Okay, we'll just hold this for a second.
And then we saw another one which also had some long fin rays, and that was an ophidioid called Dicrolini intronigra. And both of them cruise the substrate using their specialized fin rays as sensors. And again, this is a Galatheid squat lobster.
Okay, so where would you like to go now?
Related to the hermit crabs.
Okay.
Despite the fact that the Gulf of Mexico is one of the most explored bodies of water in the world, its close proximity to the U.S. mainland, as well as the number of human interests in the area, including oil and gas reserves and fish stocks, there are still areas of it like this that have never been explored before. This area we are exploring today, known as Henderson Ridge Mid-South. We're at a depth of 1,170 meters. All of this has never been explored, so these colonies are all new to us in terms of their location here. So this light purple one is not one we have seen yet during this dive.
Okay, video.
Well, I think we'll need help on that unless you can.
Lasers off, please.
You have an eye for it.
A plexaurid of some kind?
I'm guessing. If we can zoom in a little tighter on that, if possible.
We're going to get a few seconds of wide for
Yeah,
our
that's great.
pilot
Sure.
video, and then we'll zoom in.
Yeah, that the hard substrate on which the purple one is attached, this looks like it might be dead octocoral axis encrusted. This is a different kind of octocoral. See if we get any help from shore.
So I'm just reading one of the background essays that was prepared for this expedition. You can find that online at www.oceanexplorer.noaa.gov. Within EX 1711, our current expedition, there are a number of background essays which provide some great information on various habitats and dives we were expecting to do during this expedition. And one of those essays is on deep sea corals. And there are some really great factoids in it. Like, for instance, there are more species of coral in deep water than there are in the shallow waters. In the Gulf of Mexico, there are more than 250 species of deep sea corals, compared to about 100 or so species in shallow waters. Many of these, of course, include stony branching corals, cup corals, octocorals, and black corals.
A few seconds of this, then I'll tilt up slowly.
And in the last 10 years, at least 10 new species of deep-sea corals have been described from the Gulf of Mexico. And of course, that means that new relationships have also been discovered, for instance, with those commensal animals living within them. We've also seen such relationships such as Chasion quinquidens, which the deep-sea red crabs that are commercially targeted, they seem to have a particular affinity to coral colonies of Madrepora oculata, which is a type of stony coral that has this very obvious zigzag morphology.
I think they did.
We haven't seen any of those yet on this dive, but we have seen those during this expedition.
Quick thing, Diva. Both Eric and Asako are having difficulty with this pale purple one. I've asked them, and Eric says it's a good candidate for collection.
That is great to know. Thank you, scientists ashore, for pointing that out. Pilot, are you in a position potentially to perform a collection?
Yeah, we can do that.
Yeah,
okay, so Eric and Asako and any other coral biologists ashore, just a snip of this would be okay, correct? Anything in particular you need us to get?
About it video.
Yeah. We're not going to go for the dark purple. We're going to go for the.
We're just getting a close up for our highlight videos. And you wanted to get a few seconds here. Go ahead.
Sure.
Your hydraulics are up.
Okay.
So you can really see why we call these octocorals, because of the 8 tentacles.
Thank you.
And also octocorals, the tentacles always have those little side branches, which you do not find in stony corals, black corals, or anemones. Yes, as long as the whatever you collect has some branches to it, that would be great, because the branching pattern is frequently important.
So our scientists ashore are a bit stumped by this one, hence the reason we're going to be collecting it. Our collections tend to be rare animals, unknown animals, or very characteristic animals. And it's these kinds of new observations that add to this growing body of knowledge about the Gulf of Mexico, the unknown parts of the Gulf of Mexico. Who knows, this may be a new species of deep-sea coral, perhaps. But once we get it up on the ship, we're going to preserve it, and then it's going to be sent to the Smithsonian Museum, where all of our biological samples are deposited, and they are then available to anyone who may want to work on them. They can request them, and then you know, maybe someone may do the DNA work as well as look at the morphological characteristics of it, so how this piece of coral will look. And who knows what they might find. It may be a known species, maybe an unknown species, but this is the beauty of exploration.
Well said.
Wow, it's actually a lot smaller than I thought.
Yeah, we can get lasers on real quick before we cut it.
I think that arm makes a lot of things look small. What was that? You on lasers?
Sure.
I should have said that background essay I was looking at is by Peter Etnoyer, and he's a marine scientist specializing in deep sea corals from the NOAA National Center for Coastal and Ocean Science.
Getting a little scared there. You want to zoom in?
Really great read, if anyone's interested.
So, is this fine?
Oh no, that little squat lobster's
Yeah,
running away.
He's not having a good day here. Is this going to be fine for you if I
Yeah,
cut
that's going to be great. Thank you.
Okay.
Oh, taken. Oh, no. Okay.
Good. We left some for posterity. And you saw that little squat lobster shoot off the bottom. They can swim by clapping their
when you're ready.
Curved abdomen underneath, like a lobster does. This is our first sample of the dive. And it's going into the port inward inside box, bio box. And these bio boxes, these receptacles into which we collect things, are insulated, and both against light and temperature change, so that when we bring these specimens back up to the surface, they are still at the temperature at which we collected them. And when we
wouldn't mind bringing the mini Zeus over.
may have to plunge our hand into that
Should
to
we just do it
retrieve
real
the
quick?
collection
Thanks.
to the specimen, it is very cold. What we do then, we bring them on board, put them immediately into chilled buckets of seawater, bring them into the lab. We have labels made up immediately with all of the collection data and as much identification information as we have. And we photograph them with the labels. We also frequently take small subsamples for DNA analysis, put them into vials, and so we have to include in the photograph the little numbered vial, so everything is recorded. And then in many cases we will
Where to next?
take a small portion of the specimen.
If we could just continue on to the SEEP 1 waypoint and then move along from there to SEEP 2 and so on.
Okay, we'll keep an eye out.
And we're just, there's a particularly, before we go there, a really
Yeah.
large colony down to the left.
Yes.
We're keeping a particular eye out for large coral colonies as well
exactly
as
what we wanna
chemosynthetic
see.
habitats. So yeah, this is great.
Let's move over in that direction.
And so we also
exactly what we're
photograph
hoping to
small
see.
bits of
Physically
the
move the ship,
specimens
or just-
smaller
Yeah,
specimens
yeah.
under the microscope.
180.
180.
We also photograph first we'll photograph the
And
label
let's go do
under
like
the microscope,
20 meters
and then
or
we'll
something.
photograph the
You got it.
specimen. And then they are preserved mostly in 95% ethanol and into Ziploc bags.
Go ahead, Nap.
And eventually transferred
Yeah, good copy. What do you
into
got?
heat-sealed bags for more long-term storage, because these will not make their way to Smithsonian until after
Good copy. I'm
all
gonna read
of
that
the
back. I've got
cruises
20
in
meters
this
bearing
part
1,
of the
0 speed decimal 2. Is that correct?
world are completed.
Copy.
That
Stand by
is
for input.
through probably next
So
summer.
actually, we've got the ship underway.
You've got more tether. You can definitely get over there.
Yeah.
What does that mean?
Bend a little bit.
So I can't get all the way over there, but we can start with some of these.
All right,
Yeah, so if we could just get
corals that
some
are
great
nearby.
overview shots, and then
speed
if we could get a
decimal
couple quick zooms, that would be great.
Okay.
Thank you.
So the ship is underway, so we will not be able to get as close as we perhaps like. But these mostly look like the octocoral Paramuricea and the astroschematid snake star. I believe the genus is Ophiochreus.
They look like there may be a couple different species of brittle stars here.
That could be.
There's some variation in color. Some are bright pink, some are orange, some are a little bit whiter.
And there's just to the right of the center of the screen, there is up in one of the fans a chirostylid squat lobster, different from the ones that we've seen earlier today, that typically cling with their very long claws up in the sea fans, waiting for unsuspecting small prey to drift by.
To your right, but it might be wrong.
Take a look.
And again, remember that our laser dots are 10 centimeters apart, 4 inches.
Go in.
So maybe this fan right here is perhaps a foot across. And this does look like a Paramuricea with these snake stars, and there's a small anemone upright, as well as that chirostylid. And these fans can get quite large. And Eric tells us that in particular this looks like the Paramuricea form B as in baby 3, which is still undescribed but genetically distinct from all of the other described species.
Got any more zoom?
Yep.
Okay. Nice.
That's full zoom.
So these ophiuroids are likely Astroschema clavigerum, and these would be different from the ones we saw a couple of dives ago, which were Ophiochreus. And they're of course termed snake stars because of their very curly arms that sort of slither along when they move. They're also sometimes known as serpent stars.
They are in the same major group of brittle stars with the basket stars, which have branched arms. All of the other brittle stars that we see typically on the sea floor cannot coil their arms like this.
Any other creatures here you want to take a closer look at?
No, just some great overview shots of the... but this is a great example because you can see that the right part of that colony had their polyps open, and then the left part had all the polyps retracted, so it looked almost a little naked. But this is a great example of these coral habitats providing that 3D structure which can then house many different invertebrates. So just on that one colony there were at least 3 species of invertebrates living within the branches.
I was going to come to this one that's in the bottom center, or if you prefer, I can move up slope to those ones further away.
That sounds great, thank you.
Okay, all right.
So it looks like here they're living on orthogenic carbonate, which are associated with areas where there was or still is hydrocarbon seepage.
And just as a follow-up, Ken Sulak tells us that rather ugly, blunt-headed fish that with the ridges on its head that we saw hovering above the bottom earlier was a fish called Stephanobarix monet. It's not a midwater fish, even though it was just above the bottom.
Now,
but it's a hovering species that will hover
Position
above
move
the bottom.
Has been completed.
And prey on organisms in that what we call the benthic boundary layer, or the seafloor bottom layer. And there was an eel, which we mentioned, a synaphobranchid eel that had an inflated-looking snout. And Ken tells us that that inflated snout, equates to a sexually mature male with a seasonally enlarged olfactory structure called a rosette. And that brings up an interesting point. Even though we are in deep water here, and that's a beautiful shot of the serpent star with a small shrimp next to it on this Paramuricea. But even though we are in 1,100 and almost 70 meters of water. That's over 3,000 feet down. There are seasonal signals. There are seasonal variations in the amount of detritus that comes down from the plankton above, for example.
Can you try to
and
get a
the
close-up of
organisms
squat
here
lobster?
can respond to that.
Where was that at?
Is
He's up. Okay.
right there.
We can do that. Try it from this angle.
Okay.
And this is a chirostylid squat lobster. You can see it's quite different from the ones on the sea floor that we saw before. Those very long claws waiting for prey. And again, 3 pairs of walking legs. The 4th pair are small and tucked underneath the carapace. And the abdomen is tucked underneath the body.
So these are likely from the genus Gastropticus.
And I don't know if you ever see them on the sea floor. They're always clinging to these various sorts of octocorals or other erect
come out.
Cnidarians like black corals.
And, I'll go ahead and move up to those upper colonies there.
And right of center looks like another colony of the one we just collected, the unknown pale purple octocoral.
And it looks like something's created quite a big burrow there. You can see this big, shelf, discolored shelf almost, where it's pushed a lot of sediment out of its burrow
frame that
and into the surrounding areas. That colony must have,
okay, go in tighter.
I don't know,
We'll start with
15 of these snake stars on it.
At least. Oh, there's a, that's a tiny little shrimp.
I
That
think
looks
that's
like an opossum shrimp.
Yeah.
Yes, a mysid with its dark red brood pouch underneath. It has no abdominal appendages, and it's swimming with the outer branches, the feathery outer branches of its thoracic legs, which are called the, those branches are called the exopods because they're exterior.
Got more zoom.
And it is unrelated. It is separate from the true shrimp, the commercial shrimp,
okay,
and the cleaner shrimp,
better request
for
for
example.
lasers on this fan.
It's most closely related to the
That's
isopod pill bugs and the amphipods, the beach fleas, and their allies.
I guess we can zoom in just a touch. Let me frame up the, perfect.
That's a beautiful image.
And so this colony is nearly 1 meter across. You can see the lasers at the bottom of the colony. That's
Okay.
really wonderful. Thank you, Pilot.
Do you want to get lasers off for a few seconds?
Yeah, that would be great.
Okay. And he's making a coral focused.
Emily is.
Oh, Emily.
But,
Emily's making the coral focused highlight long form. So.
I think that's probably good.
Okay. You can come out. Anything else here, watch lead?
No, we are good to continue on. Thank you.
Okay, we're good. So where are we? So where do I need to go for SEEP 1?
All right, SEEP 1 is pretty much directly north.
Okay.
Maybe like...
Not that that's a precise point.
Yeah,
anyway,
but if we were,
yeah.
340 would be exactly the direction D2 would need to travel.
340.
Do you want me to give Sirius a bump to put her right over?
Maybe we'll do a little scan with the sonar.
Copy.
Yeah, that would be great.
See if we can find something.
Looking for some bubbles or hard targets.
There's
Ooh,
a
so
little outcrop there.
Yeah.
Oh, we can take a peek at that.
Oh, it looks like we're coming up on a couple ahead.
Zoom in a little.
So it looks like we're moving into an area where there may be some more of these fragmented pieces of carbonate.
You want to see any of these corals here?
No, we can just keep moving on.
Okay.
So this, we can see most to the left is that plexaurid, which we just collected. Then in between there's a chrysogorgid, and then off to the right, the possible stoloniferan.
Looks like potentially some features ahead here.
It's definitely a depression.
Yeah.
You want to, let's see, it's 20 meters out for you.
I'm just going to do a quick scan.
Copy.
I think I will scan more to the north.
So that one was like 280 if we want to go back there.
I'm going to go all the way north of trash. Obvious. What did you guys see yesterday? What did it look like?
It was a it was a mound, so they saw the sonar hit on the mound.
Yeah.
So
you didn't see the actual bubbles though. Oh, okay.
So right now you're like 15 meters from possible target.
Okay.
And I don't really see anything on sonar.
Yeah, we'll just do a quick sweep. Also looking for hard targets for outcrops. So
it's definitely. Looking at your sonar, it's more to the west.
Yeah.
At least the bathymetry.
Right. Okay. We'll bring it back a little bit. A lot of particulate in the water. Quite a bit of current.
Which would make sense. We were seeing bubbles in the water column. It'd probably be more west.
It's true. Yep.
Based on the current. But that's what we thought yesterday, and it didn't pan out. So.
Okay. Well, why don't we go toward that little depression, and then we'll...
Copy. So that's, you're looking 280.
Yeah.
Copy. 30 meters, 280.
Sure.
Happy?
Sure.
Great.
Coming back down.
Go ahead, Nav. Yes, we're ready.
These coral colonies we've been
position
seeing
move, range 30 meters,
indicate how reliant they are on that hard substrate. Most species, and I say most underlined, do rely on this hard substrate as a source of stability, so that they can attach to them, and that's where they'll anchor for their entire lives.
Quite a bit of current, roughly south to north.
But then, of course, there are some species we've been seeing, such as that Lepidopsis caryophylla, which are able to anchor in sediment.
Nav bridge.
Those are much rarer, though.
Position move initiated, range 30
here we've got
meters,
a couple more colonies of
280, speed decimal 2.
Yellow Paramaricia. Some more of these purple stoloniferans, as well as this light purple plexaurid that we just collected.
So watch just in case it's useful. The current is definitely moving generally south to north, and it is not too strong, but I'd say probably moderate.
Okay, thank you.
Mm-hmm. So those are the corals we saw earlier. So that was 285. Is that what you did?
We're moving 280.
280, okay. Any interest here, watch lead? Looks like a lot of the same stuff.
Yeah, just some quick zooms would be great.
Okay.
For instance, this Chasion red crab underneath the colony would be a great example of a relationship that happens down here with these deep sea corals. So here we've got a couple more of these beautiful colonies of Paramaricia, and this one's got quite a couple commensals on it, as well as one of those maybe homotheid but Venus flytrap anemones in the background that are also found in areas of hard substrate.
And so this is interesting, that crab, that looks almost a hybrid between the golden crab and the red crab.
Or at least it's just molted, so it looks pretty fresh.
Okay.
Fresh and clean.
So that would probably be a red?
Yeah, I think so. The golden ones tend to be like more sandy colored.
Okay.
But these ones are known to have an association with deep sea coral colonies, usually Madrepora oculata, but in this case it could just be here sheltering amongst the carbonate rubble and or outcrops in the coral colonies.
And look how widely spread the claws of that little chirostylid squat lobster are. It's just stretching. Come to me.
Another Chasion there sheltering under that coral colony.
And Ken Sulak reported on our chat room a nice historical note that I'd like to read while we're moving along here. While the Okeanos Explorer missions are the first to explore certain areas of the Gulf, as we've talked about, there were many earlier undersea expeditions with the Johnson Sea Link submersible, with remotely operated vehicles and drop cameras, sponsored by the Bureau of Land Management, now BOEM, and NOAA's Undersea Research Program, which used to be NERP.
Quick zoom on these back mats on the right.
Also, there were, of course, long history of sampling by trawls and grabs that was accomplished by ships like the U.S. Fish Commission steamer Albatross, the Blake, the Oregon, the Silver Bay, and Oregon 2. And so they produced enormous collections of fishes and invertebrates.
Here.
And those have informed much of what we know about the fauna of the deep Gulf of Mexico. And both Ken and I have spent much of our professional careers looking at those collections. That was great, Ken. Thanks very much.
So this looks like a tiny little chemosynthetic patch. We can tell that from the black reduced sediment, which has a lot of the white bacterial mats growing on it, as well as you can see this really cotton-like fluff growing on this carbonate here, and those are much thicker bacterial mats where you can actually see the individual strands of the bacteria, which is pretty cool. But just to the left of it, we've got a live tube worm, and so this would be one of the chemosynthetic tube worms, which rely, instead of getting the nutrition from surface-derived material, these would use chemosynthesis to create their food via bacteria within their body in an organ known as the trophosome. So if it were to stick out of its plume, you'd see it's bright red, and that's for hemoglobin, which transports oxygen around its body. very similar to us. I'm not quite sure if this is a Melobrachia or a Scarpia, but this is one of these cold seep, or rather chemosynthetic endemic animals found at cold seeps and hydrothermal vents around the world. Well, not around the world. These tend to be
nav bridge
specialized to the Gulf of Mexico and the Atlantic.
Position move completed.
Great, thank you.
That's great. Thank you, pilot.
Okay, want to come out?
So Eric Cordes, who's had a lot of experience not only with deep-sea coral habitats in the Gulf of Mexico, but also with cold seep habitats, says that this one looks like Lamellobrachia, and it has the very obvious lamellae on the plume.
Oh, little squid, yeah.
So that's just a reminder that this entire area may be, or is geologically active. There is probably subsurface seepage happening of hydrocarbon-rich fluid, which that tube worm is able to use its root, the end of its tube, which is burrowed deep down into the sediment, to mine those sulfide-rich compounds. And of course, that is linked to these coral colonies we're seeing, in that there's a reaction that happens which creates these orthogenic carbonates, which these corals then use as a form of hard substrate to live on.
Drop-offs.
A little depression over there.
25 meters out. Yeah. There's one. That one's right in front of you, but past that, you can see on sonar there's a larger depression.
I think it's important to mention, especially in the wake of Ken Sulak's comments about the history and the collections that have been made, the reason we can identify many of these things is because
yeah, sure. Is that generally toward the seeps?
We have specimens that were trawled or dredged up and
it's a little more
put
south,
in museum collections that will
so
allow
the seeps are
us
pretty much
to
going just west.
Really examine these things, look at them under the microscope.
Generally,
there are various
just keep doing westward moves, and you should still be able to reach it.
Fine anatomical features,
Sorry,
their
Ashley,
molecular data, and it's this basis of physical specimens that allows us to tell you
let's just
what
put
we're
another move
seeing.
west then.
And of course,
I'll just do 30-meter
as
moves
the
west.
example
Sure.
of that
should keep us in line.
pale
Sounds
purple
good.
octocoral
So now that
indicates
she's at lunch,
there are still things out here that we don't know what they are. They perhaps are undescribed, which
ahead,
is
Nav.
one of the reasons we collected that.
Yes, we're ready.
And it may turn out, for example, that there is some small fragment or specimen tucked away in a museum, perhaps
Roger
the
that.
Smithsonian,
Position move, range
or the
30
Museum of
meters, bearing
Comparative Zoology at
270,
Harvard,
speed
or the
decimal
Field
2.
Museum, that was collected anywhere from, who knows, 5, 10, 15, 50, or 100, or even 150 years ago, that has just sat and nobody has gotten around to examine it.
It's a lot like what we were
And
seeing
so
yesterday.
the museums also
said was
are
biological.
amazing repositories, not just for what we know,
pluming debris,
Nav
though.
bridge.
but also what we don't know.
Move
As an
initiated,
example,
range 30 meters, bearing
A
270,
couple of years
speed
ago,
decimal
I went
2.
up to Smithsonian. I've been working there for much of my professional life at intervals,
Right past,
making
moving
visits
west.
and
All right.
working on their
Lining back
collection.
up. You said
And
270?
I found that there were a
270.
number of
Copy.
stacks of shelves with literally close to
it's like
600 jars
see the bridge.
of crinoids that had never been identified. And crinoids, of course, are my specialty. So we ended up shipping 7 55-gallon drums of crinoid specimens in jars down to my lab in Florida. And my graduate student, Brenna Hayes, and I spent 2 years identifying them all. And most of them
Wanna go in
were
on the fish?
indeed known
Sure.
species. And here's a little, looks like a little rat tail.
cruising in the current.
And there's, you can see there he's working against a fairly strong current over the bottom, and a bit of rope there. That pale green right in front of him is a bit of discarded twine of some sort. At any rate, just to wrap up that story, among those 600 jars of crinoids, I found one species that was probably described 100 years ago, but the original specimens have been lost. So now I'll be able to re-describe it properly because the original description was very poor. And I found 2 specimens of another species that were new to science that I had just described the previous year. So you never know what you're going to find in a museum collection. And they are hugely important repositories of the Earth's biodiversity. They can tell us what we have had, and what we may lose if we're not careful. And that looks like one of those Venus flytrap anemones coming up, almost like a catcher's mitt. But that may be a different one. It's much paler than the ones we've seen, but there is some variation. And there's a bit of sargassum weed, dead sargassum weed rolling over the bottom.
There's a bunch of stuff to your starboard.
Yeah. Any interest in the anemone?
Yeah, just a quick zoom. It looks perhaps like what we've seen before, but it's got the knobs on it, but it's a much paler version of what we've seen before. And then there's a small couple of orange anemones and the almost ubiquitous squat lobsters sheltering underneath. Thanks very much.
Okay, come out.
And that looks interesting there. What do we have?
An little outcrop with a fish there.
There's a fish sheltering behind another one of these isolated cobbles of blackened orthogenic carbonate. And that's the same Venus flytrap anemone, but it's facing away from us, so we don't have to zoom in on that.
Okay.
Thank you.
Fish is kind of hiding anyway.
Here's some different kinds of burrow traces. Another little eel. We've seen those before. I might mention also as a follow-up to that, my little commentary about museums, and that is we still probably do not know and have not described the majority of species of life on Earth. There are over, I think the general consensus now is close to 1.7 million described species. Majority of those are insects, of course. But we keep finding new things. People have estimated that we may have only described a tenth of the known species on Earth, or even more.
Yeah.
And that's a bit of sargassum weed in a dead bivalve shell in a crater. No, go right ahead.
Current's picking up.
Hey everyone, I'm Alex Nancy Foster Scholar here in Okeanos, and I am reminding everyone that the NOAA Office of Habitat Conservation and NOAA Fisheries is doing a live tweet. So all of you who love Twitter and love to follow us on Twitter, they will be sharing pictures, chatting up with people, and answering questions. So just remember to use the hashtag DeepSeaTV17. That's hashtag DeepSeaTV17.
Thanks very much. That just gives you another idea of the variety of ways we're trying to get this information out to the world. And as I said, taxonomy has become one of the unsung sciences. It's fallen by the wayside in many respects. That is the part of taxonomy that actually looks at the whole organism and describes it. And it's really, if I may state in a widely held opinion, it's really a shame because there is so much left out there to discover that we still do not know about. And unless you know what's out there, you don't know whether it needs protection or not, whether it's important to the human condition. Does a deep sea unknown sponge have new biopharmaceutical properties? Or who knows what?
Something back there.
And we're coming to some interesting topography here. Yes, much more vertical relief over this ridge, more hard substrate.
There's some good stuff.
Oh, yeah.
All right.
That's where we were trying to get you to go
that
earlier.
That knob in the center is probably a contracted flytrap anemone.
Wow. Just mounds. Yep.
And I'll need help from the geologists ashore because
scientists walk down the room.
Do you want to move serious
tell
like 10
us
meters
what some of this rock is, possibly asphalt.
218. I'll put it in.
Yeah, that sounds good.
Lots of what look like bacterial mat and probably dead bivalve shells. The white fragments you see there. There's another flytrap anemone and a Gracillochinus sea urchin on the left. We've seen those before. But this relief is rather different.
Roger that. Position move. Range 10 meters. Bearing 220. Speed .2.
When you get settled, if we can get a zoom on that big rock.
The one on the left here?
Yes, sir.
Sure.
So Eric Cordes is telling us through the chat room this mounded seafloor is often found over gas hydrates. And we'll just take a close-up look at some of these rocks here.
Can zoom in a bit.
Probably orthogenic carbonate, according to Eric.
Move has been initiated.
Lots of, a number of little squat lobsters, the Galatheids on it. Very rugged texture here. Geologists, anyone want to chime in on whether we need a closer view? Well, we're taking a closer view, which is fine. Get an idea of the texture here. Again, what has been described as, I believe, vuggy, V-U-G-G-Y, to describe a texture that is filled with small holes, sometimes a geological term, vug, V-U-G.
All I'll say is this is probably carbonate, but there's not much else to say at this point.
Oh, okay, thank you. There's a little tube worm. Okay, we can zoom out, pilot.
Okay.
Thank you.
Let's come out.
And let's see what else we have in this vicinity.
Slight line there.
Some discarded line, probably fishing line.
So I was in the middle of lunch, but I've run back upstairs because there are bacterial mats and shells everywhere.
Not seeing anything alive as far as shells.
ROV nav, bridge.
Go ahead, bridge.
Last move complete.
Thank you, sir.
So these chemosynthetic habitats are all often seen within these depressions in the seafloor. And so here we can see there's a lot of reduced sediment as well as bacterial mat. And so that indicates to us that there's probably some hydrocarbon seepage in the area. And I'm not seeing any live clams or... I think there may be a mix of clam and mussel shells here, but I'm not actually seeing any live animals. So that could indicate to us that this is a colony that was once thriving, and since then the hydrocarbon seepage has begun to wane, and it just isn't at the levels that many of these bivalves need to sustain their lives.
The black one.
How far are we from that outcrop that's right in the photo right now? Is that one we could sample potentially?
Sorry, you asked how far we are from which outcrop, Carolyn?
This outcrop that we're looking at right now with these nice jagged carbonates here on the side. I'm wondering if I can't judge distances, obviously, if we're close enough that we could potentially sample a piece of that.
Oh yeah, if you did want to sample something in this area, we could absolutely facilitate that. You just have to let us know which outcrop exactly and which piece of rock exactly.
Well, I think I'm not getting emails from my British colleagues, but it's obviously a strange time there. But I think this one we're looking at right now is quite good.
It does sort of look like fabric.
So what we're looking for is a piece that doesn't have a lot of shell hash in it.
Okay.
Because that makes it harder to date.
Right.
And not a lot of holes. And you just need to take the manipulator arm and try to just get some off. And it's preferable to get something that looks like there's some trash there.
Yeah, it's just trash.
Okay, let me speak to the pilot. Pilot, did you hear that?
Only part of it.
Okay, so basically we would like to collect a piece of carbonate, but we would prefer a piece...
Stuff you're looking at right now.
So we'd prefer a piece that isn't too... doesn't have that many holes in it and doesn't have any obvious shell hash attached, within it. So that stuff we were moving away from was actually pretty good.
Okay.
And the stuff that's just in the sort of center right now with the sort of white patches on top of it is not bad either. There's like a little underhang under there. That would be a good place as well.
The only thing that concerns me with that is I don't know if we'd be able to break off a piece of it. Pilot, do you think we would?
Can you, let's get the lasers on and then you can reference exactly where you want. If you wouldn't mind tilting your camera down a bit, Josh. Okay, so lasers are on. Can you reference the section that you're interested in?
Carolyn, could you say, with regard to the position of the lasers, where exactly you were thinking?
Well, actually, the spot just up from where the lasers are now is fine, where there's robust rock up there, or over to the left is fine as well, about 30, 40 centimeters to the left is fine. What you want to avoid is the kind of more shell-filled stuff that's down at our lower... I know this is very frustrating for everyone. I'm sorry.
No, it's okay.
Something like if you can try to get the manipulator arm to break off a piece of that might be possible. I don't know. It's probably going to be pretty crumbly.
I might be able to touch my. I don't know how hard it is.
But if it's not crumbly, you don't need to waste a lot.
Might be crumbly.
Okay.
Okay. Well, we're going to try and see what happens.
See this little protrusion here.
That's great, and I'm just hearing now from BGS, and they also think that part of the top there would be great if you could try it.
Stuff that's laying in the depression and stuff like that.
Yeah. Okay. I think everything's pretty safe. See the tether kind of going up.
Yeah, I mean you're fine.
Yeah, Sarah should be settled, not moving, so should be good.
we come out a bit, Annie?
So that's full wide.
Okay. Yep. Hydraulics are up. So she said it might be pretty crumbly.
Yeah, if they don't think it's possible, that's fine as well. I don't want to waste everyone's time
We're just
here.
settling down, and we haven't actually tried yet, so just give us a little bit, and if it is not possible, we will absolutely tell you, don't worry.
Like that one below the pillar might already be broken off.
Yeah.
There's a pretty good view
No,
in
that's
Mini
what they
Zeus.
want, but
Yeah. Yeah, can we get Mini Zeus up? Now that we're stable. If you can help me keep an eye on everything, Sean, would be great. Okay. Well, I'll just poke around, see what's loose, I guess, to start with.
I think Carolyn was also particularly interested in the non-loose stuff, if we could break off some.
Okay. All right. So what about this little pillar here, if I'm able to get a chunk of that?
Yeah, so Carolyn, how does that look? This pillar that the lasers are on, is that too holy, or is that something that would be okay?
It's probably not the best, but let's just give it a shot. I think it's okay. Like I said, I don't want to waste everyone's time, and sometimes I'm not the expert in what these samples look like. I'm just getting emails from Britain, but go ahead and try, sure.
that was already broken loose.
Crab's
And
like,
there's a crab under it.
Perhaps a little more than you wanted.
That's okay. We've collected far bigger on the Okeanos Explorer.
This should be okay.
So if that's okay with you, Carolyn, we will pop that in the rock box.
Oh, yeah, that's fine. That's more than enough. I'm sure I'm going to get a funny email from Nottingham in a few minutes here. But yes, sure, that's great. Thank you.
We expect like 10 papers from this one, given the size.
Yeah, that's
This is nowhere near a record for us. So.
It's probably pretty light too compared to the manganese encrusted ones we were getting in the Pacific.
Yep. All right, can you
Let me know
tilt?
if you want the drawer out?
If you could just tilt Mini Zeus so I can see the elbow joints and stuff, would be great.
Actually, the verdict from the British Geological Survey is perfect with an exclamation. So.
Great. Okay, pilots, thank you very much for that wonderful collection. As you heard, colleagues at the British Geological Survey have said that was a perfect collection. So our second collection of the dive, the first being a Acanthogorgia, as far as I remember, Plexaurid coral that we were unfamiliar with, and now a piece of carbonate rock, so potentially that will help to help in our understanding of the origin of the area as well as what's geologically happening here.
And I'll bring the mini Zeus back over for Roland. As you know, he likes
just as an indication,
between the toes.
specifically, we've been collaborating with BGS for a few years now,
I think video's gonna switch out.
they're specialists in dating these rocks with uranium thorium methodology.
Thanks,
so
guys.
that allows us to know when the methane emissions happened. Thanks.
Wow, great. Thank you very much, Carolyn. Thank you for taking the time to explain that to us.
I would agree with what Eric said earlier. These mounds look very much sort of like what you see with inflation with gas hydrate below. Obviously, we can't know for sure, but they do have that appearance.
And do you know how that would affect the biology, Carolyn? Why, for instance, we're seeing so many dead clams?
No.
Sorry.
We all have the same thoughts, right? Toxicity, disease, maybe they are no longer above their supply of nutrients, so I don't really have an indication. Usually in hydrate areas you would see pretty rich biology.
So I'm just going to continue doing some nice wide shots until you request a zoom watch lead.
Sorry, what was that?
I'm just going to keep scanning around with and getting some nice wide shots until you request a zoom on something.
That sounds great, thanks.
Yeah, I don't see, there's a lot of little depressions around, so I don't see a strong direction for next move yet.
Yeah, we'll just I'll just go out at a tether length for now.
Yeah, I think that's good.
You want me to lose the skids?
Absolutely, yeah. You can lose the skids anytime, just call it out.
Yep.
Trying to be better about that, so we don't have ROV in our highlight videos.
So Eric Cortez was just commenting that there may actually be live clams in the area, just they tend to live burrowed just below the surface of the sediment, so we may not actually be able to see them. But in areas that I've worked in before, such as off Kikimjeni, Grenada in the southern Caribbean at some seep sites there, we were able to see the siphon of the clams sticking out of the sediment. So really interesting, but there's just such a huge abundance of dead clamshells here that this would have been quite a community when it was thriving. Yeah, so Eric is now suggesting that perhaps in more high flux environments where there's more of this hydrocarbon seepage, you tend to see them protruding from the sediment, but in this case they may have to burrow down deeper to get to the good stuff that they need to create their food, those chemical rich fluids.
How many different
We can also see a couple of squat lobsters. It looks like 2 different species just at the bottom of the screen, some white and some pink, as well as there were some Chasion quinquidens, which are the commercially targeted red crab species.
Can zoom past the skid rolling. There we go.
And even though there doesn't appear to be a lot of obvious life, it's possible that this shell hash that we're seeing could actually harbor a pretty rich macrofauna community.
Nice,
so those are the size class of animals down from megafauna. Megafauna tend to be what you can see in imagery, such as what we are doing right now. But below that, you tend to get animals which are like a centimeter or so in size and smaller, and a lot of them live within the sediment, but here they may be living amongst these shells in what could be quite an abundant or diverse community.
Okay, you can come out.
So Eric has also just said that gas hydrate is probably slowly accumulating and releasing methane at different times, and it'll get oxidized very slowly and generate that sulfide that these clams need. But these shells may be particularly old, and as a result we can see some of them actually encased in those carbonate rocks. Some of the orthogenic carbonates from nearby were dated to be at least 12,000 years old. So these could be almost fossil clams we're looking at here. Ones which would have thrived in a different time when that fluid was, pumping out a lot harder from the much deeper sediments.
Is that a different shrimp?
I think we've seen that guy today, but we can do a quick zoom.
So just a quick reminder about the mechanism that allows these incredible oases of life, these cold seeps and other chemosynthetic habitats to exist. It's known as chemosynthesis. So of course, in shallow waters and on land, the basis of all life tends to be via photosynthesis. And so that is when plants, the primary producers, use sunlight to create their energy. Whereas, of course, in the deep sea, there isn't any light, especially at these kinds of depths, nearly 1,200 meters down, 1.2 kilometers. There is no light down here. So as a result, we don't get any plants. And the majority of life on the deep sea floor does rely on food that comes from the surface layers of the ocean. So plankton, phytoplankton, zooplankton, the poop of organisms, the old shells of organisms that they may have shed, all of that will drift down into this deep sea in the form of marine snow. And that will then settle on the sediment, and that is what most animals down in the deep sea rely on. But as you can see in the sedimented areas, there doesn't appear to be, a lot of obvious life. Whereas in these areas, there at one point was a lot more life. And that is because of this chemosynthetic seepage. So, well, the seepage isn't chemosynthetic, but because of chemosynthesis. So instead we have, of primary producers, which are plants in the shallow waters, we have bacteria. And these bacteria use these chemical-rich fluids, which are seeping out of areas that are usually associated with oil and gas exploration and exploitation. They have a close relationship, those two things. And so this fluid will be rich in sulfides as well as methane, and those chemicals are then used by different types of bacteria to create food for various organisms. Sometimes the bacteria will grow in mats on the sea floor or on animals, and there they'll be grazed upon by other animals. So just like at other deep sea habitats and other habitats around the world, shallow water, on land, there tends to be this food web or food chain, which is you have the primary producers, which on land are plants, down here bacteria in these environments, and then there'll be grazers. So on land that could be like a cow or herbivores, whereas down here that might be a snail, which is found at these habitats, or some kind of animal that will feed on these bacterial mats directly. But then you also get these really special animals which are actually endemic a lot of the time to these habitats. So that means they occurred nowhere else. And they will actually have some of these bacteria within their body, and they can use these hydrocarbons, the methane-rich fluid or the sulfide-rich fluid, to create food within their own body, and that is where their form of energy comes from. And then, of course, because these are oases of life, they are very, very productive areas. You do also get animals that come in from elsewhere in the deep sea to make the most of this productivity. So you'll get predators moving in, large fish, large crabs, squat lobsters, that can come in and then pick off some of these chemosynthetic animals or more endemic animals.
Just scan up this
that
ridge and then maybe
perhaps
we'll
rely
line
on the
back up to the
bacterial
west and
mats or, in fact,
move that
are
way.
chemosynthetic
Sounds
themselves.
good. Okay. So watch lead, we're just going to finish doing our scan of this area, and then we'll get set back up, pointing west, and move toward waypoint 3.
Sounds good, thank you.
Diva, is this, before you go, is this the first sort of continuous crust that you've seen? Even it's broken up, but is that more substantial than what you've seen in the last while?
I mean, this is the most amount of crust we've seen in one area, but I suspect a lot of this area, as we saw from our back scatter, may in fact just have this carbonate crust underneath a layer of sediment, and we may just be seeing outcrops of it here and there. But I could be very wrong about that because I am not a geologist.
Well, it sounded good to me. Yeah, this is certainly more rugged terrain, more vertical relief than we've seen, than we saw earlier in the dive, and more extensive outcrops. There's another bit of water hyacinth. Is
that what that is?
Yep. Shallow freshwater plant, which is actually invasive in the southern U.S., clogging canals and things.
Okay. Do we want to do a hop toward the west, or which direction do you want to go?
So, let's see. Seep 3.
Which way is the continuation of the ridge?
Seep 3 is 280. So it kind of depends.
I think we
It's nothing huge on sonar. Everything's kind of various depressions.
Let's make a move toward waypoint 3, and then just kind of we'll do a scan along the way.
Copy. So waypoint 3.
Sounds good.
Do you want to go? So cut to waypoint 3?
I guess we could.
Or do you want to do a little
to the northwest
little
a bit?
L?
Yeah.
All right. So how about we put in
285? That sounds good. Yep.
Okay. Copy. I'll do 30 meters 285.
What do you detect as the direction of the current down here?
It's been moving south to north.
Is that a puffy pink coral center?
It might be.
So we are switching some of the ROV crew right now as we pause for a little bit to switch personnel, and we will head to our next destination after that.
Stand by for input. ROV nav bridge, move initiated 30 meters, bearing 285, speed decimal 2 knots.
Good copy, thank you.
You're welcome.
All right. Hey, Don, next move we just put in is 285 for 30 meters.
Okay, you just put it in just now?
Just put it in, yeah, so you're not swinging.
285, what'd you say, 30 meters?
Yes, sir.
Copy that, thank you.
Welcome. All right, so we're moving away from your headset. All right, just put in a move in, 285, 30 meters.
Roger.
Yeah, Dan kind of told you we're just going to cheat and go to waypoint 3.
Copy that.
You got a rock in your starboard forward, and then you got a bio sample, a little piece of coral in
port inner.
Port inner.
Once you get in place, if you want to introduce yourselves.
It's brown coral.
Yeah, of course. The watch change hasn't quite finished yet. We'll wait for the last guy to swap in, and then I'll go for it.
Very good. This is an enormous block here. Look at the
right
size
now,
of that.
This depression that we've been
And
surveying.
there's a
so you've seen all this.
what looks like a paramuriceid fan on the right-hand side.
Is it 258 or 285?
285.
285.
And I just put it in
that
2 minutes.
eelfish
You won't
on the
feel it
far
yet.
right?
But yeah.
And it looks like there may be, I don't know, look like sort of subfossil bivalve shells blackened on the left-hand side of this block.
You said this was a depression, you said?
Yeah, we were looking for...
Did you find the bottom of it? Is there anything up?
We did. We found some mounds, but they're not active.
Oh, okay.
But once upon a time, it was spewing something.
Gotcha.
Okay.
Copy
that.
Seen a lot of fish today.
Roger.
There's two kinds of shrimp on the bottom there. The paler one was Glyphocrangon, and this red one just drifting off screen is probably Nematocarcinus. When we're all settled in, the next Glyphocrangon shrimp we'll probably try to zoom in on, see if we can get a good look so that Kevin Rademacher on shore can give us an ID. There are several different species found here in the Western Atlantic.
I think you got one coming up slightly right of center.
And there's one coming into the middle of the screen right now.
I got it.
Right on the lasers, boy. Great accuracy here, pilots. See what we can see.
As close to it as you can. They got the very interesting horns.
Okay. I'll come around for a side shot here a little bit. How's that rolling?
It's good.
Okay, planting down. Come on in.
So again, this is Glyphocrangon. Magnificent shrimp here. Its carapace is rather thick and covered with these spiny ridges.
ROV nav bridge, that move looks
I don't
like
think it's ever fished commercially because it's the exoskeleton is so thick and there's not much meat on them. Those huge orange eyes are pretty impressive.
Excellent.
It's got a couple of tall spines on the first abdominal segment. And if you could pan a little bit to the posterior, I'm not sure if the tail fan has any significant features or not, but that's a beautiful shot. Thank you. Look at that. Spines right in front of the eyes there.
For it.
A curved rostrum pointing forward. Sometimes, of course, you have to look at the mouth parts of these animals in order to verify the identification. But I'm hopeful that Kevin will be able to give us a name. He says we just need the head. The spines on the carapace here are characteristic. So he's checking it out for us.
Video's clear.
Pilot's clear.
Okay. Let's see if we get a name on it.
It's pretty soft stuff. Just buried that toe.
Yeah. Looks like it's stuck to it too.
Okay, guys, the ship move is complete. So let me know when you're all set for another move. We'll be going 2-7.
And what have we here?
5-ish.
Okay. Yeah, we can keep them rolling in as much as you feel comfortable. I don't know how much layback we're getting today, but
not a ton, I don't think.
Yeah, I'm fine with it.
Sirius is just right off the transom there.
That's another
let's do 3-0.
cobble of this blackened
What'd you say the
carbonate
heading
rock.
was then? 275 or 285?
Come in partially, yeah.
And Kevin suggests that
copy that.
That armored shrimp, the Glyphocrangon, looks like the species Neglecta. And this is more of the same kind of orthogenic carbonate rock that we've been seeing. Thank you.
Yeah.
With its resident squat lobsters and small anemones.
Nav, Bridge, go ahead.
Can we do another ship move, please?
Yes, sir, go ahead.
So I'm just going to keep my face sort of pointed this way into the slope for now.
Yeah, that's fine.
Bridge copies.
That's great.
I venture off too far to the port, just let me know.
Is that a whip there below the fish?
Stand by for input.
Like a fish in a hole?
There's another rat tail.
Can come in partial for ID.
See what that little, yes, this is, what is this? Well, it's, looks like a worm tube that's covered with growth of some sort. Can't quite tell what that is. There's a lot of sediment on it.
ROV Nav, Bridge, move initiated. 30 meters, bearing 275, speed decimal 2 knots.
I'm trying to stop my
You're welcome.
lateral. Sneak in on it.
No. I'm not sure what that is. Does not look like an octocoral.
A little bit closer.
It looks like it's overgrown. Probably one of the ciboglinid, yes, one of the ciboglinid chemosynthetic worms. Very good. Thank you very much. I'm not sure what those things growing on it are. They may be agglutinating foraminiferans, but I'm not certain. I see some small polyps, which are probably hydroids on the underside, about halfway up in the screen now. Very tiny little polyps, so that may be all of them. All of these may be a form of hydroid that is growing along here. They'll grow along runners, and then the polyps will pop up from along the runners at intervals. Thanks very much.
Yep. Clear when you are, video.
Both Bob Carney and Carolyn Ruppel suggest that some of those large blackened lumps we've seen might be asphalt or asphalt-like.
I'll fly near it and see if it chimes in, otherwise we'll keep going.
And who is this lonely fish sitting on the bottom here?
Mini tripod.
That's only about 3 inches long. That's pretty impressive. It looks like a short fin tripod fish of some sort.
Good.
Doesn't seem to be a lot of interest. Do we want to keep going?
Yes, that is a tripod fish, a very short one. And are you ready to introduce yourselves, gentlemen?
Yes, we are. So for the lunch swap, we have a new shift in the front row and at clipping. We'll go ahead and introduce ourselves. Sitting pilot currently is Jeffrey Lanning. Sitting co-pilot.
Our sitting navigator is Chris Ritter.
Co-pilot is Don Libertori.
And just off camera to the right is Roland Bryan in video.
And we have, and we're all here with the Global Foundation for Ocean Exploration.
The other voices you'll be hearing on the dive today are myself. I'm Diva Amon, and I'm a research fellow at the Natural History Museum in London. I'm one of the biology co-leads on board this expedition of the Okeanos Explorer, and I tend to specialize on chemosynthetic habitats and the animals inhabiting them, as well as our impacts in the deep ocean.
And this is Chuck Messing. I'm professor at Nova Southeastern University in Fort Lauderdale. I specialize, I'm an invertebrate zoologist, and I specialize particularly on the sea lilies and feather stars, the crinoids, but also deep sea coral communities.
Hi, and I'm Lauren Jackson. I'm from NOAA's National Centers for Environmental Information.
And that's another one of these Galatheid squat lobsters in its little hole.
Seems like most of the burrows have been full of squat lobster today versus yesterday being empty.
Gonna call that a
one
complete
weird
move.
fish.
Copy, thank you, Bridge.
I forgot about that fish. That was weird.
Okay, guys, move is complete. You ready for another one?
Yep.
Keep the same heading. I'll do another 30.
Okay.
Nav Bridge, go ahead.
Hey, we'd like to repeat that last move. 30 meters for range, 275.
Les Watling from the University of Hawaii has joined our chat room.
Meters, bearing 275, speed
He's a corals expert.
Stand by for
He's had, we have Kevin Rademacher, who has weighed in on the identity of the shrimp. Carolyn Ruppel is a geologist. Robert Carney covers the waterfront. He knows a lot about a lot of different things. That's another one of our glyphocrangon that Kevin identified as neglecta. And we've also had Ken Sulak, ichthyologist, who has been studying the fishes of this part of the world for several decades.
Yeah, when you get a chance, can you
he and I were in
drop my
graduate
sonar
school
down to
together.
50, please?
Sure.
We don't want to keep it scanning out for.
Yeah, I just bumped it up from 50.
Additionally, in the chat, I can also
Just
see
to
Amanda
kind of see
Demopoulos,
what's on
who's
the
been
horizon.
science
I'll bump it
lead
back.
many times
We're
on the
still
Okeanos
down to
Explorer.
this
She's
bowl,
also
I
at
think, huh?
USGS. Andrea Quattrini, who is at Harvey Mudd. She knows an awful lot about the deep sea corals and fishes of the Gulf of Mexico. We also have Asako Matsumoto, who is joining us from Japan. Thank you very much, Asako. And she is a cnidarian expert, coral expert. We've got Eric Cordes, again, another person who has, studied the Gulf of Mexico in many different ways, but especially cold seeps and deep water corals. And Nolan Barrett, who is a student at Harbor Branch Oceanographic Institute and works pretty closely with Shirley Pomponi, who was one of the bio leads last year. Thank you very much to everyone for joining us. We do appreciate having you virtually on board with us. Oh, and I forgot to say Scott France is also with us. Sorry. Scott France is also with us. He is at Louisiana State, Lafayette.
Lafayette,
yeah.
Yes.
And he's been pretty integral in all of these Okeanos Explorer expeditions in the Gulf of Mexico this year. Well, this being the first of 3. And then also we're joined by Tina Molitsova, another international collaborator who is joining us from Russia. And Tina is an expert on many cnidarians, but especially black corals. So again, thank you all for joining us, especially those who are in some ridiculous time zone right now.
Yeah,
yes, indeed. Just reflects again the wonderful nature of this operation, where Diva and I, with some experience in different areas, have now access to this enormous... Ooh, what is that? Look at that.
Also, sorry, Scott France is at University of Louisiana at Lafayette, not Louisiana State. My bad, sorry. Pilot, if we could get a look at this colony, that would be great.
Look at that. That's a first for the this trip, this dive.
Just gonna
see
stay
what
within
that
the
is.
length of my leash, look around in this immediate vicinity. We'll reach that in just a second.
So it looks like we've come down this slope and come upon quite a couple of very large colonies of various types of corals. We're not sure what that white one was that we just passed, but we will be going back to take a good look at it. But it does look like we've got some of the yellow Paramaricia, some of the purple Plexaurids, which we collected earlier, and we're not quite sure of the identity of. And you can see a lot of them have their commensals as well. So we've got lobsters, serpent stars.
And they're all of those fans are oriented. They grow in response to the mean flow direction. They generally tend to cup into the flow, which suggests, from this standpoint, that the general flow might be from right to left. That's going to be really interesting to see what this coral is. This is a stony coral for sure, a scleractinian. That knob coming left of center is a contracted sea anemone.
We can also see some of the stoloniferan purple coral that's encrusting those rocks, carbonate rocks off to the right.
Okay, video.
So that looks like it's pretty zigzagged.
So I'm still not 100% certain when I see these whether this is. Well, we've got three choices. We've got Madrepora oculata, Solinus milia, Variabilis, and Lophelia pertusa, and I don't think it's Lophelia because the calyces are not that exert.
Both Eric and Amanda are weighing in and saying this does look like Madrepora. It is quite zigzaggy, but this looks pretty different to the Madrepora that I've seen from the Mid-Atlantic Ridge. But I am not very experienced in corals, so maybe there's like a different growing morphology here in the Gulf of Mexico to there.
And they have, these corals will exhibit a wide range of growth forms, not just perhaps geographically, but local environmentally, depending on the flow regime. For example, we get much more densely branched forms of this. This is a very spindly version relative to the ones that I've seen, and there are two shrimp there and a squat lobster on the left. And the polyps are out and feeding.
That's great, Pilot. Thank you.
Full screen.
And you could have seen there that those the tentacles on those polyps did not have the little side branches that the octocorals do. And all of that is dead and not quite dead branches down in the center there. Some broken off. Be interesting to know how those broke off, whether a large fish came by and knocked them off.
I think immediately above to the right
or
of
whether they fell off on their own.
Cup coral, right
maybe some
now.
bio-eroding organism in the lower branches weakened the branches.
Okay, I'm going to reacquire this sort of hard substrate and sidle along and see what else there is to see in it. It's really overgrown with this.
A lot of those stoloniferans here. Oh, can we have a look at this
sponge?
White, yeah. I'm not sure if it's a sponge. It might be some kind of soft coral.
That'll be interesting. If it is a sponge, I think it's the first glass sponge we've seen on this dive, yes?
Yeah. Yeah, they've definitely been
line up in the current a little bit.
Pretty rare so far.
And there's a lot of dead coral branches right here.
Mm-hmm.
And yes, that is a glass sponge. Faria, I think.
Our pilots know what they're talking about, by the way.
Very nice. That's...
Dock to the lower right.
Yeah, just beyond the view below and to the right. Now it's about halfway up the screen.
Top right corner.
Oh, I see.
I think that's probably just a closed anemone.
Anemone, that's probably
But then there's a a baby Anthomastus or Pseudoanthomastus.
Could be.
Just to the left of the screen.
Polyp. Yep.
Oh, yeah.
It's just one
Bottom
single
left.
polyp.
Yeah. Jeez, it's so small.
Yeah, you're right, Diva. That looks like a contracted anemone.
Mm. There's also a corallimorph, potentially. Oh, a couple.
Yep, those little pale, transparent gray anemones on the left side. Could be corallimorphs, that's right.
And then just in front, can we get a better zoom on this bush-like animal? Just center screen now. It looks like a,
Do we have any more, or should I
tree.
sneak closer?
Oh, there we go.
Those are hydroids with a gooseneck barnacle attached. These look like plumulariid hydroids. And small anemones or possibly zoanthid polyp down below. Next to the corallimorph on the left. So these, structures has got a main axis with side branches, so each one of these looks like a feather, and the side branches of the feather are lined with extremely tiny polyps that look like, for all the world, very simple anemones. And those white dots are the reproductive structures that have their own special little housing along the branches.
Yeah, of course. When you've got what you need, video. Some debris.
And a starfish at the bottom.
Sorry, what, Roland?
So there's a starfish went off bottom.
Oh.
White.
I'll back up. Have you seen that yet today? Watch the sea star at the bottom of the screen. Like, it might be a piece of white coral tricking us, but Roland spotted it. There it is.
So here we've got a sea star. I am not 100% sure what sea star that is yet. Perhaps Henderson?
Maxim.
Copy. Sneaking in a little.
And I'm guessing all of those small polyps, that looks like another kind of stoloniferan. It's an octocoral. You can see the pinnate tentacles. The tentacles have little side branches on them. And it's not got an erect stalk. All of those polyps arise from runners or stolons, and there are a couple of little brittle stars on the left-hand side sticking their arms out, little white arms sticking out from within the rocks. And much of this looks like coral debris, dead branches.
Video's clear.
Pilot's clear.
All of this dead coral looks like dead Madrepora, so at some point there would have been some very large bushes, or not bushes, colonies of Madrepora, that calcareous coral that we saw at the beginning of this slope.
Good eye, thanks.
Yeah, we can do another move that way.
There's also some fishing line, it looks like, caught up.
Okay, we're going to do 40 meters, 275.
Ooh, big jump.
Let's do 20. In case we see another one of these.
Yeah.
You seen this bushy Chrysogorgid or something here?
Yeah, we've seen this Chrysogorgid already.
Yeah, there's a Chrysogorgia poof on just off the bottom. We've seen that before, and more of the purple encrusting stoloniferan octocoral at left.
Bridge copy is 20 meters at 275
And that orange one in the middle might be a Swiftia again.
Oh, stand by for input.
We can snap to the possible Swiftia. See if that's what we think it is. Come on in.
I think so.
Is that enough to...
Yeah, I think so. That's most likely that Swiftia.
I can get closer if we need.
It's a bit more planar than the other, but that may be the habitat. Not quite as bushy. Thank you.
Come down a little bit, give you a little bit more.
It's okay. I'll just come around.
Okay.
This direction. That way. Make some leash. Oh, there's a fish hiding right there above the lasers. Come in on the fish. Let's see what that is.
Ah, that's a good eye, pilot.
You thought you could hide from us.
I have no idea what that fish is.
That's what we go for.
Indeed. Above it, you can see the encrusting octocorals. Polyps were contracted, some of them. They look like little lumps.
I'm settling in. You can start in again.
A little tilt. Max soon.
Copy.
So this is definitely not a rat tail.
I was afraid if I got too close it would be
As
too
far
dark.
as I can tell,
Yeah.
it doesn't have the longer anterior fin rays. It's got a couple of chin barbels.
Hey, co-pilot.
Go ahead.
I want to see if you can deploy the starboard swing arm a little further.
Sure.
And maybe light this up. Or the port one. I'm not sure which.
Okay.
Would be best, but see if one of those can work a little magic for us.
Okay.
Kevin says it's an ophidioform
yeah,
fish.
let me go inboard on the port one, maybe.
Just be careful with that.
Yeah.
So that can put it in one of several
Can hit the
families.
shilling,
Yeah.
Let's go in
You
a
got
little
it.
bit.
Yeah. You could also deploy a little too, I think.
And
A little
those
lower.
chin barbels,
yes.
apparently, according to Kevin, that's actually the
Nope, a little
pelvic
bit
fin,
too far. All right, let me
fin rays, which,
take it up
in,
easy.
as in some other fishes as well, they have migrated, if you want to call it that, evolved so that they are placed anteriorly and function as little bottom-feeling antennae, hunting for prey
Try
in
going
and
down
on
a
the
little bit,
substrate.
too, with the, or actually up, I guess. No, down. Right? We're all the way down. That's. Never mind.
Got a little fringe on the snout there too.
I think we've got everything
above
they
his
can use to identify it.
nares, above his
Okay,
nostril.
I'm clear when you are.
Thank you.
Okay. Back outboard.
Picking up. Next, go pilot.
And up.
Super.
So an encrusting octocoral like this clavularia, this
RV nav
purple
bridge,
encrusting
that's a complete move.
octocoral, and some of the others that are low growing can reproduce asexually by fragmenting. Basically, a piece
Yeah,
of
guys, that move
the
is
colony
done.
can separate
That move is done.
due to some physical event, the rock breaks or something like that. And so you'll end up with two colonies that can grow independently of each other.
Throw eels straight ahead, line.
Okay. Yep, I see it.
So here we're beginning to see a lot more of these carbonate outcrops. It's much less fragmented, and we're
Big
beginning
hole.
to see, full-on beds
Yeah.
being exposed. We can also see a lot of this coral rubble around the base here. Again, perhaps Madrepora oculata, as we saw some earlier.
And those broken branches look relatively new. They're not discolored yet.
This is just so contorted, these rocks.
It
Yeah,
looks,
it does.
it looks
Yeah.
like
Almost looks like
it
a
looks
lava
like
flow.
cooled lava.
Yeah. Yeah, it really does. And just to remind you, we don't only have one set of cameras down here. We've also got Sirius, our ROV, which sits above D2. D2 is the main feed, so that's what you'll be seeing on camera 1. Whereas on camera 2, which you can switch over to, you get the view
Coming on
from
this guy.
Sirius, which is a fantastic, much larger view of the scale of things with D2 usually in frame. So we can actually see that this feature that D2 is focusing on in relation to the vehicle.
And this, I guess, is a synaphobranchid eel.
Yeah, so I think
Good
this is
view
the
of
one
the,
that Kevin was referring to as
stuff.
Iliopsis. That's probably not pronounced well. Iliophus, sorry.
I think we've probably agitated this guy enough.
That's rock, or is that a tar flow?
This?
Yeah.
Sure.
Where's that white thing next to the crab on the right bottom?
I don't know.
So Bob Carney's suggesting that this is actually asphalt, which you could definitely understand. It does look like asphalt, some sort of, bubbling out. Oh, there's a very big polynoid down there.
Ah, scale worm.
So in Trinidad and Tobago, where I'm from, we have huge reserves of oil and gas, but we also have quite a lot of asphalt, natural asphalt seeps. And some of those are on land. One of the biggest ones, we call it the Pitch Lake. And surrounding that area, you do see extrusions of asphalt similar to this. And they do have these very, structures that look very similar to this, but then the actual lake is just this ever-growing asphalt area, which you can dig out a bit of it, and the next day will be completely filled in. It really is a very strange, and it means that the roads in the area, you can make a beautiful straight even road, and then before you know it, a couple of weeks later, it'll be all contorted up and difficult to drive on.
The feature we just came across, this is Carolyn.
Bridge copies.
Sort of flow.
Next move, 0 meters, 75 speed.
Wait, Carolyn. Oh my God, this is amazing.
Cool.
Stand by for input.
So this is the first time, sorry Carolyn, I'm so sorry to interrupt you, and you can probably explain this a lot better than me, but this is the first time I'm seeing asphalt seeps.
Nav bridge, go ahead.
Those are bubbles of asphalt. Those are
upward. Stay put.
Copy.
So all of this black stuff is asphalt, which would be molten.
This is an amazing...
They're in the frame.
Yeah.
No, go for it, Carolyn.
Definitely make sure someone gets a clip of this. This is beautiful.
Do you want to give us a bit of a geological explanation as to what is happening here? Would you mind?
I don't think Bob's not on the line.
No, he's not. You're the only geologist on the line, but that's not the reason I've asked you.
Zero lights.
You've just been giving really great explanations so far.
I don't know a lot about asphalt, but like any other sort of hydrocarbons, they can seep out, and that's what you're seeing here. But you're also seeing they're very viscous. So you'll see some of them in the current. We just saw one a second ago. You see how they're kind of probably filling from behind, like these sort of blobs. But as you know, when you lay a road, if you lay it in cold weather, it's very viscous. So probably what's happening is these sort of bubbles that we're seeing coming from the seafloor. This is just amazing. They may have some gas associated with them, given its morphology, or maybe not. And it's this material that's just sort of sitting there holding onto the seafloor. I'm guessing
some of this, look, it's kind of dull,
break
like
off.
it's been sitting like that for a while.
What I started to say before was when we came over that one outcrop, it was clear that we were... and
keep panning around, like sort of in this zone to the right. Maybe we'll find more liquidy formats, maybe even bubbles.
In those rocks. And in fact, Bob and I've mentioned a few times the asphalt possibility here. I don't know whether it's been found in this leaf block, in this area, in Flower Garden Bank, in the Gulf of Mexico before. I Googled quickly. I didn't find much evidence, but Bob would know better. Maybe he'll tell us in the chat room. Okay, and Bob just put up active spot on a much larger asphalt flow. So what he's saying is there's a big flow that we flew over, if you will, and that this is sort of the most active spot now of the asphalt coming out that sort of land on the seafloor.
The edge of it there.
Yep. Shrimp on the left.
Absolutely amazing. Thank you so much, Carolyn. That's really great. But some of these morphologies of these very viscous droplets, you can see where they've sort of stretched away to release, but haven't because they've been so viscous. And some are actually kind of like fishing lines almost, swaying around in the current. So the only time I've seen something similar like this to this, which I wasn't on the actual expedition, I've just seen a video of it after, is at the Corophenoides, which is also in the Gulf of Mexico, but in the southwest of the Gulf of Mexico, actually within Mexican waters, I'm pretty sure. And there, you could actually see in some places these droplets, breaking off, and these very strange morphologies of all of this asphalt, which is oozing out of the seafloor here.
And it's on top of that, despite the nature of this, we still have squat lobsters all over the place. I saw numerous of those palp worms, which folks suggested earlier were ketopterid worms in the family of parchment tube worms, waving their slender pair of palps in the water. And, of course, an urchin. So they don't seem to be dissuaded by the nature of the substrate here.
So
Would these
those
be
who are
like
in the
newer
dive,
seeps since there's not as much stuff around them? Like the far
we
right
did come across
dot.
that beautiful asphalt field. Jimmy Austin
See
was
that
narrating
dot
that.
just on the right?
And it was
Oh, yeah,
flowers
There's not
that
any matter around it?
were-
Yeah, right.
My
So that
guess,
means
I
they're
don't know
new,
if
like,
it's true, but my guess is that would have been slightly
formations
less
of seeps.
viscous
Or
asphalt
maybe it
and
has
richness,
lesser.
because what we're seeing here just
Yeah.
definitely looks like
Of
it's
the
got a very
gaseous
high
flow
viscosity
coming
kind
out.
of being held there as these individual
See,
droplets come out.
bottom of the screen.
Okay, and
left-
Bob Carney
left
just
of the-
posted on the chat
Yeah,
that
the
German
small
workers
fish
had the
looking
idea
thing.
that
Yeah,
these were very
purple
hot flows. Apparently not due to a lot of volatiles mixed in with the asphaltenes. But BP has sampled and analyzed some of these in the past.
Thanks very much for that, Carolyn and Bob. There's another one of those fishes.
Guess they've seen it.
So I'm just looking at the paper on the Campetionals, which was found in the southern Gulf of Mexico.
I
And
haven't.
similarly, they describe these as being lava-like flows, just like Chuck said, of solidified asphalt. And in that area, they covered more than a square
Should lasers
kilometer,
go across
which
this
is, of
fish?
course, quite a lot. But that site was a lot deeper than
lasers?
here.
Yeah.
Currently,
Sorry.
we are at 1,100, nearly 60 meters depth, whereas those were at 3,000
Pull you
meters
up to a
depth.
medium wide
And
shot.
just like
Look
here,
around.
they saw some chemosynthetic tube worms, which we did see. I'm not sure if we can find some more in the area. As well as they
Full
found
wide.
many bivalves colonizing the area. And just like the asphalt here, it appeared to cool and then contract after discharge. And so that's why we're getting these globule-like protrusions from the seafloor.
Yeah, we can look around in the vicinity.
And they
see more of this.
suggested that
Put another,
asphalt volcanism
maybe 10 meters
does
in to get
create
us
a
closer.
habitat for chemosynthetic life that could be widespread
So you can-
in
looks like
the Gulf of
everything's
Mexico.
going
And so
that
this
way
is
and
a
up.
great example of yet another one of these habitats. Yes, the chemosynthetic nature isn't developed to a
us
big
closer.
degree, a large degree, but we definitely did see some tube worms in the area. That was absolutely amazing. That's the highlight of my
Purple.
expedition so far.
Now,
For
Bruce,
sure.
go ahead, sir.
I'd like to get a small ship movement, range 10 meters, bearing 275 degrees, speed 0.2.
Bridge copies.
What I found
10
fascinating
meters,
was the
bearing
different
275,
shapes
speed
of
2 knots.
those,
Good copy.
Stand by
I guess,
for-
semi-liquid asphalt drops, protrusions. Some of them were bulbous, as if they may have had some gas inside or something, or were certainly less
Or more
less
buoyant.
dense.
Yeah.
Yes. And then there were ones that trailed to a fine filament. Very different.
Some more fish.
There's some more dead
Nav bridge move
probably
initiated,
madrepora
10 meters,
branches
bearing
and
275, speed
yellow
2 knots.
Eurythia fans,
Copy, thank you,
and
Rich.
more
You're welcome.
of the
It's all laying
purple
down, right?
Yeah, it's like
stoloniferan encrusting octocoral and
Tilt
more
center a
gracilichinus
little bit HD2.
sea urchins, the white sea urchins. We
Got
see
it.
there's what looks like more coral growth in the distance there on some elevated substrate.
It's so funny. In this paper on these asphalt seeps, they actually say the morphologies of these deposits were often blocky or ropey, similar to or Pahoehoe. That's not pronounced right.
Ahoy
Pahoehoe. Basaltic lava flows, just like we were saying.
What incredible insights we bring to these observations, don't you think? I may be overstating that a little bit.
And some more of these very large colonies, at least for what we've seen during this expedition so far, of plexaurids and these encrusting stoloniferans and chrysogorgids and acanthogorgids around colonizing these
Just doing a
fish
scan
on your
carbonate
left toe of
outcrops.
HD2.
Rat tail. Okay, I'll push on ahead here.
Okay.
Just scanning how far this goes and make sure there's something to see.
What looks like a rat tail just about to go off screen.
Bridge,
As well as
move
we're
complete.
coming upon this lovely little hill here
Nice
with
little
these
pirouette around this, make
quite
our
large
coral
colonies.
editor happy.
Pilot, if we could get some good shots of this, that would be great.
Of which?
Both wide angle and focused, please.
I think the coral community.
Okay. Start uphill or up current from it a little bit so I can drift and stay in the shot. Okay, you want to come in a little bit, just sort of frame the area.
Oh, that's lovely.
And some more of the glass sponges as well in between.
Yeah.
And most of this on, or at least some of it on, all growing on dead coral branches. Chrysogorgiidae on left.
Sponge right where the
Glass sponges, the purple stoloniferan clavularia, and these tall, pale yellow Paramuricea octocorals with their resident ophiuroid serpent stars and chirostylid squat lobsters, and all oriented in the same direction, reflecting the pattern of mean bottom flow.
Want to kill the lasers?
That, of course, doesn't mean the current always comes from the same direction, but overall, in the majority, it must to direct that growth pattern. And my observations are limited, of course, but I have never seen the this particular kind of squat lobster, this Gastroptychus, off the sea fans on the sea floor.
Yep. Yeah,
and I've only once seen one of those serpent stars just at the very base
on the one with the lobster
of one
and
of the
sort
fans.
of pirouette with the tight.
Otherwise,
which right here,
right where the lasers are,
always up coiled in the branches.
So sort of just show both sides of it.
Okay, losing the lasers.
I'm in trouble with pirouetting in this current orientation. The current is not agreeing with me. When I push through it, it makes me rock.
And again, dead bivalve shells as well.
We'll get it. Yeah, I'm going to get this the way you want it, Roland.
It's probably cereals pulling on you.
Oh, that's probably right. Thought I had more tether than that. Yeah, Roland, I can't actually get all the way around this.
Copy.
Sorry, buddy. Yeah, that's the tether spinning me off. Just trying to figure out.
Like that. And
pull wide.
What is it doing?
You can turn the lasers back on for size.
Thank you,
here.
pilot.
Yeah. Yeah, we'll get some scans of the lasers here so we can get a good idea of size of these colonies for you.
How about a super tight of the coral coming under lasers now with the big squat lobster on it? It's a super tight shot.
I can move you in tighter too. Yeah.
Working to ensure that we have enough good coral video for highlight.
Yeah, come on in.
Copy that.
Nav bridge, go ahead.
And can we do another ship move, please?
Roger that, go ahead.
Range 15 meters, bearing 275, speed 0.2.
There are also some very small shrimp in these corals, so just this colony in front of us is providing a home to 3 different species. The Gastropticus squat lobster, which is the Charostelids, which there's more than one of. There are also some tiny shrimp, as well as these Astroschema serpent stars. Absolutely wonderful footage. Thank you.
Nav bridge, move initiated, 15 meters, bearing 275, 2 knots for speed.
That's clear.
And our depth now is 1,156 meters.
And we are at a site known as Henderson Ridge, mid-south. This dive site is in an area that may be proposed as an expansion zone for the Flower Garden Banks National Marine Sanctuary. And so, of course, this dive has very large management implications. The data that we collect on the communities, the habitats, as well as the geology of this area will feed back into that process. We were hoping to see a lot of corals, just like we have on the screen, because this area was predicted by various models to be favorable to deep sea corals. So again, we are helping the southeast deep sea coral initiative. And a lot of that information is going to be really useful to them. And as well as these coral colonies, which are living on these orthogenic carbonate deposits, as well as what we just saw, some asphalt extrusions, we also were hoping to see some methane seeps, because last night when we were mapping the area, we came across several bubble streams. And that usually indicates the presence of a cold seep. But they are not easy to find, but we are seeing that this entire area does appear to be pretty geologically active. We've seen various places where there's a lot of reduced sediment, a lot of bacterial mats, as well as some chemosynthetic fauna in pockets. At that asphalt seep just a second ago, there were some tube worms. Before that, we'd seen a lot of areas of dead vesicomyid clamshells. So this does indicate that this entire area may be quite active, or was at some point at least, and still is in some places. But now it's provided this wonderful hard substrate for all of these corals to attach to. This area has never been explored before. North of here, there have been a series of deep submergence dives with ROVs and AUVs and submersibles, but never south of here, where we are, well, south of that area where we are exploring today. So all of these images are completely new to us. We didn't know what we were going to find when we got down here, but it has been a truly wonderful dive so far.
Watch lead, Nav.
Go ahead, Nav.
Hey, I found Seep 5.
Oh.
So, it's right on the way from Seep 4 to waypoint 3, just kind of southwest.
So how far away is Seep 5?
It's a ways away. 500, a little over 500 meters.
Okay, and we have...
We've got 3 hours left.
3 hours left. Okay,
yeah.
Just that helps to gauge things. Thank you.
Sure.
So as we showed yesterday, when we had a similar scenario, we had these bubble plumes that we had tracked up in the water column, but we didn't know quite where they actually escaped from the seafloor, and that of course would give us the location of these potential seeps. So the points we're working with are just kind of rough estimates. They're meant to guide us, but of course searching in the deep sea is not difficult. We're able to narrow down the scale in which we are searching from first by mapping and seeing what geological areas stand out, as well as by having seismic data, which Boehm very graciously gave to us, so that we could see potential areas where there was gas upwelling. And so then after we are able to find those areas, we can then run over it with the ship's multibeam, which can then detect bubble streams when we're looking for seeps. And so that again can narrow down the scale of the area which we will be looking with our ROV. But it's still not easy. It's, of course, this habitat is completely dark. We have a visibility of, I don't know, 20 meters perhaps. And so it means that we're basically wandering around in the darkness looking for a habitat which doesn't particularly stand out in the darkness. So it's not easy, but yesterday we did find that there was a predominant south to north, current. And as a result, we did find one small seep yesterday, which was actually located north of that bubble target. And so it is thought that when we saw that stream of bubbles in the water column, at that point that we saw it, it had already been pushed north by that current. So perhaps the same thing may be happening today. But of course, hunting seeps isn't our main priority on this dive. Our main priority is looking for these incredible deep sea coral habitats that host such inhabitants like the one we have on screen right now.
Copy that.
That's the beauty right there.
Thanks very much. That was great.
Bridge, move initiated, 25 meters, bearing 275, speed, decimal 2 knots.
Good, copy.
This again is Swiftia, one of the octocorals. And as we move along, a little quote from Patrick Stewart.
So
let's see what's out there.
Excellent.
What is?
Yeah.
Oh.
Sure your eyes are not just getting out of sync with the clock.
Time travel.
Of course, all of our depth measurements, both below the surface and altitude above the sea floor, are metric in meters, and our lasers are 10 centimeters across. That's metric, of course. The entire scientific community, both in the U.S. and abroad, uses the metric system. But it's interesting to think, when I first began working in graduate school towards my degrees in marine science, biological oceanography, many of the depth records at that time, which was several decades ago, were in fathoms, which was the traditional depth measurement, fathom being 6 feet. So first we had to translate from fathoms to feet, and then we went to the metric system. We translated from fathoms to meters, or feet to meters. So just as a gauge for you, there are 1.83 meters in a fathom, and there are 3.28 feet in a meter. So tuck that away.
Yeah. Seep and ye shall find.
So a meter is just over 3 feet. And a fathom is just less than 2 meters.
Some more monofilament there. Yep. On the bottom. Just going off screen now, you see just the tail of it's on the bottom of the loop, about that big. Ah, sorry, yeah, it was to the right.
And our current heading is just a little south of west.
So pilots, we're going to be
now coming more to the west.
Coming in and doing a fishtail here,
There's
coming down to the southwest.
One of those probable
So,
hormathiid
we're right over
Venus flytrap sea anemones.
now, so
I wonder why so many of them have been contracted.
just Sirius won't drift one way uniformly for a little while.
Okay.
Now turning to northwest. And Diva, correct me if I'm wrong, we are basically due south of the Mississippi Delta?
It might be because
Or
we're entering a depression
still
here.
east of
See,
that.
This is like, then again, it could be tidal as well.
Lauren says we're a bit west of the Mississippi Delta. West,
Yeah,
well,
so
of course,
maybe
south
it's
of
kinda
it.
swirling down here.
Is that a potential stingray or fish top of the screen?
It's all right.
Plant material.
And we are now moving over
ROV
mostly
nav
open
bridge,
sediment
call that a complete move.
towards our next,
You're welcome.
the next seep record that we have. That's our estimate of where the next one might be. And here's another water hyacinth that swept out of probably the Mississippi. Interesting that of all of the terrestrial vegetation or freshwater vegetation we have seen on any of our dives, this is by far the most common. I think we saw one other item apart from the sargassum weed, which of course is marine, floats at the sea surface. But water hyacinth is the only freshwater
So
plant
D2's current heading will be more like what we wanna
remains of a
do,
freshwater plant that we have seen.
So if you want to, it'll be 235, 230.
Copy that.
So
So I just want to re-emphasize
when you're there,
that
call in a move.
how much differing substrates can help to increase the biodiversity of an area. So of course we started off in an area that was pretty soft sediment, and while that of course does have its own fauna, many of which may be infauna, so animals which burrow down into the sediment,
Tilt up and look side to
there
side in this
didn't
area.
appear to be that many deposit
It's a nice,
feeders, you know, sea
vantage
cucumbers
point.
and other types of, wow, look at this.
That's beautiful. Look at that grove.
Oh, and all behind it as well. Okay, we're going to have to spend a lot of time here. I'll continue on that comment
Just give us
a good while later
side-by-side
once
here.
we've passed all this by. But I mean, this is an example of what I was going to say. These asphalt outcrops or carbonate outcrops or just any kind of hard substrate, these little islands we're seeing, do create these little oases of life for hard substrate dwelling animals such as corals and sponges. We haven't seen that many sponges today, but wow, if you look off in the distance, there are loads of these very big colonies. And if you look at the lasers, those are 10 centimeters. So many of these are well over a meter in width.
It's quite not quite a forest, but a grove.
So we're just climbing the the local high in the area. And so a reason that these corals could be
can
dwelling
fly
here,
over some of the
apart from the
beauty
fact
part.
that there are these wonderful little outcrops of asphalt and orthogenic carbonate that they live on. Here, I think this is actually asphalt, but they have that hard substrate they need to attach to, but also being on the topographic high, they are pushed up into the water column. Here you can see this current rushing by with all the particles. And so they'll be in areas of slightly higher current, and that will bring much more
Corals would
food.
indicate they're
The
coming from
particles
behind
which
the coral,
float
right?
by, which these corals rely on, to them and past them. So they will be in a much better position for thriving, given those two variables. If that current were to get too high, of course, that could work against these corals. But here it seems to be a perfect little medium. And that's the reason we found these very large and abundant communities.
And one thing I don't think we've mentioned about these colonies, and that goes for the stony corals, for the octocorals, for the hydroids, and that is all of the polyps are interconnected so that they are basically little, well, they are colonies, but you could view them also as somewhat as socialist communities, because the food that is ingested by some polyps, some of that is shared by the rest of the colony. And of course, many of the cells of the tissues that connect the polyps cannot feed for themselves. So they rely on nutrition captured by the feeding polyps, which is distributed among the entire colony. And of course, you can imagine that the polyps adjacent to the sea floor may not get as many get as many food particles contacting them, being ingested by them, as the ones up at the top of the colonies. So it is obviously, this sort of thing is a situation where each of these polyps are connected and they all rely on each other. And of course, they start out as a single polyp that grows and divides asexually. And here's a beautiful anemone. And look at that, in the middle of its oral disc, its mouth is elevated as a slit. Haven't seen that quite before.
I remember seeing something similar during Capstone, the 3 years in Capstone, but I can't remember what exactly they were.
And I mentioned the other day that these anemones have muscles that can contract them, and when the muscles contract, the water in the interior is pushed out through the mouth and sometimes through pores in the sides of the anemone. But in order to re-expand, to re-inflate, these anemones don't have antagonistic muscles. They have a groove at either one or both ends of that slit-like mouth that has cilia, and the constant flow of this constant whipping of the fine microscopic hairs generates a current of water down into the interior cavity of the anemone, which fills the entire interior. It's called the gastrovascular cavity because it functions both in digestion and in circulation. So there's only a mouth, there's no anus, and the remains of any digested meal that are unwanted have to be released out through the mouth.
So this could be from the family Intecnernidae, and perhaps the genus Actinernus. Those do seem to have that sort of elevated mouth. But in that area there was a solitary cup coral in the background, some of these very large Paramoricea, and then of course all of the commensals which we've been seeing throughout the dive, these chirostylid squat lobsters and the serpent stars, and there are probably some shrimp down in there as well. But also around you can see these deep purple stoloniferans. So those are the ones connected by a ribbon. The polyps are connected by a ribbon.
The skeleton that supports these octocorals is made of a particular kind of protein material. It's sort of similar to fingernail, although not identical. But then surrounding that is a layer, one or more layers in the tissue that covers the skeleton of microscopic, sometimes not quite microscopic, bits of calcium carbonate called sclerites. And the individual polyps may have sclerites in them, even in the tentacles, or as a lining along the shaft of the polyps. And these sclerites are have to be examined under the microscope. They are frequently very important to the taxonomy, to the identification of these sea fans. And so it's a combination of characters, the branching pattern of the sea fan, the nature of the axis that supports the sea fan, and the nature and distribution and forms and ornamentation of the sclerites as well.
So, interestingly, I just spotted 2 tube worms behind. Which seem a bit out of place, but then when you think these are potential asphalt extrusions, it makes a bit more sense.
And
so
what
it looks
is that spherical thing? Is that a fish?
No, that looks like a jelly.
A jelly. That looks like
Paraphila.
Paraphila heading in the wrong direction. This is a midwater jelly in the group of medusae called the coronates, because you can see it has a circular groove around it. It's got a conical umbrella or bell, and halfway up that bell is a groove that's characteristic of coronate medusae. And then the edges of the umbrella are scalloped into those great big flaps. And this is a midwater jelly. And you can see also the sort of an interior darker cone. That's the gastrovascular cavity or stomach, basically, in this jelly. And it's black, it's opaque. And one of the hypotheses is that because many of the prey items of this jelly are bioluminescent, once they are eaten, they are not quite dead yet, and so they may flash. And if that belly, if that interior gastrovascular cavity was transparent, they might advertise the presence of the jelly to other predators. So the hypothesis goes that maybe this opaque, dark lining of its stomach shields the interior prey, bioluminescent prey, from advertising the presence of the jelly to any potential predators.
Could we even look at that bright white colony? I don't think we've seen any, not bright white, sorry, yellow. We don't think we've seen any like that so far.
The whole character of the rock here, you can see how it looks very as if it flowed out. So it's very indicative of asphalt.
So this is asphalt, Carolyn says. Yes, thank you. And this looks like an Alapsamia rostrata. It's a different species of coral than we saw earlier today. The one we saw earlier was Madrepora oculata. And if we can zoom in here, we might see why we call it why it's called rostrata for a rostrum, because at least when the skeleton is cleaned, there is a prominent ridge that runs down one side of the cup into which the polyps withdraw. And it typically forms these fan-like colonies. And when you look at those polyps, you can see that unlike the octocorals, they have more than 8 tentacles, and the tentacles do not have those side branches. So this is a stony coral, and it is one of only a handful of deep-sea branching stony corals The vast majority of deep sea stony corals are solitary cup corals.
Full screen.
That's great. Thank you very much. And you can see there's also some dead branches there between these two yellow portions of the colony. And it looks like in the distance there are large numbers of tube worms of some sort on those rocks.
Clear.
So I've just been asking some of the scientists ashore what their thoughts are on this dive so far, and they are all absolutely over the moon. This dive has far surpassed any of our expectations. It's had a bit of chemosynthetic habitats, these amazing asphalt extrusions, some of which were still partially liquid, and many of them which then hosted these incredible deep-sea coral communities, as well as they were seen on orthogenic carbonates. But this is far surpassing any of our expectations for this dive, and a lot of them have seen things which they didn't expect to see, which is a really great result.
Boy, there's a lot of that purple clavularia on the rocks here. Look at that. It's just covering the place. Small orange anemones.
It's not holding, huh?
Yeah, it's really bouncing around off the asphalt.
Potential tubes to right of lasers. Also potential fish in line bottom right.
Yep, great to have you piloting. Yeah, if we could just continue perusing around this area for a bit, and then we want to be heading over, as Nav knows, past waypoint Seep 4 and onto our waypoint 3. Thank you.
Yeah, well, actually,
that white patch in the top right.
Waypoint 3 is a little off
for
from
tube
the
worms.
direction of waypoint 4 to waypoint, or sorry, Seep 4 to Seep 5. Do we want to hit waypoint 3?
No, we don't have to.
Okay.
Slightly above and to the right
Just
of
go right
lasers.
to Seep 5.
Yeah, I think
In
let's
the
just like wherever we see the most interesting stuff, as long as it's in the general direction
Gotcha.
of waypoint 3.
Understood.
Just see some dark spots, which could be small overhangs or more tar seepage. Yeah.
So I'm just reading a background essay from the Gulf of Mexico expedition in 2014, one of the ones that the Okeanos Explorer did back then. And thank you, Carolyn, for pointing this out to us. So asphalt seeps were first discovered in the Gulf of Mexico. They were first discovered in 2004 at that site on the Campeche Knolls, 3,000 meters depth. And then after that, there were reports of asphalt volcanoes off the coast of California as well as off West Africa. Yeah, so we could do a quick zoom on the orange anemones. We haven't really taken a good look at those yet. Yeah, we've more been bamboozled by the purple corals. And so, interestingly, the asphalt extrusions we're seeing here that are now hosting all of these incredible communities, these anemones we're looking at right now, the purple stoloniferans, this cup coral, those are related to that tar lily that was found during the 2014 Gulf of Mexico expedition by the Okeanos Explorer. So that was a pretty famous sighting, at least as far as the ship goes. A lot of people reminisce about it quite fondly and funnily, in that it was supposed to, they thought it was a shipwreck, and you know, they were prepared. They had all these archaeologists lined up on shore to explore this potential shipwreck, this anomaly they were seeing in the bathymetric data. And then once they dived on it, they then found this huge, enormous asphalt extrusion that had split into, I think, 5 different sections, or no more than 5, and had formed what looked like this asphalt flower on the seafloor, or as they said, a tar lily. And so because of telepresence, they were able to switch up and, you know, get all these archaeologists off the call, since what they were there interested in wasn't there, and instead get a bunch of geologists in to weigh in on what they were seeing. And so this is just another form of that asphalt being extruded onto the seafloor. Thank you, Carolyn, for pointing that out.
Sure. I'm not fine. I'll do 40. 30. Yeah.
Now, Bridge, go ahead.
Hey, another ship move, please. Range 30 meters, bearing 230 degrees, speed 0.2.
Bridge copies. Next move, 30 meters, bearing 230, speed 0.2 knots.
Copy.
Stand by for input.
So earlier this summer, I attended the Chemosynthesis-based ecosystem conference in Woods Hole. It was in August, and at that that conference, some of the researchers showed this really well done video on the Campeche Knolls asphalt volcanoes and seeps at those sites. And I just remember sitting in the audience and thinking, this is one of the coolest things I have ever seen. All of those asphalt, you know, droplets stretching out into the water column, and all these different weird shapes of these asphalt globules. And now today we got to see it ourselves. And so this has been an incredible dive, being able to see an asphalt seep, even though it was just a tiny one, live myself from on board the Okeanos Explorer. It was completely unexpected and very welcome.
Starting to drift off a little. I can follow you around if you want. This is interesting.
So all of these little shards we're seeing on the floor, I think, are more stony coral, whether they're Anelopsamia or Madrepora, I'm not quite sure, but those would be remnants from probably some large colonies that were found in the area once upon a time.
Yeah.
We've now also seeing these urchins, no, what is it? Gracilis affinis, that are deposit feeding around. This is probably quite a productive area. These sediments are probably quite rich in organic matter. And so they're probably thriving around here. You can see in the distance how many of those white spheres there are around. As well as we're just hovering over this little patch of black bacterial mat, white bacterial mat, which is all in this layering on top of this reduced sediment. This really is just a very extraordinary area of the Gulf of Mexico.
Oh, just sargassum.
So this is some more of that sargassum we've been seeing. I think we've seen some on nearly every dive. A lot of it is floating by the ship. And it does have to end up somewhere. Sometimes that'll be beaches, and sometimes that'll be down in the deep sea, where it is then food for many different animals down here.
Shifted my head a little to the left because I was a few degrees starboard of that. No, I was off a little bit. We're doing 230. It was at like 237, so just lining up a little better with the move.
I think as deep sea biologists, we're pretty spoiled in terms of amazing sights. We do get to see some incredible things pretty often, but so as a result, it takes a lot to actually kind of shock us and have us keep going on about something, and that's kind of how I feel about that, about that asphalt seep. That was one of the coolest things I have seen in a very, very long time.
You want another one? Oh, no, I'm good. I was debating updating the bailout plan, but I think we're good. Southeast. Yeah, we are. Got another couple meters.
Here we've got some more of this asphalt extrusion. It looks, and as we were saying before, it looks almost like cooled lava in a lot of ways. So it looks like coming into view, we've got a couple more of these very large coral colonies living on these asphalt extrusions. These look like they could be very large stony corals.
Oh, yes. Oh, yes. Look at that.
And so stony corals are extremely slow growing, and so these large colonies could be quite old.
Can we go in with lasers on?
This is beautiful. This is like a deep sea forest.
Mm.
Oh, so it's not actually asphalt. What the darkened areas we're seeing here are dead colonies.
Yes, yes.
Looks like there's something large and purple.
And they're growing in a very
above the corals.
Strange
Right above
morphology or growing form. It's
behind that
It's
coral.
like they extend out horizontally and then curve upwards.
Should zoom to that coral on the aft.
Nav, Bridge.
Good.
Yeah, it's a complete move.
Roger. Thank you, Bridge.
You're welcome.
Complete move, pilots.
Oh, that's lovely.
So that looks like Madrepora, and we can tell
right.
that now that no,
But
I think it is.
Could this be, I'm not familiar enough, and I've just been bumped out of the chat room. Could this be Madrepora carolina? I don't know how deep it gets, but I know it formed monoplanar fans like this in the Bahamas. But that was in 350 meters of water, which is a lot less than this. So not sure. I don't know what the diversity of growth form is in Madrepora oculata.
While we're waiting for an answer, these stony corals are, they're like China. If we were to try and collect some of this, it would literally just shatter. It would be like if you just dropped a piece of china on the floor. It would just break apart extremely easily.
Full screen.
They are notoriously difficult to collect as a result. So Eric is now saying, and Andrea as well, are now saying that they think this is Madrepora oculata, because, as for the reason you were saying, Chuck, Madrepora carolina is pretty shallow. But Daniel Wagner is just blown away. He can't stop saying how beautiful this whole site is.
It is.
Eric has also added that he hasn't seen Madrepora carolina outside of the Florida platform in the Gulf of Mexico. But I remember we collected quite a large piece of Madrepora during the last expedition I was on, which was to St. Paul and St. Peter's archipelago, or rocks, in the middle of the Atlantic, just outside of Brazil. And once we brought that piece of Madrepora on board, we put it into a tank to see if it would photograph well. And there was just so much mucus being extruded from this coral. Like, it was unbelievable how mucusy these colonies can be. Andrea is also saying that there's also Madrepora profunda, a third species, but she thinks that may occur deeper in the Gulf of Mexico. There has been so much to look at, so much to take in, so much to discuss during this dive. So right in view right now, we've got these light pink-white colonies of Madrepora oculata. Then off to the left, we've got what looks like a chrysogorgia. It's a bit hard to pick up against the sediment, as well as those purple stoloniferans, Clavillaria. And then we've got those bright yellow, another type of stony coral. So those are Enelopsamia, that colony.
I'll let you push out, but you guys let me know if you want another move.
Just amazing.
Actually, was saying let's head southwest, but if we see things that we want to chase, feel free.
Potential plastic
Features.
bag.
Is that plastic caught before?
Yeah, it looks like it.
Okay, another sordid reminder of our impacts on the deep ocean. We are at a depth of 1,145 meters, quite far away from land in the Gulf of Mexico, right down in the southern area of the Gulf of Mexico, and yet
Fish
still
hidden
here is a
inside,
plastic bag.
White tip.
Some really large colonies of Madrepora here, and also this Enelopsamia is a good, you know,
it's
80 centimeters across, 70 centimeters across, nearly a meter.
And you see all of the dead coral branches that we've mentioned. I wonder, let me get my words straight out of my mouth. I wonder if how many successions of live colonies there have been on this site that we can see here. Were most of those dead corals all alive at once, or were
Still maybe give it a minute.
did they represent a succession of the same sort of isolated colonies that we see now? And how long has this been going on? Are those dead colonies 50 years old, 500 years old, or 5,000 years old? And there's that. Did you mention the Anthomastus? There's,
no, I think that's an anemone.
Yes,
Yeah.
you're right. That's a flytrap anemone.
You guys okay with another 30? Ready for it?
What's that glowing in the distance?
It looks like a shiny fish. But I may be wrong.
I'm going to guess shell.
Nav bridge, go ahead.
Ship move, please. Range 30 meters, bearing at 230 degrees, speed 0.
Oh no, it's the inside of a bivalve shell.
Oh, yep. 30 meters, bearing 230, speed decimal 2.
Yep, Bathymariola shell. So those are the very large chemosynthetic mussels we can get. You can see the 10 centimeter scale in front of them, and those that particular individual was probably about 20 centimeters long. And when you compare that to the mussels which, you know, we eat, those that are commercially available, those tend to be less than 10 centimeters in length, whereas these deep sea mussels, these Bathymariola mussels, can get up to, I think the largest on record is about 37 centimeters in length. So they're pretty big. And this may gross a lot of people out, but a lot of them also have commensal worms that live inside of them. So each one will have one large worm living inside of it, and they spend their entire lives together, potentially. And then also some smaller worms that can live in between there as well.
Left.
So those little platforms look oddly similar to the one we saw yesterday that had live mussels in the middle of them. Oh no, yeah, you're right, Don.
ROV nav, bridge.
Go ahead.
Yeah, just be advised, I've held position for a second. We just had to ramp up on some thrusters.
Yeah, lots of coral rubble of this stony coral, and then in between that, it looks like the stoloniferans have actually colonized those dead coral skeletons, the purple.
With the 25 meters that we did not get out of it.
Understood. Yeah, if you guys are comfortable with that, let's go ahead and try that additional or remaining 25.
Yeah, I didn't see anything that led me to believe we couldn't do that. I didn't get anything in the red or anything like that. Just that's the first ramp up I've had on this watch, so I just wanted to stop it before it got too high.
Understood. Thanks for letting us know.
Yeah, no problem. I'm going to go ahead and plug that last 25 in and see what we can get out of it.
Copy that.
All right, I'll get back with you.
Sounds good.
You guys copy, Bridge? Yeah, you just halted the move. They're going to keep it going here now.
So while these coral colonies are some of the largest we have seen during this expedition, just north of here, in an area where there has been quite a bit of exploration so far, they have found coral colonies of Madrepora oculata and a couple other species that in some cases were between 6 and 10 feet long, which is
we're looking good right this second.
really something.
Understood. Yeah, sounds good. We'll be moving pretty much southwest to our port beam there for the remainder of the dive. So if you want to try a change in heading or something, that'd be fine with us.
Yeah, right now we are into the move and it's settling out. So I think we're good.
Okay.
I'll let you know if I see anything different. I think we're good though.
Copy that.
All right.
Thanks.
Yeah.
So it looks like we're leaving this area where there were live coral colonies as well as some dead coral colonies. We can see we're leaving these overgrown coral colonies as we move out into the sedimented area. Here we're seeing another one of these Gracilichinus affinis urchins, as well as some more of these dead shells. To the right of the Gracilichinus, we've got a dead vesicomyid shell, so that's one of these chemosynthetic clams, and you can see how the shape of it and the color of it differs from that Bathymodulus shell just to behind the Gracilichinus. So this is a small, well, not small, I mean, it's still at least nearly 10 centimeters in size. It's a small white, very circular almost shell. But then if we were to zoom back out and look behind the urchin, there was a Bathymodiolus bivalve shell that was open, yeah, glinting off in the distance. And you can see that one is much bigger and much more elongated. And is almost like, yeah, like kind of like a boomerang shape. There are, in fact, some species, a species known as Bathymodiolus boomerang, because it is one of the largest species. I think that's the one that holds the record, about 37 centimeters in size, which is, I mean, over
Enormous.
a foot. Yeah, absolutely enormous. But it has like a crook in the shell, so it does resemble a boomerang quite a lot. I'm not saying that's what this species is, but you can see how those two shapes do differ a lot between the clams and the mussels. But they are both chemosynthetic. The mussels tend to use the methane in the fluids, whereas the clams do tend to use the sulfide-rich fluids, component of the fluids.
So, Diva, this Bathymodiolus, is that in the mussel family, Mytillidae, or is it in its own family?
No, I'm pretty sure it's in the Mytilid. No. You know what, I'm just going to stop guessing. Hold on.
It looks like a mussel. And the
so,
other...
of course, it is, yeah. It is a mussel, and I want to feel it is within Mytillidae, but then has its own subfamily.
Oh, okay.
But
Copy.
yeah, so Mytillidae family, and then subfamily, I think, is Bathymodiolinidae, Linnae, yeah. And then Bathymodiolus.
And what is the other clam that we saw, the other bivalve we saw? What is that related to? Do you know?
I'm not sure whether, I think that one may be a vesicomyidae,
okay.
but it could also be a thiosaurid. But I'm not so good with the clams. But it's definitely one of those two families.
All right, let me see what that's related to.
ROV nav, bridge, move, complete. We didn't have any issues with that.
By the way,
at all.
thank you, Don, for accommodating both of those zooms on the different shells.
And are all of the vesicomyas chemosynthetic? Do you know?
I think so, yeah. I think they're pretty exclusive to organically enriched habitats, which are mostly chemosynthetic based.
Which is interesting because the genus was described in 1886, before anybody knew about this sort of thing. There's another Paraphila, coronate medusa. Let me just take a moment to say that we should not use the term jellyfish if we can avoid it, because it's a very vague term that applies to a lot of different gelatinous planktonic organisms. So things that we traditionally think of as true jellies with the umbrella or bowl or dish-shaped umbrella should be called medusae, after the Gorgon Medusa from Greek mythology, who had the snakes for hair and turned people to stone if you looked at her. There are other things that are gelatinous that are, I mean, you could use the term jelly for things like siphonophores, which are colonies, whereas this jelly is a single individual. A siphonophore, like the Portuguese man of war, is a colony of a combination of individual polyps and sometimes modified medusae that are all attached and function together as a unit. But calling that a jellyfish is not the same thing as calling this a jellyfish. And then we have the gelatinous relatives of the sea squirts called the salps and the pyrosomes, and they are actually more closely related to us than they are to these jellyfish or medusae. So they're just as gelatinous, but so we should be really careful when we use the term jellyfish, because it can apply to a lot of things.
But I do think this species of jellyfish, this Paraphila, pretty easy to remember. I remember I used to like that one in exams because it was, pretty easy. but it is one of the most well-known of the deep water, I don't want to say jellies, but jellies, in that it's thought to have a worldwide distribution. They have been found up to 7 kilometers in depth. And yeah, they're quite common.
And one of the reasons they can be well understood is because in far northern waters or southern waters, they can get into much shallower water in places like the Norwegian fjords, where it's easier for folks to study them. Coming back to some more hard substrate again. And according to the World Register of Marine Species, which is our general arbiter for the identification and taxonomy, the classification of marine organisms, that clam that we passed by, the dead clam, the Vesicomyia, is actually in a family related to the Venus clams, which include the commercial hard-shelled clams, the blue point and quahog clams, and many others. And that looks like a little rat tail again. And I keep getting bumped out of the chat room here, logging back in. So we've probably seen this before. This is probably Corophenoides. Not sure. It doesn't have the black rim, so it may not be the same species as we saw before. And those little dark brownish tan little tree-like structures sticking out of the sediment are probably the agglutinating foraminiferans. I'm not sure if I mentioned what those are on this today's dive before, but foraminiferans are single-celled organisms, although they have many nuclei, and they radiate extensions of their cells for feeding, like an amoeba radiates pseudopods, but these radiate branching extensions called reticulopods because they're often mesh-like. And the best known, oh, that's an interesting one. Maybe perhaps a juvenile of the other. If any ichthyologists ashore can help us out. But the forams are best known from their planktonic species, which produce a sand grain-sized shell or test of calcium carbonate, and they float around in the plankton. And when they die, the shell sinks to the sea floor, and they occur in vast enough numbers that they form vast deposits of sediment on the sea floor. As a matter of fact, much of the sediment that we see here may be the shells or tests of planktonic forams. But there are only 40 species of planktonic forams, where there are over 8,900 species of bottom dwellers. Some of them, of course, produce, oh, what is that? There's a squid up there, yes? A different one than we've seen.
Ooh,
yes,
beautiful.
Perhaps. Not sure if that's a short fin that looked like an omistrephed. Perhaps.
No, you went
I don't think it was a Heteroteuthis.
I was thinking that
Histiotuthis,
apart
rather.
from, sorry to interrupt,
Sure.
apart from the short-fin squid we've been seeing, there haven't really been that many other cephalopods of any kind. We saw that first octopus, which was a moose octopus.
Moose octopus, yes.
During the first dive, but since then, apart from those potential ilex squid, which have been around quite a lot, there hasn't been much else. I think that's a different species to what we saw before.
Probably.
But that's been kind of it on the cephalopod front. But sorry, carry on.
No, that's all right. Andrea Quattrini has suggested that rat tail we just saw a little while ago was perhaps a hymenocephalus. It did look different from the Corophenoides. It didn't have that black rim to the eye. But so just to wrap up the foram story, many of the bottom-dwelling forams do produce a calcium carbonate test, but many others just glue to themselves sediment grains and extend their little protoplasmic extensions out to capture particles of food. And there are hundreds and hundreds of species of those. And we see them all over the seafloor here. We see them on hard substrates. And the folks who work on forams are sort of a different group. We have, the fish people, the invertebrates people, and the people who work on the more single-celled organisms. So I'm not sure how well those are understood here in the deep sea. A couple of other options for those rat tails are the genus Kumba, K-U-M-B-A. I wonder if there's a species Ya, Kumbaya. Pardon me. And so that was from Andrea Quattrini about Kumba or possibly Nezumia, two different genera of rat tails. There are a lot of rat tail species.
Here we've got a couple more of these Bathymoriola shells, some of which, as we were discussing earlier, could be quite old. The ones which have lost that shine are obviously a bit older, and I think, I can't remember who it was, I think it was Eric, said earlier that some of these could be as old as, 12,000 years in other areas of the Gulf of Mexico where they found these aggregations of shells. They have dated them and they have proven to be quite old. So these could be very old communities that existed a quite long time ago.
And Kevin Rademacher suggested that of those two rat tails we saw just recently, the first might be a Nezumia and the second Trachanurus sulcatus. We're throwing out all of these names. Of course, no one apart from the ichthyologist is going to remember any of them, but that's okay. We want to give you a feel for the diversity of things here, that there is just even though we go on and we, there are maybe acres of empty mud that we see and scattered dead shells that may be centuries or millennia old, there's still an incredible diversity of life down here. And, of course, we have not got an inkling of what is living in the sediment, nor do we have an idea, not at least from our observations here, of the smaller organisms, what are called the macrofauna, from only 2 millimeters down to, say, 0.5 millimeter, or the myofauna from 0.5 millimeter down to what, I think 50 micrometers or something like that. There are different size categories that people use, because the equipment you use to collect and study these organisms is different for the different size categories. If you use if you take a sediment sample and you pass it through a series of sieves,
starboard side,
you will get different organisms of different sizes collecting on the different sieves, the different screens.
You want to go to this?
And they have different roles to play in terms of recycling nutrients, feeding on the organic material, providing food for larger organisms.
245.
Some of the smaller organisms in the sediment include things like nematode roundworms. There are small crustaceans, tiny crustaceans, usually less than a millimeter long, called harpacticoid copepods.
Go ahead.
They're relatives of the copepods that are abundant in the plankton, but they're sort of unsung because they're buried in the mud generally, and they're very, very difficult to identify and work with. But they are sometimes in enormous numbers. Then, of course, there are forams. There are tiny little crustaceans that have a bivalved clam-like carapace called ostracods.
I know. I think it's the backside.
And a number of other different groups of worms.
Tilt down, we can start on that.
And that's a whole different universe that requires sampling with different methodologies, taking core samples or grab samples of the sediment and carefully sieving through it. And there's really no way we can access that from this expedition.
Coming in.
Here we have another probable paramuricid, maybe, unless this is, and Kevin reminds me of another group called the tardigrades, the water bears, and they are many-celled organisms. They're animals, but many of them are no more than 50 micrometers long. 50 micrometers is 1/20th of a millimeter, and a millimeter is 1/25th of an inch. So we are talking small, microscopic creatures.
Full screen.
And of course, all of the organisms you see here, virtually all of them, the brittle stars, the soft corals, the sea fans, the black corals, the clams, they begin life. Of course, they're produced as a result of fertilization, and the fertilized egg is usually launched into the plankton, where it develops as a microscopic larva, which has its own role to play in the food web of the ocean, providing food for larger organisms as well as feeding themselves. And Andrea says these yellow fans are all paramuricea,
more or less on our tracks.
And in the clade or group B, a clade means in classifying things, a clade refers to a portion of a evolutionary or phylogenetic tree that includes a common ancestor and all of its descendants. And I talked a little bit about that, and those are the things we try to identify for classification. So Octocoralia, the octocorals.
Thing on the right edge of the screen looks weird. Maybe a large crab.
And there is a deep-sea red crab.
Get a quick snap on it.
So just for example, Octocoralia is a clade, or what we also call a monophyletic group. It includes its common ancestor and all of its descendants. The mammals are a clade, for example. But the reptiles are only a clade, a common ancestor and all of the descendants, if you include the birds. Because the birds are a branch of the tree that arose from the dinosaurs, which of course were reptiles. So we try to use our classification scheme both as an information retrieval system and as a way of looking at the history as well as the diversity of life. And he is waiting, he or she are just waiting.
ROV nav bridge, move is complete.
Copy bridge.
To take on D2.
Lots of dead bivalves here. And these are both the, that long one next to the crab, that one looked sort of like an oyster, but I don't know my deep sea bio. So that's
yeah, we can definitely see the Bathymoreolus, which are the longer ones, and then that would be one of the...
Right, the clam-shaped ones are the vesicomyids?
Yeah, but the fact that they were still closed kind of threw me, because I was like, oh, some of these are alive.
I sort of like the higher flying.
But yeah, some vesicomyids as well. But then yesterday, remember, we saw that Solimbiatrax, yeah, which is another type of chemosynthetic bivalve, and so those may be here as well, the third family of chemosynthetic bivalve, but we haven't seen those yet today. But those shells are also very distinct.
Do the vesicomyids sit on the sediment or burrow
or
partly? Or?
So the Bathymoreolus mussels sit with some of their body within the sediment, but most extruding. They're very obvious to see. We saw them yesterday, how they were arranged in the sediment. But the vesicomyids and most of the chemosynthetic clams tend to burrow nearly completely or mostly at least. So when I have seen them before, they've maybe had a fifth or a fourth of their shell protruding from the top of the sediment, as well as their siphon. But in some cases, Eric was mentioning earlier, when the flow is lower, they will actually completely bury themselves, and you may just see a little bit of the siphon, if anything. So they could be here alive, we're just not seeing them.
Understood. Yeah. And do the Bathymoreolus, are they like other mussels that they attach with a byssus, a clump of bristle threads? Okay.
Nah.
Sorry. So exactly as you just suggested, Chuck, just like shallow water mussels, they do use byssus threads to attach to hard substrate of some kind, or to each other sometimes.
I wonder what that
and
is.
In case you aren't familiar, byssus is B-Y-S-S-U-S. And these are adhesive threads that mussels secrete from a gland.
Rock structure ahead.
Material comes out as a fluid and runs down their very narrow finger-like foot in a groove, reaches the substrate and becomes a hard thread.
Don't see anything living
and a
yet.
mussel can actually move slowly by releasing some of its threads and adding more as it moves slowly along the sea floor. Of course, mussels are most familiar to us as very shallow water, sometimes intertidal species, sometimes highly edible species. And look at this structure here.
I think some of these look like they may be alive. Maybe it's wishful thinking, but from the way some of them seem to be standing up, that could be some live bivalves. Plus, there's quite a bit of bacterial mats here, indicating, there could be this area. But as I was saying before, you tend to get these zones around chemosynthetic habitats.
Look at that. It's a ring of dense. Wow.
So this kind of concentric, right next to where the seepage is, there'll be live mussels. And then, outside of that, there'll be this dead ring of mussels sometimes, mussel shells, which tend to have their own fauna living amongst those shells, which is different from where you get these live mussels, which is what we are looking at right here. And then, of course, you've got these orthogenic carbonates, which are also found in this area, and those can harbor big deep sea corals, as we've been seeing already during this dive.
And so these carbonates, this is a much paler rock than the asphalt. Very different texture to it.
Yeah, it doesn't really look like the lava we were seeing earlier, lava in inverted commas. But no, there are definitely some live mussels here. These ones do look quite small, being a relative term when you're talking about these mussels. But I'm not sure which species it would be. It'd be one of three, either Bathymoreolus childressi, Bathymoreolus hecri, or Bathymoreolus...
Stand by for input.
I was doing so well. Something yeah.
Pilot, can you zoom in first at the base here? Certainly.
Sorry, so it was Bathymoliolis brooksii, Bathymoliolis heckeri, Bathymoliolis childressi, and there are loads of bubbles coming out of the top of this feature. Oh no, that's okay. Yeah, and we're going to be here for a little while, so just be prepared for that. So we are actually really close to a site which we had put down as Seep 4 during our surveys last night. We did detect potentially this bubble plume in our bathymetric data in the water column. And it seems like such a small stream, and yet we were able to pick it up very easily on our multi-beam bathymetry. So yeah, as we can see, these mussels are very live. You can see the shells are partially open, and they've got some of their siphons extended. Well, you can also see there are some polyplecophorans, which are chitons.
Drifting back. Right, that's the chitons are, again, a different major group of mollusks. So you've got these bivalves, clams, oysters, mussels, etc. You've got the cephalopods, octopus and squid, the gastropods, the snails, cowries, whelks, and conchs, and then the polyplecophorans, which means many shell bearer, because their shell is divided into, I think it is 7 overlapping units or valves held together.
The bubbles right between the skids.
So those chitons might be Leptochiton micropustulosus, which is a species which is found from the southern Caribbean all the way up to the Gulf of Mexico, and they are very common in these seep habitats. We can also see some echinoids. Those are likely, or urchins, those are likely not chemosynthetic, but they have moved into this very productive area to either munch on some bacterial mats or perhaps some of the other small animals living here. But you can also see there are some of the larger predators, as I was mentioning earlier, some of the Chasian quincunans, the red crabs that have come into, I know they do eat mussels, well, at least Chasian feneri, the golden crab does. It does eat these Bathymoliolis mussels. Oh, and in between, we've also got some Alvinicaris shrimp. So those may be Alvinicaris muricola, or there is another species that's found in the Gulf of Mexico as well.
And these are restricted to cold seep environments, aren't they?
Pardon?
Are these shrimp restricted to cold seep environments?
Well, not just cold seeps, but also vents. But yes, chemosynthetic habitats, most certainly. And actually, if this is Alvinicaris muricola, it has been seen at from as far down south as Brazil on a whale fall that was discovered down there. Then it's been seen off Trinidad and Tobago at some cold seeps found there. Off Grenada, cold seeps also there, Barbados. And then if you jump up to the Cayman Trench, it's been found at hydrothermal vents there, but not in the numbers as the other dominant shrimp has been found. And then again, we move up to the Gulf of Mexico, like Chuck and I were chatting about yesterday. That prevailing current from the southern Caribbean moves up to the Gulf of Mexico, bringing all of these cold seep chemosynthetic animal larvae, as well as other deep sea larvae, with it to the north. So then we've also got some snails in between. That one looks like Canoia meroglypta. It's got a very ridged shell, and again is a very common seep inhabitant that occurs from the southern Caribbean all the way up to the Gulf of Mexico.
That snail was just off left video, next to the chitons right there. Top center, 2 chitons and a snail.
Partial.
This is magnificent. This is amazing.
Yeah. But do you see what I mean about how the live mussels...
Yes, sort of half in and half out, some of them.
And so...
So you've got some of them sort of half buried in the sediment, and up the sides of this mound, they're clinging to the hard substrate.
Yeah, and so these mussels, again, it depends on what species of mussel this is, but some tend to use the methane more, some tend to use the sulfides and the chemicals more, and some tend to use a combination. And I think a lot of these Bathymoliolis species can actually have a mixotrophic diet. So not only will they use the chemosynthesis-based nutrition, but they'll also filter feed as normal mussels would, so they can kind of switch between or supplement their diet if needed with one or the other.
Ship it serious.
And
nice
Diva,
and close.
can you, I'm guessing here with these concentric bands of dead shell, so the the populations of mussels would track the emanations from the, which is, I guess, not continuous then, that these things release material or develops
looking to get the light pull
cyclically or sporadically over time?
So yeah, it's completely patchy. What this may have been is a much larger community, and now the flow is waning slightly, and so you can see how the community may have decreased in size, and so now the most amount of flow is coming out in the center of this area, or perhaps it could have migrated, but as we were saying yesterday, these cold seep habitats are quite ephemeral. They do switch on and switch off, not super rapidly, but they do in the grand scheme of things. Oh, and there's a little fish right at the bottom of the screen, just below the lasers. So I'm so pleased that we've managed to find one of our seep targets that we had found mapping last night, and this has been a great example of using all of our tools that we have on the ship to try and locate these habitats.
Will there be any elevation of temperature associated with these? I mean, they're called cold seeps, but I had no idea.
So if there were, it would only be by a couple of degrees. Cold seeps are cold seeps because they do have this hydrocarbon-rich fluid, this chemical-laden fluid, but it doesn't tend to be higher than ambient temperature.
Okay.
If anything, it would just be a couple of degrees. They're not like the hydrothermal vents where you have this superheated fluid in a lot of cases, sometimes over 400 degrees Celsius, gushing out of the seafloor. This is a much more gentle process.
Look at that. And is that a coral?
This is Carolyn, and can you hear me okay?
Yeah, yes. Carolyn, we can hear you great.
Dita? Okay. Yeah, I'm sorry, I'm in my car, so I'm on speaker. So a few things. First of all, the fact that you found bubbles here, as I said yesterday, almost any emission of bubbles, even the smallest amount seemingly and the smallest bubbles, is usually enough to cause the contrast that we detect with the hydroacoustic methods, like the multi-beam sonars and the split-beam sonars. So it's great. You probably have found it. It's like finding a needle in a haystack, and it's great that you found it. Something for listeners to know is that we're at a water depth and a temperature right now at the seafloor that is well inside the methane hydrate stability zone. So in theory, methane bubbles should not exist. They should be a frozen form of methane hydrate. However, we do find seeps in these areas, often because there might be a heat source at depth. And in the Gulf of Mexico, the northern Gulf of Mexico, the primary, it's not a source of heat, but because there's so much salt, it's very hot down there because of the thermal properties of the salt. And the salt can be very near the surface. So that's one reason why you can have these deep seeps in the Gulf of Mexico and not just have them clog up all the time with gas hydrates. And I'll stop there because I'm not sure how well you can hear.
That was excellent, Carolyn. Thanks very much. A great explanation of that. Because, yes, exactly, seeing the bubbles under such hydrostatic pressure at 1,100 plus meters is pretty impressive.
Diagonal down left.
And that looks like a little colony of perhaps Madrepora at the very peak of this accumulation here.
Pretty high now.
And a number of these
in HD2.
deep sea red crabs.
Okay, yeah.
So looking at this community here, this is a great example of these, especially if you look in the Sirius view right now, we can see D2 hovering above this little oasis. And so we can see that this area, which is obviously where these hydrocarbons and this chemical-laden fluid is coming to the surface, or at least closer to the surface of this sedimented area.
Right above
and it's forming this little oasis where we're seeing a huge aggregation of life. You know, there are probably a couple thousand mussels just here in this little patch, all squished together. And then additionally, there's shrimp, there's chitons, there's crabs, there's urchins, and they're all just smushed into this very small area where because of the high productivity and this abundance of food, just in this tiny area that's resulted in this explosion of life, this very abundant, not super diverse, but abundant and prolific area of life. Yeah.
And look at how circular it is. That's just very cool.
This is ridiculously, they're never this perfect.
That'll give you a better angle for the bubbles. Yeah.
Call it bullseye seep.
Yeah, unfortunately, there's already a very famous cold seep on a Vancouver margin called the Bullseye Bench. So we'll have to think of another name.
How about target seep? Maybe we'll get some funding to come back from the drug chain.
Target?
The drugs. Target.
Aren't they like a grocery?
Yeah.
Or whatever.
Pharmacy, grocery, yes, a little supermarket.
They're coming out up there. It's just sporadic. See them.
Very, very cool.
Yeah, so this has just been a dive that's far surpassed our expectations. We have gone from deep sea coral forests to asphalt seeps, and now to very traditional, methane seeps.
And one group of organisms that is, at least to me, of course, notable by their absence is there is not a single crinoid. And
you would notice that.
I would notice that, but that was also the case on the expedition I went on further west in the Gulf of Mexico some years ago, where we normally might find sea lilies growing out of the sediment. We did not find them, and we only found a couple of feather stars, and this is very much unlike the fauna that we saw along the West Florida escarpment, which was completely different by and large. Very few overlapping species.
Really fantastic. Can we zoom in on the top of this feature? It looks like there's a couple, those cup corals or anemones?
And a little branching coral at the very tip.
As well as we can see these Munidopsis squat lobsters that we haven't seen anywhere else during this dive, this particular genus. And that's because, again, Munidopsis, a lot of the time, can be associated with chemosynthetic habitats. Oh, as well as this fish that's floating down. We could do the fish first, because it might move away.
Unless it's a squid.
You might have called it, Roland.
If you could catch some of those Munidopsis, I may be able to identify them. We have some specimens here at Texas A&M that have already been identified.
Yeah, I remember Willis and Linda Pequenot did an awful lot of work on deep water Galatheids back in the early 1970s.
Is it possible to zoom on the squid?
That's weird.
I feel like we've been saying that a lot.
Yeah, but look at that. Its fins are undulating. We have not. This is very different. Where's Mike Vecchione when you need him?
And you can see, what are the two feeding tentacles?
Well, yeah, there are two longer feeding tentacles there. This is a new one on me. I know a few midwater squids. Oh, there was the bottom. I didn't expect that.
Ooh, look at that color change. That was awesome. And
away he goes.
Amazing. What?
Scott, with respect to my comment about the lack of crinoids, Scott France mentions that when he was a grad student, he went on a cruise to the West Pacific hydrothermal vents with the intention of collecting amphipods for his dissertation project because they were so abundant in the East Pacific vents. And, of course, they didn't find a single one.
And after that, he decided to switch to deep sea corals.
At some point, at least, that's true.
bubbles.
Yeah, that you could see on that squid. You could see the two slightly longer tentacles sticking out from among the long arms. There's another little point of nomenclature. Octopuses do not have tentacles. Octopuses have 8 arms. Squids have 8 arms and 2 longer tentacles. Just so you know.
Thanks, Scott.
Kevin said that squid was imitating a pogo stick.
So yeah, these are little solitary cup corals found around the top of this feature, as well as that is not a coral we have seen today.
That is Solanasmilia.
Oh, we did see some of the beginning of the dive.
Because you can see the calyces, if I remember correctly. Correct me from shore if necessary, but the calyces are elongated. They're sort of oval or even sort of kidney-shaped. And I'm not good enough with my solitaries, but that conceivably could be Javania.
Partial.
They look large enough if they're not Desmophyllum.
Transmitting the manips on quad screen. Going wide.
So Eric is commenting from shore, Eric Cordesny, saying that he thinks that is Solanasmilia, and the fact that it was under a rock before and now right next to a seep is quite weird. He has not seen this coral being located in those types of places before. Oh, can we actually see the spot where those bubbles are coming out of? And those bubbles, it's amazing. They just seem so small, and yet that's what we're able to pick up from the ship. You know, 1,100 meters above.
We looked at.
That's that sonic contrast. And either Eric or Scott, I don't know if you got a chance to see that quick zoom we had on the solitary corals, but is either Desmophyllum or Javania possible identifications, or is there something else that has those big septa that come out and run down the side outside the
about that earlier.
The calyx?
Interestingly, there are usually more than one gastropod species found at these sites, but so far I can only see one, that Canoia miroglypta. Yeah.
Yeah, that's the same one as before.
But sometimes we can see like trophon type ones or other species. And yesterday at the seep I was just about to say, yesterday at the seep we were exploring, we saw a couple brittle stars. And but here we weren't, but nope, we just spotted one. So in a lot of the Gulf of Mexico seeps, surprisingly, some of the gastropods that I was just discussing have actually been replaced by 2 species of brittle star.
And the brown you see on some of the shells, I believe, is the outer organic layer that the mussel lays down, called the periostracum, which is apparently worn off or has not been produced by the others or is covered with sediment.
And this Chasian quincunans crab, you can see it looks, kind of black, has a lot of barnacles on it, just generally like it's been through a lot. But that means it's probably quite close to molting. It's probably been wearing this carapace for quite some time, whereas we saw one early in the dive that looked shiny and new, and so fresh and so clean, and so that this one is probably getting close to that molt period.
Pilot, could you please drop?
Ooh!
Oh, you see something else?
Can we zoom on that fish?
Oh yeah, that little fish there, just below and to the left of the lasers. 2 little black eyes. Like a little white tadpole, right?
Yeah, so it
right smack middle.
So that looks like an eel pout fish or a zoarcid. And those are a family of fish that are commonly found at chemosynthetic habitats. I am not sure which species this is.
That was a good spot.
No, in a lot of seeps in the southern Caribbean where I've been working recently, we do see much more of those eel pouts. That one is Pachycara caribbeum. But here they don't seem to be as abundant.
Is that as big as they get?
No, they can't. Well, I don't know what species this is, but
Okay.
at least the ones in the Caribbean tend to vary quite a lot. Eric is saying there are so many different species in this one little aggregation. And he's pretty surprised by the amount of chitons as well. Oh, there's another one.
That's the
same one?
one.
Okay.
Drifting back. You say go wide? Okay.
That's
Go
wonderful.
wide.
Okay, I feel bad to be hogging all the dive time, but this was a really
quite extraordinary.
Yeah, a really incredible habitat to spend some time at.
How much time do we have left?
We have Nav. What time are we coming off bottom?
Got an hour and a half left.
Okay, so we still have an hour and a half left. We are close to waypoint 3. Between waypoint seep 4 and waypoint 3, which
Something
you cannot
above
see
it
right now on the
looks like
on the
Tina
quad
or
screen,
something
but that's
coming at
okay.
you.
But we are going to head up the topographic high. What is that?
That hanging in the water above, just above the pinnacle, another squid of some sort, maybe the same.
Can we get a zoom? It looks like it's shining. Is it a ctenophore?
I think it's,
Well,
yeah,
let's
it's
take
not
a quick
squid.
look. Or a siphonophore,
like,
as the gas gland reflecting. What is that?
Yeah, those ribbons that are...
Oh, yes,
Lasers
it's just got
flare.
something caught in that wing. This is a low-bait ctenophore. I've never seen a,
Pulling out.
a yellow one before like that. Yellowish, sort of almost not quite mustard colored, but that is strange.
You drift it back up into the darkness. So you tilt up.
I assume the red inside must be yeah, I think so. That's probably, he's caught a gas bubble. And away.
Oh, that is hilarious. So perhaps one of those methane bubbles
Got caught.
got
So,
caught in his lobes. Oh.
Someone sent me a one-panel cartoon of a parallel to this today of a fish that was fitted with a pair of styrofoam blocks on its tail fin because of transgressions, was sent to sleep with the humans, floated up. So I'm afraid that ctenophore may be doomed to go to the sea surface with that bubble inside it.
Yeah, and that plus that bubble is just going to expand for
Right.
a while. So that ctenophore is in for a rough ride. Interesting.
Never saw one with that coloration before.
Yeah, that was pretty weird, wasn't it?
Try one more
And,
time
and
to get
I'm
the
assuming
bubbles
that
go
dark
straight into the
red
camera lens.
mass in the middle was its stomach gastrovascular cavity.
Okay.
There was another mass beyond this one and to the right a bit. It's just right, I think, right of center beyond this mound. Can't quite tell if that's it or not.
So I just want to remind viewers, this is a seep that no one knew existed before right now, pretty much. We knew that there were seeps in the area. There have been seeps found much more north of here, but this area has never been explored, and so this is a brand new coal seep site to science. which is pretty extraordinary.
And I guess that's just a mound. I thought there was something on top of it. Well, here's some. A really rugged relief here. And very few mussels. See a few down in the crevices maybe.
And some more of those urchins.
And that circular pattern of dead shells again. Very interesting.
So I'm not a geologist. I'll preface this statement with that. But the reason that yesterday we saw this cold seep on this sort of mound, and again, this cold seep was kind of on this little mound, could be because of that buoying of fluids and gas underneath. That chemical-laden fluid, that hydrocarbon-rich fluid, as well as gas, that is what fuels that environment, perhaps may have buoyed the sediment on top, maybe? I don't know. I just find it's a bit, conspicuous that they've both been found on little mounds.
So, Tiva, so sometimes in these deep water environments like this, as I said, gas hydrate is actually the stable phase for methane. So that's a methane ice, a solid form of methane. And so some of these places, and some of us were chatting about this earlier today, sort of behind the scenes, some of these places probably have little domes of gas hydrate right under the surface. And then probably warmer fluids coming from depth can circulate through those and carry methane and other sulfides, et cetera, up through those areas. So that may be one reason that we see these sort of flat mounds as an association with the steep areas.
So, Carolyn, how would you guys be able to confirm the presence of methane hydrates? Just interested.
Well, without punching something into the sediment, which has been done, Ian McDonald and others have done that over the years, it's a very hard substance. And the other thing is that at least in for example, in the hole-mounted chirp systems like the Knudsen that you guys use, you'll see a very hard return on the bottom, but that's not really diagnostic. It's just indicative that there might be something right below the surface.
Not necessarily.
As I said before, in the New England margin, there's a place where we studied this with OER, with ocean exploration, where we do think there's a lot of hydrate right below the surface sediment cover, and then there are these sort of seeps that come up through it, and it's probably very sort of overall similar, except that margin doesn't have the hydrocarbon basin, the world-class petroleum basin underneath it like the northern Gulf of Mexico does.
Amazing. Thank you so much, Carolyn, especially as you are driving home. This is the magic of telepresence, people. Carolyn Ruppel is driving home from Woods Hole to Boston in her car right now, but yet she is able to follow along with our ROV, which is 1,200 meters deep in the Gulf of Mexico, and contribute to what we are seeing during the dive. Absolutely incredible.
Nav Bridge, go ahead.
We just had a shift change down here. I just wanted to know what's the condition up there? How are we doing with the waves and the wind?
Bubbles will probably show up pretty good.
With this heading, is it good, or do we need to
see if I can darken it?
As far as I'm concerned, we're perfect.
Copy that, and then just to confirm, you have payload southeast, correct?
That's correct.
Okay.
So what do we have? New nav.
This is a great overall shot of this community, this chemosynthetic community here. You can see the darker patches of mussels are live mussels, and then there are several different areas of those. We can also see the glowing white bacterial mats, which are, you know, spruced around between, and then around that is this halo of these dead Bathymodiolus mussel shells.
Co-pilot. In SIP 5, we're skipping the new and the old waypoint 3s. Bearing 235, that's correct.
There's the bubbles.
That's a question for
Done.
the back row. No, the co-pilot was just asking me if we're done with the SIP or we want to start heading over to SIP number 5. All right, copy that.
Yeah, what you got there is really nice. Sort of pirouette around it. I'm going to lose the skids. Just keep the bubbles at the bottom of the screen as you get the top. Can you tell which direction they're going? Which is 80-ish degrees.
Let me say that again. What an absolutely extraordinary area of the Gulf of Mexico this is.
Indeed.
Yeah, we have time. So if we start heading there at an average of 0.2, it'll take us about 65 minutes. And that would leave about, well, only 12 minutes over there to check it out.
Would it be
well, but we can go at 0.3 and then we'll get there with 20 or so minutes.
Pilot and nav, we don't have to get there, but when we are heading down that way, would it be possible to try and stick to the topographic high, or if we see corals, because we're trying to see as many corals as possible, but also every now and again, if we could just scan the area for bubble streams, if we're high enough, maybe the sonar will pick it up anyway.
Slowly go down to sit on top
do you have high pack right?
I do.
So this camera is as far down as you can get, keeping it in view.
Yeah, something like that.
Now I'll zoom to it. You just
I guess it depends what we start to see. If we see the corals are more along the flank of the slope, or if they're more along the top, we can kind of make a decision on the fly. But yeah, proceeding towards the point SEEP5 generally. That sounds great. Thank you, Nav.
Good framing.
So I think we're getting our final glimpses of this new cold seep site. Yep, we've got Bathymodiolus mussels, which are the very large mussels occupying most of the screen. I think this may all be one species. But they are different sizes. So either it's two species or there have been different stages of recruitment. So there may be several different populations of this species here.
Yep,
we can also see the Leptochiton micropustulosus. polyplacophoran, which is that chitin animal with the segments attached to the shell of that Bathymodiolus. We've also got a gastropod, potentially Canoe miroglypta, which is one of the chemosynthetic bacterial grazers found at cold seeps in this area of the world.
Right,
and then we've got this beautiful urchin, which
section of the
I don't think it's the one we thought it was. Maybe that one's Gracilichinus alexandri, because it has a pinker tinge.
Yeah, all right, I'll buy that.
And we have been seeing Gracilichinus affinis throughout the dive, but that one was whiter.
Yeah, it does look different.
Tilt the camera up
as well as there's a little zooacid fish,
so
probably from the genus Pachycara. I'm not quite sure of the species, as well as some Munidopsis squat lobsters, these white guys, as we're looking off at this carbonate mound. And then some more of these, what would be top predators in this environment, the Chasian quinquidens, red crab, as well as making the most of this carbonate outcrop. At the very top of the carbonate outcrop, we had a couple corals. Really beautiful, and a great example of the zonation you get in these habitats, and on very small scales, and also how patchy these habitats can be, that there can be this abundance of life, and then just 10 meters to the right, we're back into sediment. And again, some great shots on camera 2 from Sirius.
Good job. Thank you, sir.
Thank you very much, pilots. That was great.
You're for what?
Alright, co-pilot. So the science lead requested for us to not go straight to SIP5, but try to stay kind of going up the ridge. So maybe we'll cut it, instead of going this way, maybe halfway. Maybe I'd say 240, the first move, and then we'll see. They're looking for coral, so we'll see. We'll take a decision then.
Yeah. All right.
Yeah, 240 is good.
All right, yeah, you can call moving.
Okay, 240. Let's do 20. Do you want to do 25?
I'd be fine with 30 or
all right,
40.
Let's do 30.
Yeah, that'd be
to begin with.
Go stop it. Yeah, sounds good. It's shallow.
Nav bridge, go ahead.
Next move, please. It's gonna be range 30 meters, bearing 240 degrees, speed 0.2.
Does tether act quite a bit differently than the previous tether? It seems more malleable, which is nice. Yeah, definitely.
Good copy.
Yeah.
Stand by for input.
It's got a little more range before you get that hard tug. Yeah. Seems to flow nicely. Yeah, for sure. Acts a little different, yeah, but that S.
So we have a little bit more than an hour left in this dive at Henderson Ridge Mid-South. It's an area we've been exploring for coral communities, which we found some absolutely spectacular ones of, as well as chemosynthetic communities, which we just left one. So it's been a pretty good dive so far. We've also found some asphalt seeps. So last night we detected 4 different bubble streams, which do indicate sometimes these chemosynthetic habitats. We have passed by 4 out of 5 of them. Sorry, it was 5 of them. We have passed by 4 of them and found 2 chemosynthetic areas, one which we do suspect was linked to that bubble target we imaged. And now we're going to be heading across the local high in this region, this area, sorry, to see what may be living on that, and then finally finishing up close to our final seep target, hopefully. We've seen a huge range of invertebrates, many echinoderms, many cnidarians, many arthropods, Just a real diversity on this dive.
Mollusks, polychaetes. But I wonder what activity strews these shells randomly around the sea floor. Is it? I have no idea. It's.
So is it like a chicken before the egg thing? Perhaps this whole area was active once upon a time, and then it's died off, and some have been buried and some have not. Or perhaps they were centered more around some other area, and when they died, scavengers then dragged them off.
Okay.
Like for instance, the Chasion quinquidens we've been seeing. I'm not really sure, but they do seem to have quite some difference in size. Can we just get a zoom on that mound ahead? That's not the mound we just came from, right?
Negative.
No. It looks like there are some like stringy things closest on the side closest to us.
Aye.
They could just be dead corals.
Let's go zoom, roll.
Yeah, just a quick zoom.
Copy that.
Just a quick zoom, yeah.
Yeah, dead coral.
Oh yes, small branches.
That's great, pilot. Thank you.
Sure, we're headed that way too. So
okay.
if you wanted a closer look, just
there's something purple in the middle. That may be that clavularia encrusting octocoral growing over a dead branch.
I got excited and thought it was an octopus. It's not.
That charismatic megafauna.
Gets everyone. Gets everyone.
Yeah. Although it's funny, sometimes when I take my invertebrates class on a field trip and I'm trying to focus on the corals and sponges and crustaceans and so on, and a school of fish swims by, they get distracted. The students get distracted.
That's so funny because I think a lot of marine biologists would say that invertebrates are just so much
purple
cooler.
Maybe
we're biased.
Yeah, I guess.
But
that's totally biased.
It's like I have this fish tank at home. It's a freshwater tank, but we've got loads of different species of fish in it. But then we have 2 crayfish, and we got them when they were like a centimeter big, and now they are, I don't know, 8 inches long. And they are by far the best thing in that tank to watch, definitely. But again, maybe it's because I'm biased.
That's it. There's an interesting. Is that one of the heterocarpus? That's something different. I can't
Difficult
tell.
to tell from this angle.
Yeah. Need a profile view.
But some more of these, as we were saying, some of these stoloniferan clavularia.
Yeah, that looks like heterocarpus.
Corals, as well as squat lobster behind.
He's ready for his close-up.
Did you want a tighter look on the shrimp?
No, not necessary.
Okay.
Thank you, though.
Sure. Okay, video. I think we can come wide here.
Pulling full wide.
Those also look like some dead branches of octocorals in there as well.
So in the distance, can you see those?
Oh yes, there's something out there.
Communities
Mounds.
looming out of the depths. It looks like, again, we're about to enter our, 3rd, 4th coral forest for this dive?
That's what it looks like. And that mostly looks like stony coral.
Yeah, definitely.
Again, all oriented. They've all grown up over the decades in the same orientation, indicating that the flow is pretty uniform down here.
1 or 2 meters
If
left
it
on this
came
move,
from
co-pilot.
different directions at different times, we would expect these to look far more bushy, taking advantage of flow from different directions. But these all seem to be oriented in the same pattern at right angles to the primary direction of flow.
Copy. Yeah, they said that getting to SIG 5.
And again, similar to what we were seeing before, a big portion of it is dead.
Yes.
But then very, very much alive colonies as well.
Co-pilot, what were you saying about the port swing around there? We might
port lower looks like it's just
and even more further in the background to port
fish left of lasers.
But this I'm assuming is all. It looks like it's all Madrepora oculata, Diva.
It does look like it. Oh, whoa! Look at that
There's
big
an
bunch.
interesting, and that's a strange purplish gray fish.
Can we get a quick zoom on that?
Front and center.
Just snap zoom here, Roland.
Pushing.
That looks a bit different.
That looks way different. Look at his lips.
I think we've seen a member of that family before, but I don't know what. It's an ophidiiform, I guess.
Perhaps bithytid.
But maybe somebody on shore.
Kevin stepped off for the day, unfortunately.
Okay.
But yeah, it looks like a brotula, one of those. Not an ophidiid, necessarily. But perhaps from another family within the Ophioidiforms.
Ophidiiforms.
There you go.
Great shot. And can we get a zoom on one of those white coral fans when you have a moment?
Pulling for full species. Tilt up.
Tilting.
And it's just hanging out there, chilling out.
And video's clear.
Copy. Just come wide. We want to get a zoom here.
There's a Paramuricea, although that's
see if we can get it from
purple one. We've mostly seen the yellowy orange ones today. And we'll get that sea star eventually. So this again looks like Madrepora. And there's a little, is that a homola up top left? Little homolid crab.
Stand by for input.
I think.
I can come in a little, Roland.
Yeah, that'd be great.
Yeah, because I'm
okay. I'll
pretty
creep in.
far out there.
Copy. Hold there. Hold there.
Holding.
And there's it looks like they've formed a an arcade over an empty tunnel almost. This arcade of coral branches.
Okay, let's come back in.
Spanning an empty area, and in the distance underneath there's a squat lobster and half a dozen or so solitaries. And here we have, yeah, that's a homolid crab.
I think it's a car star.
No, it's holding. I see it's holding its two hind legs up in the air, up in the water. That would normally, it would hold a glass sponge or something else like that. And then there is a small shrimp. Well, I don't think it's a car. Maybe it's a chirostylid, but look at the way it's holding those legs. I can't tell.
I agree with you. It is a really weird way for it to be standing, but
Right.
but I do think it looks like a Gastropticus.
And here are some those. I haven't seen those galathioids before.
No, that is a new sighting for this expedition, I think. Definitely not on this dive. Those that look like new squat lobsters.
And Scott France tells us he thinks this is Madrepora oculata. And I would
I'll
get
look at that
just
picture, but you're a funny crab. Maybe belong to a family called the Homolodromiidae.
Okay, that's what I thought. Because I thought I saw 3 pairs of walking legs and then the last 2 held up in the air. I keep saying that. In the water. Thanks very much.
Sure, I'm just
and these guys are probably in the genus Urotychus, but they're not the one that you've seen previously, so I'll have to check them out too.
Okay, thank you. That's Mary, isn't it? Yeah, hi Mary.
Sure is.
Mary
Hi,
Wixton.
Mary. Texas A&M. Thank you for joining us. She is an arthropod specialist, especially the shrimps.
And Mary, those two Urotychus squat lobsters that are now off view, one had thicker claws than the other. Is that likely a mated pair?
Let me know when you're clear. We can.
Could well be, but what you want to look at, this is whether they have a gate between the fingers. Like this guy who's gesturing at us is definitely a guy.
Okay. That's something new. I didn't know that.
Yeah, that's really interesting, Mary. Thank you.
I think we're pretty good. I don't think I've seen that overoid.
Can we have pilots? Can we have a zoom on that pair right in above center, directly above the lasers again?
Sure.
Thank you.
Stand by, one video. Okay, let's come back in.
So Mary, when you, I don't know what the delay is where you are, but we're going to zoom in on this pair. And the one on the left has got the much thicker major claw, and it's right-handed, I guess. The heavier one is right-handed. And the one on the right has got a longer left-handed keela.
A little for you.
Well, some of them, the right or left-handedness is species-specific, but in others it isn't.
Oh, okay.
Go ahead and rock back to the right.
Well, they look like they're a pair, whatever they are. Thanks very much, pilot.
You got it.
Video's clear.
I'm on the move. I got you at 60. So.
There's quite a bit to look at here, pilot. So if we could just spend a little bit of time here, that would be great.
Coral feature?
Yeah, at this coral
partial
area.
Okay. Is the ship moving, guys?
Yeah.
Let's get a stop then.
So do we?
Great.
This coral we're seeing in the foreground, it looks like the Paramerissia we've been seeing before, but it's got
purple.
purple major stems.
And we've just lost Scott,
but Les is there as well as
Eric.
and Asako and a couple other.
Do either of you know whether this is just a different clade of Paramerissia or a different genus?
Pull full wide so you can get in close unless you're using another camera.
I'm using another one.
So Scott is still there, and he says that is Paramerissia. But is this a different species to the one we've been seeing before, Scott? I don't remember seeing purple on the colonies we were looking at before, but maybe I was just unobservant. Sorry.
We'd like to make a heading change up here.
I know we've seen them. I saw them on my other cruise in the Gulf of Mexico, but.
Okay, video.
So he says there are some colonies that have both yellow and purple.
Nav bridge.
Yeah, go ahead.
There's a lovely view.
Just make sure that the center of rotation is at 8.
There's a little amphipod left of center with a yellow, pink with a yellow eye.
But look at the contrast between those two colors.
Especially this stuff.
Yep, that's
like
correct. We're going to make a heading change of 5 degrees to port for now and see how that works.
Growing up.
All right.
So Scott says a range of from yellow, mostly purple, or a combination. Scott, if you can weigh in, maybe. I remember, of course, some of the shallow water plexaurids have purple sclerites. Is this purple color due to the whatever mineral it is in the sclerites themselves?
There we go. That's better light.
So Eric is saying this is Paramerissia B3. So this is the same species we were seeing earlier in the dive, and it's this known species but not yet described species. So we do know this exists, but it just doesn't have a name yet and hasn't been described formally.
Polyps on the right are closing.
And there's just above center, behind one of the polyps, there's a little amphipod, pink with a yellow eye.
The kind of detail that D2 is able to pick up is just mind-blowing.
So Scott says the pigment he thinks is free in the tissue rather than bound in the sclerites.
Yeah, I see Andrea Quattrini's also just jumped in and said that she agrees the sclerites are not purple.
Thanks very much, folks. That's magnificent.
Okay, that's wonderful, pilot. Whenever video is happy, we are
partial.
Copy that. Go ahead, Roland.
And another shrimp.
Easier to hide my focus. issues.
Mary, any idea of the ID of this shrimp? I know that's your thing.
I got my rotator maxed out, but I think I'm just settling out here, so.
Copy.
This is going to be another
Titan
basic
two,
palemonella, which doesn't seem to be specific to any particular kind of coral or soft coral. Unfortunately, there's supposed to be two different species that are in the Gulf of Mexico, and it's almost impossible to tell them apart. And in fact, some people think, in fact, that they're the same species. It's just the description was of a small one and a big one, so that remains to be seen.
ROV nav bridge, that heading change is complete.
And that's an anemone that we have not seen before. A little one. If it was an anemone and not a zoanthid, that looked more like an anemone. We get on the east side of Florida, we get a larger white anemone with an orange mouth. Belongs to the family Sagartiidae, but that did not quite look like it.
This
So
really
long,
is
Scott.
This really is like a thicket of coral. You could almost imagine walking through that tunnel of coral with it over your head, like a rose bush kind of thing.
Originally, or quite a number of decades ago, I can't remember who it was, Wilson, I think it was, who gave terms for the different sizes of accumulations of these deep water coral colonies. And so there were, of course, individual colonies, there were thickets, and then I think this is more of a term used by, in Britain, he'd say larger than a thicket was a copse. C-O-P-S-E, which is a term used in, I guess, forestry and agriculture. I'm not sure. But look at this. These are just very impressive. This is more than we've seen. This is still Madrepora. Some of it is dead here. A lot of it is alive. And this is beginning to be an accumulation, where you have live coral growing on dead coral to a considerable extent.
See it from both sides.
And here you see that these colonies are a lot bushier because they appear to be shielded from the overlying flow by the mound behind. So they're growing out bushier than the fan-shaped ones we saw before.
Just asked the pilots to put the lasers on just so we could get a scope for the size of this colony of coral, and you can see this is many meters across, and if you include the dead coral, many more, nearly double that.
And this is interesting because this begins to approach the architecture of some of the deep coral accumulations on the other side of Florida under the Gulf Stream, where you have dead coral forming these literally windrows and the sediment accumulating and building up, and the living coral tending to occur a bit on the down current side, if I recall correctly. Sorry, John Reed's not here. He has much more experience with the east coast of Florida. But this is beginning to be an accumulation. There's
beginning as of what, hundreds of years ago?
That's a good question. How long ago? Could be certainly hundreds, I would guess, possibly more. Of course, on the east side of the Florida Peninsula, we have mounds of the Coralophilia built up that are over 150 meters high.
Push out.
And in the North Atlantic, they are even larger. And in the North Atlantic, the deep sea coral reefs have undergone periods of increase with live corals and then completely dying off and being covered with sediment and then increasing again. And there's a midwater fish that looks like a stomiatoid, perhaps. I can't tell if that is a bristlemouth. That might be I guess that's a bristlemouth. Anybody on shore can help.
That is cool.
But in the North Atlantic, those reefs are so enormous that they have been growing for hundreds of thousands of years, and the dieback and regrowth is in sequence with the ice ages and interglacial periods. So the corals die off when the ice caps expand, and then regrow when the ice caps retreat and there's surface productivity to contribute to their
I'm at 235
nutrition.
right now.
Okay, we can go back that way.
So this is certainly not that old.
So right now, SIG 5 is selected on bearing 235.
And Les suggests that the flow here is generally left to right.
30 meters good?
Yep.
It's not now, I don't think, but the coral growth informs us that the mean flow is in that direction.
Bridge.
Come back down a little.
Left to right.
Now, Bridge, go ahead.
With the dead coral up current.
Range 30 meters, bearing 235 degrees, speed 0.2 knots.
Bridge copies. Next move, 30 meters, bearing 235, speed 0.2 knots.
That's a good copy.
Stand by for input.
Pilot, could we take a look at those little little white dots just above the urchin, left of center? Those look like solitary corals. I'm not sure whether we've seen that kind before or not.
Sure thing.
Thank you. And Les, or Andrea, if you can help us out with these, maybe a genus name when we zoom in. Look at the color on that. That's beautiful. That
see if we can get
pale
from
peach.
Video. Go ahead and.
So those are gorgeous. We may have seen them before, but so these are solitary cup corals and Munidopsis squat lobsters. But any idea, are these?
Let's just keep it here.
Or bring a lower
Desmophyllum?
inboard,
I'd
just a
have
little bit,
to
maybe some
look.
more
There are
light.
so many
help you out a
genera
little bit.
and species.
Is that better or not?
Didn't notice
Let
the
me see if
difference.
I can dig that one up in a hurry. I don't have Steve Cairns'
Looks like it's
monograph
bright from the
with
top.
me, but let me see what I can come up with.
Let me know if it gets better, Roland.
Might not different. Not seeing any change in brightness.
So they're oval and not circular.
Yeah, Dan Wagner's suggesting Desmophyllum.
Yeah, that's what I would guess too from my quick scan of
You want to try to do it tight on one of them, Roland?
Yeah,
of the web, and one of the common
got the
species, at least in the
tentacles.
North Atlantic, that grows with these corals is Desmophyllum christogalli. But let's see what he says. Yeah.
Sorry.
So I don't know how many different species there are.
But
that's beautiful.
Les Watling from the University of Hawaii is suggesting that the large
Lighter
flukes of these cup corals suggest also it could be Desmophyllum. I'm just not sure what characteristics you use to distinguish these.
It's the pattern of the flutes. Those partitions are called septa, skeletal septa, and their arrangement, the number of cycles,
We are on the move.
their
looking down at you.
relative proportions,
So,
the ornamentation or sculpturing of the skeleton
Pull that
down
as you
in the
lift off.
center, called the columella, whether these septa extend over the edges as ribs, whether they are circular or oval like that. So my brief foray onto the web suggests, yes, that they are
Push out here. Full wide there, video.
Desmophyllum, I agree.
Okay, that's fine. Go ahead.
And I've gotten something on what has happened here.
The
I
yellow
think you just got kicked out.
No, but it magnified.
So as we're continuing along, this colony looks a bit different. If we could have a look at this very big colony that we're just about to pass, the one which is just where the lasers are to the left of the yellow, it just looks a bit different to the Madrepora we've been seeing so far.
I'm guessing it's dead.
It's still got commensals.
Let's fill frame here, Roll, and see if we can figure out the mystery.
Ooh, what is happening here? That's weird.
Okay, keep going in.
I'm confused by what we're looking at.
Well, the polyps are much denser.
But it looks like...
I think it's a dead coral covered by zoanthids. What do you think?
Yeah, I think that's a pretty good guess.
Especially up on the branches up towards the top.
Yeah, and you can see the sort of sedimentation in between.
Slowly go down.
Copy.
And there's a hydroid there.
Because there's some live polyps.
Some of it may still be alive, like right here, unless that is Solena
Stop the
smilia.
ship, I think, if there's...
I can't quite tell. No.
Yeah, I think you might be right. I think that's a pretty good synopsis of what's happening here.
Yeah, you can see on the branches how the tissue of spreading out from the polyps is discontinuous.
So
that's what this looks like, some kind of colonial anemone
8-10 meters
covering. Very interesting.
Yeah. Lots of really interesting sights on this dive.
Right.
Thank you, pilot. That was great.
Sure.
Watch lead. This is Nav.
Go ahead, Nav.
Just to let you know, we have a little bit over 40
here,
minutes left on the dive.
Shrimp looks interesting.
So we're going to be coming off at just at 4?
About 5 minutes before 4.
Okay, thank you.
Okay, clear.
Copy, come for one.
And we can also see these bright yellow colonies, which I think we said were Enelopsamia before. And now we're shooting off of this mound.
Yeah,
and we can see, that's okay. And it was very dramatic. And now we can see some more potential Madrepora in the distance, as well as some dead areas of Madrepora. Or that might be asphalt again. They're very difficult to tell apart. Look at this.
This is much steeper than we've seen before as the slope runs upward.
And so Dan Wagner has just pointed out a great fact in the chat room that this is a perfect example of the value of these habitat-forming corals. They form these 3D structures which, when they are alive, give a home to many different animals, but also when they are dead, they continue to create habitat for many deep-sea invertebrates, as well as vertebrates. Thank you, Dan.
All right, co-pilot, we should be at the end of that move.
Okay.
ROV nav bridge, that is a complete move.
Fish coming in from left.
Copy.
I think we can get another one in whenever you guys are ready.
I agree.
All right, same bearing. 235 is good.
Maybe 240.
Copy.
But yeah.
So,
okay, let's go ahead and get it.
Les has thrown out the idea. This looks like that Corophenoides that we saw earlier, Mexicanus, with the black ring around its eye.
Yeah, I didn't get a good look at it before, but that black ring is very distinct.
Speed 0.2 of a knot.
Bridge copies. Next move, 30 meters, bearing 240.
And Les has mentioned that in his work on the Atlantic seamounts, it was clear from the paleo temperature analysis done on these desmophyllum, those solitary, more or less solitary corals, that their numbers were real high during the last ice age.
I'm going to let him go.
He's heading out of town.
Full line.
He's on a mission there. Copy, full one.
So with the
nav bridge moving
slabs of what I guess are orthogenic carbonate here suggests that this was a seep at one time rather than an entire coral buildup.
This is Carolyn, and I think it's important to note that you don't necessarily have to have methane seeping out of the seafloor.
I think you definitely got some seepage highlight material.
So, there's flow of fluid, certainly, a supply of fluids, these microbial processes that result in orthogenic carbonate are taking place, but that doesn't necessarily mean you have to have methane or other fluids fluxing into the overlying ocean waters.
Well, thanks very much, Carolyn. Listen, one quick question about this. Is the deposition of the carbonate at all bacterially mediated, or is it strictly a chemical, geochemical phenomenon?
Just wondering how the ship is doing with that.
Do bacteria have a role? Oh, it is. Okay.
Because of anaerobic oxidation of methane.
Nothing
Okay.
really distressing, but it
Thanks.
is ramping up.
All right, let me know if at the end of this.
So we have bacteria building rocks.
Yeah, we're looking at it right now.
Copy.
All right.
And I have to say, I'm not the person to ask about bacteria versus other things, so I just call them microbes. So you'll have to get one of the other people in the chat room to say something about exactly what it is. But various archaea, et cetera, are responsible for some of these processes in some places.
Great. Thanks very much.
So we have just over 40 minutes left in this dive. We are currently at a depth of 1,135 meters roughly at Henderson Ridge Mid-South, which is in the southern region of the Gulf of Mexico. Oh, that was exciting.
That was a polychaete, I think.
Yeah, definitely. And we have been exploring a ridge-like feature. There's not been that much difference in
overall elevation or depth.
But they have it has been visible, and it has proven to affect the diversity and abundance of animals here. But also the animals have been very affected by differences in the geological nature of the area. We have seen everything from a miniature asphalt seep to several methane seeps, and then also to asphalt seeps that have been supporting huge colonies of coral, as well as orthogenic carbonates that have been supporting huge coral colonies. It really has been a dive of very mixed animal life as well as geology, and it has been a really fascinating one.
And Carolyn, if you're there, if you can correct me if I'm wrong, or Diva, authogenic, A-U-T-H-I-G-E-N-I-C, means built in place, right? Or is there another meaning for that?
I have no idea.
I could look it up.
I think that's about right. But I mean, the autogenic, when we refer to autogenic carbonates, we're definitely referring to those that are microbially mediated in terms of their production. So it's an entire biogeochemical process that occurs. So it's not just sort of the deposition. You do get carbonates. They're deposited as the seafloor that rain out essentially from the water column. But these are carbonates where there's a biology mediating to make them be deposited, which is why we suspect they occur primarily in steep environments.
Gotcha. So a shallow water limestone deposit created by ooids or pelagic deadfall of forams would not be called orthogenic.
Right.
Thanks.
I've got you at 7.5, 10.4.
Looking good.
Some more of these dead coral colonies here in the foreground. Some newer ones, which are still white. And then there are some ones that have been overgrown by various
organisms and crusts.
5 meters left on that move.
Copy that now.
And then it looks like a lot of this is coated in a white. If we could get a snap zoom at some point, it looks like some of these rocks or coral pieces are coated in this sort of white fuzz. I think it might be maybe stoloniferans, a different.
That's a complete move.
Oh, that's
different species. Do you see what I mean?
Let's take a look. Yep, bingle bangle bungle. Good eye. A little transparent.
I don't think they're stolons though.
Well, they're not. Those are octocorals, and if they're encrusting, that would put them in stolonifera. They're not telestasians, which grow up as tall branches with side polyps.
Maybe we just can't see the ribbon, the connecting
right.
ribbon.
I think that's uncovered, but I would guess that these are stoloniferans.
Is that the max down?
They're definitely octocorals. You can even count the tentacles and see that they're pinnate with the little side branches.
You want to get closer?
Yeah.
Hmm.
There you go.
So this is a potential second species
Right.
of stoloniferans for this dive.
Anybody on shore think that we should collect this one? Or is this a known taxon? Les or Eric?
Or Andrea, because throughout the dive we've been seeing these very deep purple stoloniferans, likely from the genus Clavillaria. But here, this is likely something different.
And Asako has asked whether we could pick up a small one of these.
One at the end of the heading change.
Also, Eric mentioned when I was talking about the age of the coral mounds that the whole evolution transition from seep to coral mound is probably in the tens of thousands of years. And they, he and his team cored a large Lophelia, which is one of the other deep water coral species, mound building species, in the northern Gulf, and got an age of 300,000 years. So there are areas of major accumulation here.
Change to port. We've got the current shifting around on us right now.
Copy, Bridge. Another 5 degrees to port.
That's correct.
So if we can...
Okay, so pilot, would it be possible to collect one of these pieces of rock with the stolons on them? Stolonifera?
Yes, I think so. Absolutely. Try to find a nice...
That one that we were looking at looked like it was a potential collectible piece.
Yeah, I agree.
Hydraulics spun up.
Copy that. Go
I think we're comfortable here, Nav, for now.
Copy. I don't know if they want to put it in
Okay, so we are setting up for our third collection of this dive. The first was that unknown
used at the moment.
Plexaurid, right? The pale purple.
Okay. So either
pale purple plexaurid, if I could be alliterative.
And then we also collected a piece of carbonate, orthogenic carbonate. And now we're going to be collecting a rock with these stoloniferan octocorals on them. Our collections during this expedition and all Okeanos Explorer expeditions are limited. We do tend to take a maximum of 6 collections per dive, but those need to encompass both geological and biological collections. So we need to be very careful about what we choose, and so that means that we tend to stick with species that are new or unknown or rare, or could be a new record for that area, or a very characteristic. And so both of the ones we've collected today, the biological collections, have been perhaps new or very poorly known.
Well, Daniel Wagner has just chimed in, says the only stoloniferans reported from these depths in the Gulf are the Clavularia rudis, which is the purple one, and Telestula tubaria.
You're welcome.
And Les says if they are solitary, that would be a great collection. We'll have to see. One way to find out. This rock will be, and of course then the rock may have some interest as well, not just for the critters on it. And this is going to be only about 4 inches long. If it's the one next to the lasers. I'm not sure which one they're going to go for.
Oh, so Andrea's also just said that they've collected 2 stoloniferans that have yet to be identified to species, but those actually do not look like either one of the species we've encountered today.
Brilliant.
Yeah.
Probably port outer, maybe.
Okay.
That's what I'm thinking.
Not me, not me. You can't get me. Oh, good. Get both. They're both of those rocks. Looks like they're loose.
See 3 small rocks here.
Yeah.
Looks like I have 2 of them. I wonder if they might fall apart and fall out. I'm trying not to crush them right now.
There looks like there are some polyps on that, so that should be good.
Although it might be crushing it right now. Yeah.
See how strong of a rock it is.
Well, if it breaks, you can try another.
Yep. I think there's quite a few options here.
You want to go ahead and extend?
Yeah. Okay.
And Daniel Wagner mentions that there are an additional 9 species of stoloniferans reported from the shallower mesophotic depths. That's, what, 50 to 150 meters or so depth, where there's some light, but enough for photosynthesis, but not a whole lot.
All right. Nice.
Could always go in and grab another one.
Yeah, watch lead pilot. Watch lead pilot.
Go ahead, pilot.
That was a pretty small one. Would you like an additional one? Or to throw in the same box as the same sample?
I mean...
Because it fell apart there?
Oh, sure.
I mean...
It won't hurt.
We could move on, but if you needed one, we could.
I'm inclined to say no, because I think there would have been quite a couple on that, those 2 rocks.
Okay.
But thank you for offering. I do think we're going to be able to get what we want out of that. There should be quite a few polyps on those, on that rock, which then fragmented. But thank you very, very much.
No problem.
That was a great collection, our 3rd collection.
Okay. Nice. Drawer secure and hydraulic secure.
Yeah, we didn't drift that far, so
yeah. Good. Moving on. First sample of the cruise for me.
Really?
Yeah. I poked at a rock and it like crumbled apart. It was It wasn't a. It was continuing to squeeze. Grip force 3. So it was a strong enough rock. Affirmative. All right, video. You want to snap on fish as it floats by?
Coming up.
We have Bob over there going now. Sorry, Bob.
What do we have here?
Hello.
That's interesting.
Wow, he looks like he's just eaten something very large. And is now waddling off.
What is that? Very large pectoral fins with maybe a parasitic copepod attached. That long yellow white string. Is this Chiasmodon? Is this a black swallower? Wow. Somebody from the mainland may be able to help us on this.
The food is so good on this ship, that's how I look every time I leave the galley.
I'm guessing that's Chiasmodon niger. Why don't we look that up?
Let me know if I...
Lauren Jackson is saying yes.
And, you know, it's interesting because that is one of the fish that I learned about when I was a kid. There was a Life magazine series called The World We Live In, which had these wonderful fold-outs of different environments on Earth, both terrestrial and marine. And there was one that included the deep sea. Yeah. And I remember Chiasmodon niger, the black swallower that could consume prey significantly larger than itself and expand its stomach. That was very cool. I did not expect to see that at all.
Oh, yeah. Look at this picture on Google. Amazing. I wish we could pop that in a CT scanner. Oh, I've just cut it open. See what it just ate.
All right, coming back into your view here.
That was amazing. That was such a distended.
And they get much more distended than that.
Surely when they eat something that big, though, then they are very vulnerable.
I would guess. I don't know how, you know, we need Ken Sulak to tell us about that.
Is your camera partially zoomed? Huh? Yeah, Sirius is full wide? Seems.
Yeah, I don't know if it was Chiasmodon niger.
Bingo.
But
I don't know.
Lauren Jackson gave us the family Chiasmodontinidae.
I knew something was a little fishy.
And that one looked like it had photophores, which she said was interesting.
Was that the first time you've ever seen that alive?
I think it was like
Absolutely.
Amazing. That's so amazing that you could have seen something as a child in a magazine, and now...
I remember it.
Yeah, but not... No, not that you remember it, but now you're getting to see it.
Oh, yes.
In, you know...
Well, it was remarkable that I remembered it. But there were, there were reinforcements along the way.
Looks like that move's complete, or are we underway here?
Yeah, whenever you guys
I'm
are.
not sure, but I think
Alright.
William Beebe's half
Call one
mile
in there.
down in the bathysphere may have mentioned that species too.
220.
I'm going to have to look up and see how
last
many
one was 240.
species are in that family.
Yeah, so Wikipedia says it's known for its ability to swallow fish larger than itself.
Yeah.
Worldwide
All right.
distribution, mesopelagic and bathypelagic zones.
Leave it, Charlie.
And it feeds on bony fishes, which are swallowed whole.
Video, let's come
With
in,
its greatly
see if there's anything in
distensible
here.
stomach,
Sand
it is
here.
capable of swallowing prey over twice its
ahead
length
now.
and 10 times its mass. Its upper jaws are articulated with a skull at the front via the suspensorium, which allows the jaws to swing down and encompass objects larger than the swallower's head.
Copy, that is 30 meters.
Theodore
bearing
Gill
220
speculated that
at
the
decimal
swallower
2 knots.
seizes prey fishes by the tail and then walks its jaw over the prey until it is fully coiled inside the stomach.
This is a science fiction movie
Black
waiting to happen.
Black swallows have been found to have swallowed fish so large that they could not be digested before decomposition sets
video,
in,
come wide.
and the resulting release of gases forced the swallower to the ocean surface. In fact, this is how many of the known specimens have been collected.
ROV nav
And in
bridge,
the
move
family
initiated.
Chiasmodontidae,
30 meters, bearing 220 at decimal 2
I'm
knots.
about to find out. There are, wait for it, wait for it. There are 4 genera in the family, and the genus Chiasmodon, which is the one I thought it was on very little basis, has 9 species in it. So it's not unique. One of the genera in the family is called Kali, K-A-L-I, which I believe is the Hindu god or goddess of death. You know, we taxonomists do try to entertain ourselves with naming some of these things. So a former classmate of mine in graduate school was working on the parasitic isopods. And of course, we saw the giant pill bug, that's an isopod, but there are isopods that parasitize crustacean, little shrimp, and large shrimp too. And they get in the under the carapace in the gill chamber and cause it to swell up so that the shrimp looks like it's got a cut plug of chewing tobacco in its cheek. And so there's this little grass bed shrimp called, the genus name is Thor, after the Norse god of thunder. Don't ask me why. I'm not sure who named it or why. But my classmate, John Markham, found a parasitic isopod that only parasitized Thor. And what do you think, what name do you think he gave it? Anybody watching the Marvel film series should know Loki is right. But on top of that, he even included a quote from the ancient Norse prose Edda, some of the writings about the mythology, in which Loki says to Thor, "Why chafest thou so, Thor?" And Thor responds, "Silence, thou impure being." And I'm sure the parasite makes the shrimp chafe.
Lots of these.
That's very appropriate because you just had a close-up of a Munitus shrimp that had one of those isopods under its carapace.
Oh, I missed that. Thank you, Mary.
Yeah, it looks like everything here is a little fuzzy, and that's because of these stoloniferans that we just collected. Seem to be carpeting everything in this area. They're not very easy to see, but yeah, it does look like everything, especially the rocks in the distance. It
Yes.
could almost look like bacterial mats, but it's not.
Right, or sediment.
Yeah.
Right. Yeah, it just brings to mind again the very disjunct appearance and disappearance of these different, not quite micro-habitats, but very localized habitats where for much of the dive we didn't see this stoloniferan at all, and now it is just carpeting all of the hard substrates.
Yep.
I think that feature that you're looking at now is what we're seeing.
And Carolyn, if you're still on, are these large pit-like craters, are these remnants or associated with release of anything from below the surface?
So she may not be able to.
Okay.
Because she's not watching.
Oh, right,
she's only
right.
listening.
So we've got this relatively low relief area with a lot of small slabs of the orthogenic carbonate.
You can frame it up.
But then there are three
zoom in video.
Three large craters. I don't know if they've been dug up by things. We've got some of the red crabs.
He's rearranging.
I don't know that.
Right. So
Go ahead.
Definitely excavating.
Okay, so my daughter, whom I have now fetched, is sitting here with the iPad, so she's tuned into it, showing me. So we do see so-called pock marks, as you know, associated with methane venting, but it's really hard to look at a feature on the seafloor and genetically say, you know, whether it's biological, which is your realm, or whether it's related to methane venting. So I'm not sure that we would know for certain, but one thing that I think you just mentioned, it looks like there's some like oxide staining around some of them, which might indicate, or may not, it might indicate that there's some kind of process going on with methane and with oxidation and sulfides, et cetera. But I don't know that we can tell just by looking at them.
Gotcha. Thanks very much. And here are two dueling. Well, one looks considerably larger than the other. This, I suppose, could be courtship of a sort. Oh, that's possible.
I was thinking these might be two males. Then the smaller one is about to lose this fight. And that female was clearing out the
burrow, the crater
yeah, because
there.
you definitely saw her picking up a rock and taking it out before.
That's...
Or, I mean...
Right. Let's see where she is now.
All right,
well, she's down in there and half covered with sediment. That's a lot of sediment to move even for a crab like this.
Go ahead, Bridge.
Ship move is complete.
I'm more stable, so you can
Yeah,
come
so
back
perhaps
and
she's
compare
ready
here.
to mate, and he
closer
is,
zoom
well, one of them is fending off the female that it had secured, and from this new aggressive male that's come in to try and mate with her instead. I'm not sure which one was the original male, but
Of course not, and they're covered both with little gooseneck barnacles.
and again, that's an indication of the fact that these crabs wouldn't have mated for some time.
It's just retracted there.
Oh, there's some movement here. Let's see what happens. Oh, one of them has apparently decided to back off, at least temporarily.
And what is so great about the Okeanos Explorer as a platform is that with a lot of science, you know, pure science, we tend to go to places and do experiments and we have very direct objectives, and we don't ever just sit somewhere and look. And so here we're getting this chance to observe behavior, something that we don't necessarily get the chance to normally see.
Right, and whenever you look at, in most cases, with few exceptions, studying the behavior of invertebrates requires a great deal of patience.
Yep. And so, I mean, that's where these deep sea observatories are coming in handy now, because they're able to just observe behavior. But we don't normally get the chance just to hang out and watch some crabs fight.
I'm still on heading.
Did you notice that one of them had a broken claw?
Yeah, so maybe there was some dueling before. Maybe this isn't his first attempt at trying to get in to mate with this female. I mean, we've just made up a whole story which may not be true.
Yes, because neither of them seems to have any particular interest in the crab in the burrow.
They could have been having an argument about something totally unrelated.
Yeah, one's a Steelers fan, one's a Packers fan.
So Les is just saying that the male is on the left by the looks of it. So it may not have been a mating ritual. Both may have been males. So, but then it could have been two males fighting for that female, right? If it was a female. Who knows? Ecology at its best.
As a general rule, with the males fighting, the size is important, but also intact pincers. If one is missing a pincer or it's got a broken pincer, regardless of size, it's usually going to lose.
Oh, and we did observe that. You know, one of them definitely had broken pincers, so... Thank you, Mary. And yeah, Tina is agreeing. She says apparently the female is in the nest.
What's this darkened color in here? Kind of
That
purplish.
that looks. We'll have to see what that is. That darkened material in it. Some of it looks purple. Could be the purple stoloniferan.
Yeah.
And then there's more living coral in the back. It looks like the yellow fans, possibly of Analopsamia, and some more of them still further in the distance.
You can come in. Copilot, you can turn the lasers
Well,
back
what
on.
is that? Yep, that is the stoloniferan.
But here we can see both of the species.
Yes. Yes, the pale one off right, front right, and the purple one. And then these white solitary corals look like Desmophyllum. Elongated like that. Many solitary corals are perfectly round, so this is a distinction that would narrow the field for identification.
Yes.
I think we have seen these before
And,
video, so we don't know if we need to
and it also looks like all of the dead branches are covered with those agglutinating foraminiferans.
Looks like that moves completely.
And
okay with another.
for Les and Eric and Daniel,
do you want to 25 meters?
I'd really like at some
30
point to talk about that with you, because I think these agglutinating foraminiferans may accelerate the deposition of sediment in the areas where they grow. I had an abstract. I actually went a little far afield from my regular area of scientific comfort zone and talked about that based on some of our work on the east coast of Florida on the deep Lophelia reefs.
This is not
And those agglutinating forams just covered all of the dead almost all of the dead coral.
It's out past you.
I don't know if anybody is doing any work on them at all.
Maybe a little bunch of things.
This dive just keeps giving and giving and giving. Now we're approaching yet another coral forest. We can see some yellow Analopsamia, as well as some other species looming out of the darkness
Come down for you a little bit.
in the distance.
Yeah, that's what we're seeing there. We're headed that way though, so yeah, get there. Take a peek off to starboard. Yeah, we are getting some returns over there too. Yeah. Video, can we snap zoom on this? I don't know if we've seen this. It seems like a stalk kind of sticking out.
Okay, thank you. Good eye.
Oh, it's a. Okay.
That is a dead stalk with the stoloniferans on it again.
Okay.
Very deceptive. And hydroids up at the top, I think. More dead mussel shells. Yes? No, or is that carbonate? That's carbonate. And it looks like some plant material there. Not sure. No, is that a claw, a dead claw? I can't quite tell. See that curled? Oh no, it's a galatheid. It's alive. Well, okay, there he is. Okay, that's a galatheid.
Okay, video, come wide when you're clear.
Fooled me.
It's probably a great big male.
And with that heavy claw, yes, heavy cheleped.
Oh yeah.
Nope.
Alright, so my sonar, that is kind of uphill that way. Okay. It's an option.
So we have just under 10 minutes left in this dive, so I think we're probably going to end up ending the dive in this area, which we're coming to, which is just
nice.
Another incredible coral community.
Yeah, it looks like a mound of dead branches.
With lots of the little desmophyllum cup
right,
corals.
all over it. And then on the left of center is a dead colony, probably covered by that pale stoloniferan.
That looks a bit different, though.
But the one in the middle, now leaning a bit right, looks a bit different, but we can't tell from here.
I'll get you a
We
closer
can
look.
zoom in on that.
I believe you're talking about the one lighter in color.
Yes.
Yeah, more of a cleaner look. You come in. I think right here.
So what have we got here? So, a lot of desmophyllum. And an octocoral of some sort?
This is so beautiful.
Yeah, what is that?
Those cup corals are just perfect.
Yes, but what is the white colony? I don't see
Maybe an
innate
acanthogorgid?
tentacles.
Yeah, no, they're definitely octocorals.
Oh, yes,
Like
now I
a-
can
yeah.
see. Yes, it's an octocoral. And sort of a maroon gray anemone there.
But again,
Unidopsis.
this is a new species of coral that we haven't seen for this dive
No.
yet.
No. It's not a corallium, is it? I don't know them that well in all of their...
Max zoom video?
Yes.
Copy.
Oh, Tina Molotova is now chiming in and saying it might be Anthothella, which is known from
Rotate
the
you down
sites
video.
north of here.
That's okay with you?
Okay.
Or Asako's
See if
actually
we can
saying it
kind of
could
scope
be...
around a
Oh
little
yeah,
bit.
so from the family, Anthotheridae. So they're both Tina and Asako are coming to the consensus that, yeah, it could be Anthothella, which...
And you can see there's a whole little community here. We've got a galatheid up left. We've got desmophyllum solitary corals. There's still some living coral here. I can't tell whether that's Selena smilia, perhaps. The octocoral, which is the fuzzy white. There's another crustacean center. There was a small shrimp. It's really, and there's that brownish purple anemone above center right. Just, you know, on and on and on.
Maybe you can zoom out a little bit and see if you notice anything
There's
else
baby
here.
Pilonella, and there are at least two of them in there.
Say again, sorry?
It's the genus Bathypalamonella. You have a large
Bathypalamonella,
one
yes.
and a small one.
The small shrimp, Bathypalamonella, according to Mary. Thank you.
So...
Video. Clear when you're clear.
This is the most desmophyllum we've seen, certainly. Look at that.
ROV nav bridge, the ship is stopped.
This isn't the same Unidopsis you saw
Copy, bridge.
deeper.
Come wide video.
I am.
Oh, okay. Sorry, just couldn't see my toes. I was rotated up a little bit. Got what, 5 minutes left? Not even?
Yes, about 4 minutes left.
4 minutes?
Yep.
Copy.
Pilot, would it be possible to set down in front without crushing any, without, I shouldn't say it that way. That thing that we just zoomed in on that was called Anthozoans, can you reach that without destroying the front of this construction?
I think so.
Brilliant.
Willing to give it a try for you.
Thank you very much.
So if we do want to collect then, if that's the case, some of the white bushy stuff, please.
Copy. Going in now.
This will be our last activity here. I'm going to be pushing it for time.
I suppose if we were just heartless, we would just say, well, we're just accelerating a natural process, but we don't want to do that.
We don't work that way.
No.
And this community could be very old, and it's very diverse and also very abundant in this area, but very fragile. A lot of these stony corals, as I said before, are like china, bone china, and if once they're touched, they'll sort of shatter. So we really want to cause as little harm as possible to this community.
Inner is good.
So we're well off. We're sitting well off the community on the sediment and broken dead branches in front of it and reaching out. So we may be able to get some of the desmophyllum as well.
Okay, video coming on Zeus. In HD here. And coming on Mini Zeus as well.
It's hard to get a frame of reference there. But
Watch, did you say it's going to be brittle or malleable or?
The one that the fuzzy stuff in front of your claw is what we want.
Okay.
And that should be that's a gorgonian, an octocoral, so that'll be flexible.
Okay.
The cup corals will be rigid. We'll just see what it's growing on and just grab a chunk of that.
Does this look about?
Yeah, that should be fine. See what we get. That does not look like very much of it. Yep.
Okay, so it looks like it's growing on a harder subject.
Yes, it will be.
So if we just trim some of these off the top here, might be what we need to do here.
I think your fingers still might close on that hard piece though, so it won't let you cut unless you go way up top there.
Okay. Watch, does this look like a sufficient sample?
Believe yes, that's fine. That's plenty.
Okay. I'll just gently try to break it off here so it doesn't pull through.
Okay. Starboard.
Oh, we've got a desmophyllum attached, and who knows what else is in that. A little branch broke away. That's okay. That looks like maybe a dead desmophyllum, which is good.
Video
it's okay.
Zoom on Zeus. We'll do a quick zoom here and we'll get in the box, then it'll be end of the dive.
That's fine.
All right, video come wide. Let's get in the box here. I'll just take off real quick, Chris.
Sounds good. I'm actually going to bring your swing arms in anyway.
Yeah. You could bring it out.
Copy.
Starboard.
Extending.
I'm kind of drifting down. Hold on. Adjusting my depth set point up.
Yeah, I don't know if that's a live one or a dead one. We'll have to see.
I'm drifting to port.
You're okay.
Copy.
Gonna come up with you.
And as we're drifting off the seafloor, I mean, just take in this view. We've got bright yellow Enelopsamia colonies, as well as Madrepora potentially. Huge colonies off in the distance. I mean, these are over a meter across, sometimes 2 meters across in some places. These are very old, very fragile, very beautiful, large colonies of deep sea coral.
Nice. And the diversity associated with them was just wonderful. We had the multiple species of galatheads, shrimp, anemones, stoloniferan octocorals, the octocoral we just collected, the agglutinating foraminiferans, and if we get any dead branch in the sample, there may be additional things, small things, whether they're amphipods or worms in tubes or barnacles or hydroids. It's really, there's still a great deal to be learned about just the basic biodiversity, what is out there in these habitats.
Right. Yeah. Which makes yours 0, which makes me get a turn negative, so I got to turn, we both turn right.
Okay.
I'll get
ready for that now?
Negative, I'm going to get underneath you. Let's get a 20. Here, I'll get in auto depth. You bring your rotator to 090.
090.
I'll get underneath you.
Copy.
And then we'll do a turn.
Is it stowed?
Copy that.
I'm not sure.
Yep. Okay, pilots, thank you so much for those amazing collections, that amazing piloting.
Right,
that was a phenomenal dive.
Right.
Absolutely phenomenal dive. So scientists ashore, thank you so much for sticking with us throughout this dive. It has been a best one so far.
Extraordinary.
Yeah, for me at least. Like, I thought this dive was just amazing. So we're going to have our post-dive call in about 8 minutes at 4 Eastern Standard Time. So please do join us then to talk about this dive, reminisce how amazing it was, and then move on to tomorrow's dive.
Can we bring your rotator down a little bit?
So chat soon. Thank you everyone again, and thank you everyone who's been watching from your homes, your offices, wherever. Thank you for following along.
And thank you very much for calling in, Mary, Carolyn, and the rest of you.
Looking good.
Pushing out.
Okay.
Mind taking my auto depth out.
Copy.
I'm still maintaining forward way.
Yeah, I've got you in.
Coming up easy.
Or you have me in your tether cam.
Yeah, that's right. Tight, pushing out.
Copy. Delta 21. Looks like we've got a pretty good offset on high pack, but you're working your way there.
Alright, I'm spooling up I'm coming up here.
Copy that. You want me to come up with you?
Yes, once I get like delta of like 12.
Okay, let's get you in my tether cam before we go up full speed.
Go ahead, Bridge.
Okay.
Are the vehicles off bottom?
Yes, we just came off bottom.
Roger that, thanks.
We really don't. We don't need to call up till we're done getting set up here. So.
Alright,